the session.
Thank you very much, Pragya, and it's an honor to be a part of this very informative and well-needed course. And I am so lucky, and I'm so feel so like a opportunity for me to learn, because earlier in my PhD days, I was also connected with this kind of research area. So I'm very pleased to be the part. And I really want to congratulate Professor Tandon for this course and for organization of this course. And it's my pleasure to introduce two of my speakers. To start with, of course, Dr. Anant Narayan Bhatt. He is going to present his work in the session 3, first lecture. And he's a colleague, and he's also a friend, good one. And then he is currently working in Institute of Nuclear Medicine and Allied Sciences as senior scientist. His research interests are radiation biology, cell biology, cancer biology, and then cancer biomarkers and protein folding. Again, a lot of molecular biology along with the cell biology correlation, how the radiation can impact the cellular function and how we can counteract it. So it is again a very upcoming and very challenging field too. And his qualification, like, no need to give any emphasis on, but he is very well known in the field. He has done his PhD from CDRI Lucknow. He was a researcher, EMBO fellow in University of Zurich. Then he also went to Johns Hopkins for Fulbright fellowship. And he was visiting faculty at Department of Zoology, University of Delhi, but at present he is in Mass, as I said. He has published more than 47 papers, so many chapters. So that itself talks about his good science and how in-depth science he is involved too. He was working with Dr. Dwarkanath also in many of his research work. So there is always you will see in both the talks there is always some coherence, and maybe we will learn more. And just to tell that he was been also awarded by many awards from DRDO itself, and also by in Mass. And I'm really looking forward to and our participants are also going to be benefited by his experience and by his research work. So Dr. Anand, please start your presentation, and we all are looking forward to.
Thank you very much, ma'am, for nice introduction. And as you rightly mentioned that I started, basically I also learned radiation biology at Mass only. Like as you said. So basically, I mean, Dwarkanath sir is my mentor also, as far as radiation biology and metabolic cell signaling is concerned. Yes. So we'll be talking more today about radiation biology. What Vibha ma'am has given the topic is cellular effect of radiation and clinical radiation biology. So we'll be discussing more about this today. So is my slide in presentation mode?
Yes, sir.
Okay, yeah. Okay. So we'll be talking mainly on these. This is the topic which I have to cover. Overview of interaction of radiation with the biological system, molecules to biosphere, and then chemical basis of biological effects of radiation, how basically it interacts with cell, and then when after interacting with the cell, what kind of damage it causes to the cellular system, and which leads to cell death, as well as then different kind of complications in the human body or any biological specimen. Then factors influencing biological effects of radiation, and radiobiological cell death and dose response, that will be covered by Dwarkanath sir. We discussed together. As I mean, he'll cover DNA damage and repair, and then after that he'll be talking about cell death. So I think this suits more after that. So we discuss, and then Dwarkanath sir will cover this in his lecture. So So yes, since yesterday, you all would have, gone through this, that what are the basic source of radiation, how the radiation exposure can happen. So I just want to tell one more thing, that if anybody wants to ask any question, they can stop in between, and then they can ask the question, if suppose that somebody wants to know or have more queries immediately, suppose that. So on the left side, you can see that these are the planned exposures which can happen. Medical imaging is one thing which Dr. Rath talked about elaborately. Then rescue operations are, suppose that some accident happened at some nuclear reactors or any places, or suppose that if some nuclear bombing is happening, then the people who will go for rescue operations, they are also, they know that they are going to get exposed to the radiation. So that's also comes in planned exposure. Then space flights, when people go, heavy ion exposure occurs to the people, and some studies has happened that they face neurocognition problem later on in their life. That is a chronic exposure happens, and then they show some problems there. So these are the planned exposure. And then we already know that the background radiation, which is already coming. Suppose that if the people or the cities which are close to uranium mines, or you must be knowing that in Kerala, the background radiation is relatively higher, because there is a lot of thorium is there on the beaches. So they are kind of the source of background radiation. And radon is already coming from, a lot of concretes and all. The basic radon keeps coming. You must have seen, if suppose that if you are visiting on YouTube and studying about radiation, there is a video which is a very, very informative video. If you will go and see, then they compare everything as taking banana as a unit, because banana has K35, which is a radioactive material, and potassium 35, and then that they compare, like if you are getting exposed to X-ray, how many banana kind of, suppose that 20 bananas you will be exposed to radiation. That kind of, he compares everything with banana, taking banana as a unit of radiation. So that is a very informative video. I think you all must go through. So you will have the idea that how much radiation is where, and then how people get exposed. Then there are unplanned exposures, like terrorist attack is one unplanned exposure. Then reactor accidents, strategic explosions. You must be hearing that Pokhran kind of thing which happened in India. And then you must be hearing every day that Ukraine and Russia war, it may turn anytime into a nuclear thing. So that kind of things are that can be unplanned exposure, and then it may lead to devastation. So basically, these are the basic source when radiation exposure can happen. And this is a basic understanding that what basically happens. This is what you are seeing on the screen. That's called mushroom cloud. And once anywhere bombing happens, this kind of cloud will form, which is called mushroom cloud. And like it is not only going to be limited up to very in a closed area, let's say few kilometers or few tens of kilometers or hundreds of kilometers, but it can go up to very far. You can see this is a predictive modeling of Fukushima, which happened. This nuclear accident happened in Fukushima, and it reached up to the west coast of, it crossed the complete, I think, Pacific Ocean, and then it reached up to the west coast of America. You can see in 10 days, this nuclear cloud, whatever the radioactivity was there, that reached up to this place of west coast. So it was estimated that Chernobyl tragedy put 400 times more radioactive material in the environment than Hiroshima blast. So these are the basic incidences which can expose to radiation, which can put a lot of radioactivity in the environment, and that can be responsible for radiation exposure. There was another recent evidence which happened in basically in Europe. People have seen that, suddenly some detectors which were placed in the environment in Norway, they found that some radioactivity is there. So then the study was conducted and they found that you can see the complete radioactivity cloud on the entire Europe. And they found the nucleus it was detected in somewhere in 2017. And they found that basically the cloud, the nucleus was basically in the Europe somewhere. It was in Russia. And they detected that it was ruthenium 106, which is a artificial radioactive material. It is not a natural radioactive material. So that means it was being produced somewhere in the reactor only, and it got released probably from there. So this news came. It was not a very popular news, but this news came, and then people detected, but it was not in the alarming level, but they have very sensitive reactors which can identify at very low level, and then they informed to the authorities that it should not happen. That the radioactivity leak should happen. So these are the incidences. We never know in and around region that probably somebody can accidentally get exposed to the radiation. So how basically once this exposure happens, how basically this exposure can happen, and then the person gets affected. So on the top, you can see, like, if the source is kept somewhere else and you are passing by or you are standing there, then rays may come and then you may get affected. So you will not carry the radioactive material with you, but the source is kept at a distance and you will get exposed by standing there. How much time you spend, that will going to affect. I said, as you have seen that the cloud which forms, because of explosion or anything, the cloud which forms, that may also put a lot of radioactive materials, in the environment. When you are breathing in that kind of environment, you may take up the radioactive materials inhale it. Right, or it may come on the skin and some itching is happening, or you're scratching, then it may get internalized through your skin. So that kind of internalization of these radioactive materials may happen. External exposure and internal exposure as well. So that may go and get incorporated. Cesium 137 gets incorporated. There are so many radioactive material which gets incorporated into your body if it is not properly decontamination or decarburation is not done properly, then it may get incorporated into your body. And the half-life of some of these material is very high, so they will be continuously emitting radiation from wherever they got incorporated, and the person may get unless it gets converted. And if it is insufficient amount, it got incorporated in the body, then it will keep damaging the cells nearby. So coming to the basic, once it gets, it got incorporated, or you got exposed from a source kept at distance, which is basically called teletherapy. Like in case of what Dr. Rath was telling yesterday, that radiotherapy machines. So that is called teletherapy if you are not getting exposed to brachytherapy or that kind of source. So the source will be at distance. In both the cases, radiation will pass through your body, and it may cause two different kind of effects. So on the left side, you can see the direct effect. So direct effect is what? Like when the radiation will pass through your biological material, it will deposit the energy directly to the DNA, to the biological material, other macromolecules like protein, lipid, and so many other things. So it will deposit the energy. Because of that high energy, the bonds are basically energy. So if they will get, at some bond, the energy deposited, is more, then it may break. So so the bond will break, and then the fragmentation of the macromolecule may happen. So that is on the left side, you can see, and because of that, it may lead to cell death. On the right side, you all know that our different cells, different type of cells are made up of 70 to 80% of water, right? So because of that, because radiation, what we are talking predominantly is ionizing radiation. So ionizing radiation has that much energy that it may cause the radiolysis of water. And because of that radiolysis of water, free radicals and ions will be created. And those free radicals, they will have, they can oxidize the neighboring DNA, RNA, protein, lipid, and so many other different kind of macromolecules. And because of those oxidation of those molecules, inside intracellular environment is always reducing. So if it is not in the reduced condition, then probably it will not be able to perform up to its
Did we lose him?
kind of which poses different kind of stress in the cells and lead to finally cell death. And there are chances of repair also. If the damage is relatively less, then the cell may get repaired also. Macromolecules may get repaired and ultimately leading to the survival of the cell. But sometimes suppose that if these kind of mutations may persist and cell survives, then that kind of cell may lead to a cancerous cell death. And that is what called basically the radiation-induced carcinogenesis. So suppose that if some cell survived and because of some mutations which happened at a place where proliferation got induced, or because of the damage, mutation occurred and it in the environment it survived in some way, which may lead to the high rate of proliferation, that may lead to tumorigenesis, and that is called radiation-induced carcinogenesis. So that is one of the delayed effects. At very low dose, many people who survives after radiation exposure, the chances is very high that they may have a delayed radiation-induced carcinogenesis. So this is a cell. When cell got exposed to radiation, you can see like it will first pass through the lipid membrane. The energy will pass through the lipid membrane. So lipid oxidation may take place along with the radiolysis of water, indirect effect. Then protein molecules may get exposed. DNA molecules may also get exposed. So these are the macromolecules, biomacromolecules, which are susceptible to radiation, and they may get damaged because of radiation exposure. So this is a very classical example of direct effect of radiation. So, chromosome will get damaged, and after the damage, what will happen? A repair will happen to this. So sometimes when repair happens, what happens like, some fragment got detached, and this kind of chromosome, because there are so much chaos after radiation exposure in the cell, the machinery also get completely, because the machinery is basically what? Predominantly proteins, right? Repair proteins. So they also got damaged. Sometimes some protein enzymes are not there, or sometimes if they are there, they are damaged. So they are not able to perform its function properly. So what will happen? There are a lot of chaos, and some other chromosome may get attached with some other chromosome. So what will happen? Some dicentrics, these arrows you can see on the right top image, these arrows you can see, these are dicentric chromosomes. So dicentric chromosome will form acentric chromosome. The some small piece of DNA which left out during this repair, double strand break repair. So that will, what will happen? That will left out. So it will form acentric, and that may form actually because centromere is responsible for pulling them during the cell division. So if that doesn't have centromere, what will happen? It will left out and it will form a micronuclei. and the cells which will have micronuclei, they will die eventually. So biodosimetry will cover this completely, because it depends completely on the dose of the radiation, that how many dicentrics will form, and based on that biodosimetry is being done. Suppose that if somebody got exposed to radiation, you take the blood of that person and you check the chromosome of that person. That can give you idea that a person got exposed to how much radiation. So that is called radiation biodosimetry, to identify that the person got exposed to how much radiation, and then his or her, the person's clinical management is completely depend on that, because the person will be, whether what kind of management he needs, that completely depends. So the lower panel you can see, that is a curve which talks about the double strand break, that how much dicentric and different kind of breaks or different kind of damage happen to the DNA because of the direct effect of radiation, or indirect also sometimes it happens, and then that leads to the formation of this curve, which can give you the estimate that a person got exposed to how much amount of radiation. So in like in the cell, it is not only the radiolysis of water due to radiation or some other energy that is responsible for ROS. Even the respiration, mitochondrial respiration, is also one of the biggest source. I mean, beside radiation, if somebody is not got exposed to radiation, there so many drug and so many other environmental stressors, they affect the mitochondria directly, or outside the mitochondria also. They basically, these drugs and other things leads to environmental stresses. They lead to the production of reactive oxygen species, which is the indirect effect of radiation also. So that may lead to lipid peroxidation, DNA oxidation, and then protein oxidation, which is a complex process. And that entire thing leads to the macromolecular damage in the cell, and the cell may opt for death. So basically, chemical and a lot of chemical and natural stresses may have this oxidative damage to the cells, and ROS is not always, ROS is not always seen as a negative thing. Sometimes in a very minimal, what you call like a pro-oxidant effect of a lot of these antioxidants which we eat, they have pro-oxidant effect. So sometimes they are helpful also, like NRF2 expression, which we will discuss in protein oxidation in detail. The expression of NRF2, which is an antioxidant, cellular own antioxidant machinery, the activation of NRF2 itself depend on the generation of reactive oxygen species. So that kind of regulation ROS level, then it creates a problem. So it is being generated continuously during the process of respiration. We know that, and it has the potential to cause oxidative deterioration of macromolecules, which we already have seen. So radiation-induced mitochondrial DNA damage, suppose that if radiation, the cell got exposed, what will happen? The most susceptible thing, which is, mitochondrial DNA, it doesn't have very good repair machinery. So the chances, and that is also, it is said that which is like a lot of accumulation of mitochondrial DNA, mutations in mitochondrial DNA, that leads to a problem, and that is basically responsible for a lot of diseases. Now, you track in majority of lifestyle related disorders and a lot of other diseases like cancer itself, then neurodegeneration disorders and all. If you will track them, then you will find that mitochondrial alterations are one of the main responsible for that. And why this is responsible? Because the mitochondrial DNA is very susceptible to mutations. So once the mutation will happen to mitochondrial DNA, that will lead to altered, there is a metabolism relationship between the cell and mitochondria, you know, because we learned in evolution that mitochondria came from outside. So it has to have maintained that relationship. If that relationship is not maintained, what problem will happen? That the mitochondria will be having a damaged performance, and then it will be taken over by the cellular milieu, the metabolism of the cellular milieu, and then this cell will not have the normal functioning. And if it will not have the normal functioning, then cancer or any other different kind of diseases may occur. So it is predominantly because of the mitochondrial DNA is not performing better. So respiratory functions is not happening properly. And that is responsible for a lot of reactive oxygen species production or leakage of the reactive oxygen species from mitochondria, leading to the disturbed ROS maintenance system in the cell. So what are different kind of system is there in the cell which is responsible for taking care of this ROS? That is the spontaneous decay. I mean, once the radical will form, reactive oxygen species will form, what will happen? It will get reduced by transferring its oxidation state to the neighboring macromolecules. Like, as I said, like any macromolecule, if it is present there, the reactive oxygen species will immediately oxidize that, and it will transfer its oxidation state there only. Then you all must be knowing that there are enzymes, superoxide dismutase enzymes are there. So they can also reduce the free radicals and convert it into H2O2 superoxide radical. SODs, they basically convert the superoxide radical into H2O2. So H2O2 is less, less toxic reactive oxygen species. Not, radical is not, it is not that harmful, and it has, its limit is relatively less. So it gets converted into cytoplasm. SODs are there in the mitochondria also and cytoplasm also, and it converts, superoxide radical is predominantly produced into the mitochondria, and there MnSOD is there which converts it into H2O2. So H2O2 will not be that much harmful. So then glutathione is there. Glutathione converts the hydroxyl radical. It combines with the GSH, and it gets converted into water. So water is like completely, it is harmless. So then catalase is there, which can convert the H2O2 produced by SOD into water and oxygen. So these are the systems, enzymatic systems which are present to take care of the free radical in the system. But as I told you in the beginning only, many of these are enzymes, and if radiation damages the enzyme itself, what will happen? This system will not be there, and then a chaotic system will be there. A chaos will be created after radiation exposure in the cell. So suppose that if that kind of system is there, then probably antioxidants like endogenous antioxidants like vitamin E, A, C, and beta carotene, that can take care of the free radicals generated, and that can minimize the indirect effect of radiation which is caused. So what could be the different targets of these? We have discussed already, and what could be the once they will be damaged, then what could be the kind of damage and what could be the consequence? So if DNA RNA gets oxidized, then what will happen? Scission of deoxyribose ring may take place, base damage may take place, and a strand break may take place. So because of these different regions, what will be the consequences? Mutations, translational errors will happen, inhibition of protein will happen, and protein synthesis may also happen, right? So because, like if mutation will happen, then probably because of the mutation, termination may happen. A stop codon may get introduced there. So that may happen. Or mutation may happen in such a way that probably, there is transfer RNA is not there. That kind of premature, termination of the chain may happen, synthesis chain may happen. Then in case of protein, you already, if suppose that some damage happened, or some oxidation of the side chain of protein, which we'll study in detail, that happens, then what will happen? It may lead to the formation of aggregation of the proteins. So that may happen, and then what will happen, like suppose that two different kind of molecule, let's say some transporter protein is there, and that forms adducts with lipid, because transporter protein will be in the lipid membrane. So because of oxidation, that may form a adduct, and that we will study in detail. So what will happen? It will not, it will not be able to execute its function properly. Like that is its function is like, let's say, ion transport, or some important molecular transport, like let's say glucose or any other things, for which it is responsible. These transmembrane proteins are responsible. So or the transport of the some of the message may also happen, right? The primary, receptors which transfer the message to the intracellular milieu. So if they got damaged, then the transport of the message may also not happen. So there are so many problems. But sometimes what happens, like these oxidation may be useful, as I said, in case of NRF2. You know, NRF2 is a transcription factor, and it is always inhibited by a protein called KEAP1. So KEAP1 has cysteine, several cysteine in its structure. What happens, like in reactive oxygen species may reduce it to cysteine, and once it will be reduced, it will release the oxidize it. Reactive oxygen species will oxidize the cysteine to cysteine, then it will release the NRF2, and then KEAP1 will be degraded, and NRF2 will go and bind to the nucleus, to the DNA in the nucleus, and it will induce the expression of antioxidant proteins. So that is a regulatory mechanism of reactive oxygen species. So there it is a beneficial, reactive oxygen species is beneficial. So that is one example. Then polyunsaturated lipid. So these lipids also, we will study in detail that how it may lead to unsaturation of lipids. So because of that, fluidity will be altered, and then it will lead to the formation of adducts, which will a small molecule of lipids, which is considered as as a marker also to study the lipid damage. They are called MD and 4-HNE. People suppose that if somebody is studying the radiation effect of radiation in whole animal or in the cellular level, they measure MD and HNE because this is the byproduct of the radiation-induced lipid damage. So this leads to altered lipid fluidity, altered membrane permeability, and then effects of membrane-bound enzymes. As I said that it may, if it forms adducts with the transmembrane proteins, then it may create a problem. So, unlike DNA, which we studied, it is believed that major DNA damage happens because of the direct effect of radiation. Indirect effect also cause, DNA oxidation also happens, but there are a lot of array of proteins are there, which DNA repair enzymes, which takes care of the DNA repair. And if that is not taken care, then only it leads to a problem. But predominant damage to lipid and protein is mainly due to indirect effect of radiation. And suppose that whatever the direct effect, the protein and lipid damage is happening because of the direct effect of radiation, you will not be able to identify. Let's say if you found some protein, like I took the example of NRF2. Let's say if it is a 90 kD protein, and you found because of direct effect of radiation, it got fragmented in a 30 kD and 60 kD fragments, then you don't know this happened because of the radiation or it happened because of the proteolytic enzymes. So it is very difficult to identify that the protein got damaged or lipid got damaged because of the direct effect of radiation or not. Or there are some other enzymes which are basically responsible for cleaving continuously the proteins and then making the homeostatic balance of the old proteins they damage, they make it free amino acids available for new protein synthesis. So but that machinery is continuously going on. So you don't know that they are byproduct of direct effect or direct effect of radiation, or because of the machinery, whatever machine homeostasis going in the cellular machinery. So it is believed that predominant damage which is happening is because of indirect effects of radiation in the lipid and protein. So radiation-induced lipid damage, people have seen that there is a correlation. People they have a lot of study have been done earlier, and they showed that damage to membrane is an initial step of triggering of cell death. Because like if the permeability is altered, then what will happen? Lot of extracellular material like calcium and so many other things will enter into the cell, and that may cause a necrosis and different kind of cell death, or some metal ions, fluidity will be changed, permeability will be changed. So a lot of damage may happen, and like the damage to the membrane may cause this kind of thing. So people have observed a correlation between the loss of clonogenicity and membrane oxidation, breakdown of nuclear membrane and chromosomal condensation, and damage to the organization of mitotic spindle. So they have seen that when these are involving both protein and lipid damage, and because of oxidative stress, and this leads to the this has correlation with clonogenicity. Dwarkanath sir will talk in detail about when these damages happen, how it leads to the clonogenicity, which is assessed by macrocolony assay. So lipid peroxidation you can understand in these three steps. Spontaneous, how it is defined, that spontaneous oxidation of lipid molecule in membrane by oxygen at room temperature is termed as lipid peroxidation. So it happens naturally also, because environment has, if suppose that which is called, lipid kept outside, but if you keep butter outside in the at room temperature, that also get oxidized, and that's called rancidification and all, right? So that leads to different kind of oxidation. But inside the cell, because of radiation or the naturally occurring reactive oxygen species, the lipid also gets oxidized, and it can be it can be classified in three different steps. That is called the initiation of lipid peroxidation or lipid oxidation, then propagation of that. It is basically a chain reaction process. So propagation happens, and then it gets terminated also at some point. So we will study in these three different forms. So how basically initiation happens? So water is getting, like after ionizing radiation, water get hydrolyzed, and because of this hydrolysis leads to the formation of different kind of radicals. You can see on the right side, hydroxyl radical, then proton ion, and then superoxide radical, then H2O2 and H2. So these different things are responsible mainly for damage to the lipid. So how it is defined, that the removal of hydrogen atom homolytically from a methylene carbon or an unsaturated fatty acid, which is denoted as LH, can initiate lipid peroxidation. So you can see here in this reaction that because of radiation, first a hydrogen ion is removed from there, and then it forms a double bond. So this basically further gets cleaved. It may form cyclic peroxide, cyclic endoperoxide, or hydroperoxide. So these different kind of lipid oxidation, they basically form in the lipid, and it is basically L dot, which you are seeing on the right side of the on the because of the hydroxyl radical interaction with the lipid, that is basically called lipid radical, and that is responsible for further initiating the chain reaction in the lipid. So any free radical which has a sufficient energy to abstract a hydrogen atom from methylene carbon of unsaturated fatty acid can initiate lipid peroxidation. So this is the basic requirement. The radical should have that much energy that it can abstract a hydrogen from the methylene carbon. Then comes, we have already shown the reaction with hydroxyl radical, how hydroxyl radical is basically responsible, and predominantly hydroxyl radical is responsible for causing lipid peroxidation. But like this radiolysis of water leads to the formation of other radicals also. So superoxide anions are there. So these superoxide anions, they themselves doesn't have that much energy, which can abstract a proton from the methylene carbon. But what will happen that these superoxide anions, not themselves, but in the protonated form, and like in cancer, we discussed cancer in detail yesterday. Cancer have relatively low pH because they have high metabolism and continuously a lot of acids are being produced. So at physiological pH, superoxide radical always remain in the form of superoxide radical. But what happens at low pH, like in case of cancer, in case of tumor, it will have relatively low pH, and their superoxide radical may form it may lead to a protonated form. And once that protonated form will react with lipid, that is denoted as LH, you can see in the first reaction, it will again lead to a lipid radical, and it will form the H2O2, which is again a ROS only, and that that will be taken care by catalase. So at physiological pH, as I said, superoxide radical is always remain in the form of superoxide radical, not in the protonated form. But what will happen, like the cell has a lot of metals also. So this is a very well known Fenton-Haber-Weiss reaction in oxidative stress. What happens, like the iron which is present, free iron which is present in the form of ferric, it will react with superoxide radical and it will convert it into the ferrous, reduce it into the ferrous and O2. That then superoxide radical will react with proton and it will form H2O2, and then iron and ferrous iron and then H2O2 will convert again into the ferric form and hydroxyl radical, right? And as I said in the beginning, that majority of the initiation of the lipid oxidation happens predominantly responsible radical is hydroxyl radical. So superoxide radical doesn't causing lipid initiation of lipid oxidation on its own, but on reacting with iron, it may cause free hydroxyl radical, and that hydroxyl radical will lead to the formation of lipid peroxide, right? So indirectly it may lead to the formation of lipid peroxide through the Fenton, through the iron available in the cell. Then macrophages produce HOCl. HOCl is biologically produced inside the body in macrophages. There if oxygen superoxide radical reacts with HOCl, and that also leads to the formation of hydroxyl radical. So that hydroxyl radical may also lead to the damage of membrane damage or lipid damage. So these are the indirect way through which superoxide radical may lead to the formation of hydroxyl radical. And hydroxyl radical has that much energy that it can abstract the proton from methylene carbon and cause initiation of the lipid oxidation. Then RNS is also there, reactive nitrogen species. So those reacting nitrogen species will, a superoxide radical will react with those reactive nitrogen species, and it will lead to the formation of hydroxyl radical, and that hydroxyl radical is damaging for membrane. Then perferryl is also formed, and ferryl is also formed, which are responsible predominantly for lipid oxidation. So these are alternate way through which the initiation of the lipid oxidation may happen. Then once it got initiated and it forms a L radical, that is lipid radical, it will, the more oxygen availability will lead to the propagation of these lipid peroxidation, this lipid damage, and it will cause peroxyl radical, and that will then, this chain reaction will go on, and it will continuously propagate, and it will lead to the formation of more and more lipid radical. So oxygen availability is the primary determinant here, and that is why the cells and the tumor cells, like they are deprived of oxygen, they are hypoxic. So that is why, because of radiation, radiation, those tumors are basically resistant to radiation because oxygen availability is less. So the damage caused, basically we are giving radiation to kill those tumor cells. But because hypoxia is there, oxygen is not there, so that doesn't lead to the formation of too much, it doesn't lead to the propagation of lipid radical. And those tumor, there are several other reasons also, but this is also one of the reasons why the tumor become resistant to radiation, because they are hypoxic. Then this I mean, iron is also responsible for propagation, not only for initiation. Iron is also responsible for propagation. So some of these ferric and ferrous, it reacts and then it propagates the peroxyl radical into the lipid. Then how the termination happens? Like it got propagated, a lot of lipid got oxidized, and then these lipid radicals, they react with each other and they form the chain cross-link, and then this reaction get terminated. Peroxyl radical formation happens, and then this gets terminated. And you know, like once this termination happens, it is not like the problem is solved. These chains, this cross-link will form, and you know, the cross-link, the formation of cross-link will lead to the altered fluidity. Fluidity of the membrane, and then the permeability and so many things will be compromised, and because of that it will have impact on the cell. So these are some biological examples, you know, like here you can see that because of the formation of this direct or indirect effect of radiation, a small molecule of lipid is formed. And you know, lipid is already this small MDA or 4-HNE which are formed, they are already known. They have the capability of penetrating any membrane, you know. And once they will form, let's say the ER, endoplasmic reticulum, which is full of calcium, that itself is a membranous structure. So if they will pierce it, they may lead to the formation of ionophores, and that it will form a channel, and because of that the calcium leakage may happen. So there are articles which already suggest that these lipids, fragmented lipids, may pierce through endoplasmic reticulum, and that may lead to the calcium is it is an energy dependent process. Suppose that if it is stored in the endoplasmic reticulum, it cannot come out of the endoplasmic reticulum automatically. So these kind of oxidative stress may lead to the altered fluidity or formation of a pore in the endoplasmic reticulum, and due to which the calcium will come out. And once the calcium will come out, you know, like the extra calcium in the cytoplasm is not beneficial. I mean, it may cause different kind of damage. Mitochondria has to get damaged. And this is some results from our own lab. You can see here non-malignant cell and malignant cell. They have so many these white dots, which you can see they are the damaged mitochondria because of the high accumulation of the calcium. So this kind of things can be seen in the lower panel. You can see that these are exposed to ionizing radiation, and because after exposure to ionizing radiation, the formation probably would have gone significantly high, oxidative stress would have gone significantly high, and there is a lot of mitochondrial damage and a lot of alteration of calcium homeostasis. This is again, I have taken some images from that study. You can see the top panel is several malignant cells, and the malignant cell on its own, even when you don't give them any stress, you can see a lot of these bodies are there. These small dots are there. They are basically damaged mitochondria at any given time in the cancer cells. And you can see in all different kind of cell lines, you will see that these bodies. When you give them stress, let's say we have challenged them with adipocyte, these number of bodies go significantly high, which is suggestive of this formation of probably these ionophores. It leads to the very high amount of mitochondrial damage. In the lower panel, you can see these are the non-malignantly transformed cells. I mean, they are not cancerous cells. And in the control, you will not see any body in these cells. But when you will challenge them with the drug, which is etoposide or radiation also, with radiation also we have seen these formation of these bodies happen. So this is a example how this lipid oxidation happens, and probably that leads to the formation of these pores, and that lead to the altered calcium homeostasis, and damaged mitochondria will be formed in the cell. And this will lead, I mean, you know, like mitochondria is basically responsible. If that itself will get damaged, it may lead to the intrinsic cell death. So that will lead to the cell death. So how then basically lipid damage, lipid oxidation may lead to the formation of protein. I mean, make a cross-link with protein. You can see here, this is a amino group of protein, and then malone dialdehyde will be formed. MDA and 4-HNE, as I said, will form as a byproduct. So once that will form, it will form a protein cross-link with amino group. So amino groups are already, you know, like the positively charged amino acids, they have free amino group in their side chains. So that may form, like arginine or lysine may form a protein cross-link, and that will lead to a permanent damage. And this kind of damage will happen predominantly where, let's say, in the transmembrane proteins, in receptor proteins, which are membrane-bound proteins. So this kind of formation, they are more, those things, I mean, those proteins are more prone to happen there. And once it will happen there, what will happen? The function of the protein of the plasma membrane and function of the protein both will get compromised. And because of that, the overall function of the cell will get compromised. So what could be the biological consequences? We already studied in detail. We have taken the example, but slowly one by one we will go through it. Like lipid peroxidation is highly destructive process and alters the structure and function of cellular membranes. Then peroxidation brings about change in the structure, fluidity, and permeability of membrane. Then it inactivates a number of membrane-bound enzymes and protein receptors, induces swelling in mitochondria and alterations in respiratory functions, causes loss of sulfhydryl group from membrane-bound proteins, and then it mediates DNA damage, alters RNA transport from nucleus to cytoplasm, and carcinogenesis is also related to lipid peroxidation. So one is a very popular method how to identify lipid peroxidation in the cell or tissue. So TBA is a very well-known method to study the effect of radiation or any reactive oxygen species mediated lipid peroxidation. One unanswered question remains in this radiation-induced lipid oxidation is, it is not answered till date that whether membrane disintegration is cause or effect of cell death. I mean, people see that when cell death is happening, the membrane disintegration is started taking place. So people believe that it is effect of cell death. But many a times what happens, like it may be a cause also. Membrane disintegration is happening, and that is why the cell death is happening. So we don't know. I mean, it is the similar kind of question that whether the chicken came first or the egg came first. So then we are coming to protein oxidation now. So what is protein oxidation? Protein oxidation is basically covalent modification of a protein induced by ROS, intermediates, or byproduct. We have already seen that, I mean, how the lipid byproducts can form covalent modification of the protein. So that also leads to the formation of this. We have already studied in detail what are different kind of reactive oxygen species which can basically lead to the formation of the lipid oxidation as well as protein oxidation. So these are different amino acids, you know, like in a protein, the backbone of the protein, which is like the peptide bond, that is already, you know, like amino group is linked with another amino acid and the carboxyl, so they are not free. So what is basically free is the side chain of amino acids, and they predominantly get oxidized, and that leads to the oxidation of proteins. So sulfur oxidation may happen in case of cysteine and methionine, and then protein carbonyls formation happens in all the groups which have aldehyde and ketones. Then tyrosine crosslinks may happen. Chlorination, nitrogenation, and hydroxylation may happen in tyrosine. Tryptophenyl modification may happen. Hydroperoxide derivatives of aliphatic amino acids may happen, like as you've seen in any amino acid which have a carbon chain, so methylene carbon. Then chloramines and deamination, then amino acid interconversion may happen, histidine to aspargine, and proline to hydroxyproline is a known, regulatory mechanism. You know, like HIF1 alpha, which is a protein in oxidative stress, expressed in oxidative stress, hypoxia inducible factor. So that gets, normally what happens, its proline gets continuously hydroxylated, and because of that hydroxylation, what happens, it gets degraded continuously. So HIF1 alpha is continuously being produced in the cell and continuously degrades. But in the presence of oxidative stress, what happens, it's modifying enzyme, which is disulfide, a heme group containing enzyme. So that gets, heme gets oxidized there. So that cannot hydroxylate it into a hydroxyl proline. And then what will happen, HIF1 alpha will build in the cell, and that will cause the hypoxia sensor. It is a transcription factor, HIF1 alpha, HIF1 beta, and several other isomers of it. And that will go and bind to the nucleus, and it will send the mechanism, the message that the cells have a reactive oxygen species, oxidative stress, and that will induce the glycolysis and so many other enzymes pathway of the cellular machinery. Then amino acid oxidation, adducts formation may happen. Glyoxidation, adducts formation may happen, and crosslink is very common. The cells may form crosslink. They may form the peptide chain may form crosslink with other peptide chain, and that may lead to the aggregation also many a times. So basically here the message is that it may happen that this may be reversible, this may have irreversible also. So both kind of protein oxidation may happen. And how basically it happens, like reversible, what are the classified as reversible? You can see here protein disulfides, the formation of cysteine to cysteine, and then cysteine sulfonic acid formation, methionine sulfoxidation, S-glutathione, S-nitrosation. So these are reversible. Why they are reversible? Because cells already have the machinery, repair machinery, which can take it back. Through the glutathione or different proteins, we will discuss here that how it can take it back. It can reversibly convert it back. And why the cell have a machinery? Because majority of these modifications are responsible in regulating the function of the proteins. So that is why the cell has machinery for reversible modification, and sometimes over oxidation also of these amino acids. So that is why cells have the machinery, and that is why these are reversible modification. But there are some irreversible modification happens when the excess of oxidation happens. So that is cysteine sulfonic and sulfonic acid, trichlorination, dichlorination, tyrosine nitration, then dityrosine nitration, carbonylation. So these are basically irreversible modification, and what we study the irreversible modification only as an end parameter to see that how much protein oxidation is happening. And once these modification will happen, it will lead to the loss of protein function, degradation or accumulation. And you know, the entire management of protein oxidation oxidative stress in the cell is taken care by the protein only, because they are the basically enzymes responsible for it. So you can see here that this is a protein molecule. You can see there are a lot of how many possible sites can be which is exposed to the solution, and that may be susceptible for oxidation because it is entirely covered by water. And water is basically what? Which is getting hydrolyzed. So I have shown here a small, fragment here, and you can see the lysine residue is here, which is susceptible for, oxidation. So these are the residues which can easily get oxidized. And you can see here the list is there that what different kind of oxidation may happen to different kind of residue side chains. So this is a example of carbonylation. You can see here that where this T is being formed, you can see here the carbonyl group is there, and a carbonyl formation happens. And once this carbonyl formation will happen, what will happen? The chain will get terminated here in the from the peptide bond, and this can be easily quantified. We will discuss in detail how to 2,4-dinitrophenyl hydrogen is one chemical compound which can form a permanent adduct with this, and this gets at 340 nanometer you can see its absorption. So it is very easy to identify that the radiation-induced protein oxidation in the cell. So here different kind of in the cell different kind of modifications are happening to different residues, and that is basically responsible for even formation of methoxyl lysine, which is a very common thing when people age. It shows the formation of these adducts, which is responsible. So people measure this as a measure of aging also. Here I will show you the methionine. There we show the ketose and aldehyde amino acids which have ketose and aldehyde groups. So how they modify. Here methionine, you can see that this is also the peptide methionine sulfoxide reductase system is there in the cell. And if small oxidative stress is less, you can see here this methionine sulfur group gets modified to methionine sulfoxide. But and this is PMSR is one protein which is there responsible for the change of this. It converts it back into the methionine. But if the oxidative stress is very high, then it will go further modification, and this will be an irreversible modification which will lead to the formation of methionine sulfone, and this cannot be converted. So that's how this amino acid will get modified, and once it will incorporate, in any of the amino in the protein, these can get incorporated into the protein, and once this will form even the newer protein which is formed because of this oxidized amino acids, they will not be functional. So people have shown the expression level of MSRA and MSRP. These are the repair proteins which convert back to the oxidized methionine, and people have shown that in aged subjects, this level of this MSRA and MSRP goes down, and that is responsible. Overexpression of MSRA in Drosophila melanogaster lead to increased resistance. How it is leading to increased resistance? Because it is able to repair the protein, So once the protein will be repaired, then protein can go and function again. So if you overexpress MSRA, those Drosophila, they performed better against the radiation. That means radiation-induced protein damage is responsible for the radiation sensitivity. And people have shown in neuronal cell also PC12 that it can provide improved detoxification of ROS induced by hypoxia, reoxygenation, and increased protection of cells against apoptosis. So PMSR is a protein which is well known and responsible for radio resistance also. So here you can see in the cell how basically happen homeostasis maintenance. The native protein, it gets damaged. So after damage, it may form either aggregates or it gets degraded. So once it will get degraded, it will form the pool of amino acid, and then again it will come to the biosynthesis. But as I said, if the permanent oxidation of the amino acids happens, so within the cell also, it will, even if it forms a fresh protein, then again that protein may not be very useful if those amino acids are very crucial. So every amino acid is crucial as far as structure is concerned. To attain a three-dimensional structure also, amino acids have a role. So suppose that if they get oxidized, it will not have that proper rotation. It will not have that proper, long range and short range interaction formation. So proper three-dimensional structure achieving is difficult. And then if amino acids of active site got modified, then it will not have the proper function as well. So there are many ways that protein oxidation may lead to the altered function of the of any protein. So here people have shown in mice, this study is done with the fraction of life, how much carbonyl modification may be occurring. So you can see here that with aging, the carbonylation of protein occurs more and more. And this is from 0 to 1 of the lifespan of so many different mice. And you can see here that the carbonylation of protein starts building. And why this is building? Because, you know, PMSR expression, you can see in young mice, in liver, kidney, and brain, there are a lot of expression of PMSR. But the expression of PMSR in liver, still it is relatively maintained. But in kidney and brain, you can see the level of PMSR goes down. So if the level of, in old mice, so if the level of PMSR is going down, that is leading to formation of more and more protein oxidation. And that is probably leading to the accelerated aging. So, you know, I mean, that is how we consider that accelerated aging is also a oxidative stress induced phenomena. So we already studied, I mean, we talked in detail that loss or gain of enzyme activity may happen because of oxidation. Loss of protein function may happen. In many, it is already reported. Loss of protease inhibitor activity may happen. Protein aggregation, we studied that how it can lead to protein aggregation. Then enhanced susceptibility to proteolysis. I mean, it may expose some of the proteolytic site, and that may be get cleaved by the circulating or nearby, I mean, caspases or different kind of proteases. So then abnormal cellular uptake may happen. Then modified gene transcription, as I said, like in case of an, we have shown I-kappa B, they get phosphorylated or different kind of modification, acetylation, different kind of modification happen, and that lead to different kind of gene transcription. Or if suppose that if it has to go and bind to the DNA and that portion got modified, then these transcription factor may not have the proper transcription of the genes. And there are several diseases which are reported because of the protein oxidation or altered expression, or even I mean protein aggregation. You know, protein aggregation is a big problem responsible for Alzheimer's, Parkinson, and mad cow disease, different kind of diseases. You know, like, but beside that, even atherosclerosis, rheumatoid arthritis, muscular dystrophy, ischemic reperfusion injury, we already saw, like in PC12 cells, if MSR expression is down, then I mean, reperfusion injury is more. So that study is also, I mean, people have shown that protein oxidation is predominantly responsible for it. Then cancer and then so many other diseases are responsible. So what is the solution? Again, antioxidants and then radioprotector, which there are a study in Deinococcus radiodurans. You know, like this is one of the bacteria which is very highly resistant, and people have shown that it is mainly the I mean, what they did, that they have exposed a sensitive bacteria to the radiation, and this resistant bacteria to the radiation. And what they then they did, that they found that the damage caused because of the radiation, of the same dose of radiation, is similar, but the processing of these Deinococcus radiodurans of those damage is very fast, and that is why these are resistant. And why they found it is very fast, because the protein damage in these Deinococcus radiodurans was very less, and the protein repair was very efficient in these Deinococcus radiodurans. So they took a sensitive bacteria, a resistant bacteria. They found that it is predominantly because of the resistant proteins. I mean, the resistant bacteria becomes resistant. And we have shown in, we have made a cell line which is resistant as compared to from the parental cell line. And when we checked, we found the protein damage is much less in resistant cells as compared to the sensitive cells. So this is a data from our own lab. And you can see here, I mean, I try to show it in a pictorial form. You can see cell A and cell B. Cell B is already known to be resistant, and these are different DNA repair proteins, which is either in the nucleus or outside the nucleus. But once you irradiate it, what happens? Like these white spots in the lower panel, you can see that these white spots are basically DNA damage, right? So these resistant cells have relatively less DNA damage, but the protein damage is also less in this. So more DNA repair proteins are available to execute the repair, right? So what will happen? More DNA repair proteins will go and execute the repair in resistant cells, and they will form I mean, the chances of survival of these cells which have less protein damage in the cell will be high, and that is how the cell will survive faster and it will become resistant to radiation. Then, you know, like many a times what happens, even oxidized protein, many a times, you know, like these oxidized proteins in the nucleus, you know, all the DNA is complex with protein most of the time. You know, so what will happen, that these oxidized protein, they may form its oxidation state to the nucleus, to the DNA or RNA, and they may oxidize the DNA and RNA also. So that kind of problem may happen. So carbonyl group, how to detect it? You can see here, as I said, like 2,4-DNPH is one chemical. You can expose the cell to the radiation and then take out those cells and lyse them and then derivatize it with 2,4-DNPH, and then there is a process of it, and then you can monitor the after washing, you can monitor the absorption at 370 as compared to control. If you get more absorption at 370, that means the carbonyl formation or radiation-induced protein oxidation can be seen there. Then there are immunoassays are also available. Antibodies are available, which can, you have to derivatize the protein again here, and then antibodies are available. They will go and bind to those DNPH derivatized proteins, and you can identify your protein of interest is whether your protein of interest also got oxidized or not. So using anti-DNP antibody, you can find out. So now coming back to the cellular level. So suppose that all these modifications happened. Dwarkanath sir will tell you in detail about the DNA damage and repair also. So once all these damage happens, what will happen? You can see with this arrow on the left side photograph, this cell is died. And once this cell will die, what will happen? It will release all these oxidized molecules to the neighboring cells, right? And those neighboring cells, these oxidized molecule, on the right side, you can see here that the cell membrane have pattern recognition receptors. They are basically TLRs, toll-like receptors, and they can sense these oxidized molecules. They can sense oxidized DNA, they can sense oxidized RNA, oxidized protein, oxidized lipid, and they will then cause the inflammation in those neighboring cells, right? Because the neighboring cells will bind the oxidized molecule released from the dead cell. Then that's how inflammation progresses, and in the entire tissue. And then those, you can see here the apoptosis, how apoptosis is happening. All these oxidized molecules is being released, and once this gets released in the lower cell, you can see here that these will go to the circulation, and this oxidation or these inflammation will reach from one tissue to the other tissue. And once the exposure, radiation exposure happens, some cells which are very susceptible, they will start dying at a very massive rate. And then they will not only damage to those tissue, because this inflammation will go on, these inflammatory markers will go on in the entire system, and the inflammation will reach to most of the part of the body. So this is how you can stop it, by doing this. And what are the basically most susceptible organs? They are hematopoietic system, which is in the bone marrow, situated in the bone marrow. They are fast proliferating cells. So any fast proliferating cells, as Dr. Rath said yesterday, like cancer, any fast proliferating cells are very susceptible to radiation. So hematopoietic injury and GI injury is one of the prominent thing which happens, and because of this, leukopenia happens. So immune system after radiation exposure, immune system of the person is gone completely. So the person will become susceptible to opportunistic infection, and the person may die because of the opportunistic infection. And then gut also will become the leaky because of the damage to the gut epithelial cells, gut microbe. There are a lot of microbes are there in the gut. They are good in the intestine, but once they will enter into the bloodstream because of the leaky gut, then they may cause sepsis and the person may die because of that. So these are the basically consequences which happens because of the macromolecular damage and cellular damage to the cell. And the overall what we see is a leukopenia as a marker in the body, or the gut damage. So diarrhea is also seen one of the symptom after radiation exposure because of high cell death in this in the organism. So this is it. If anybody has any question, then you're most welcome to ask.
Yeah, I think Dr. Shalja is here.
Yes.
Yeah. So there are certain questions in chat box.
Yes.
So you can read or you can ask questions.
In our message box. So first of all, let me thank Anand for such a wonderful lecture, and I really enjoyed all. So I'll start with the question by Sangeeta Chaudhary ma'am.
I think Dr. Sangeeta Chaudhary is one of the chair. So ma'am, you can ask this question yourself. You are welcome here.
She can read it. She can read it from the chat box. No problem.
Okay.
So the first question was the difference in lipid composition between the tumor cell in comparison to normal cells. So Anand, the question is there any change in the lipid composition between the tumor cells and the normal cells?
Yes, I mean the lipid. I mean there are different kind of lipid. I mean, I don't know exactly, but yes, I mean people say, like rafts are completely changed. Signaling got completely changed. So
yeah, the lipid rafting.
Yeah, lipid rafting.
As far as I think that could be one of the way they change there. And so thereafter, it affects the entire lipid rafting mechanism. That's what I thought, but I thought I would ask you since you're an expert on this area.
Exactly, ma'am. So in fact, that is lipid biology is completely altogether a different field and very complex field actually, because like people have identified so many so many protein adducts are found, but lipid, still it is not listed that how many different kind of lipid species can form because of reactive oxygen species. But
Dr. Anand, if we do, in this case, if we do a metabolomic analysis, can we not find out about it?
Ma'am, it is of course it is able to find, but whatever the techniques which are there, I mean mass and all, because lipid is always a sticky thing.
Right.
I mean, people who study lipid also, they are also very frustrated that many a times they are not able to identify all the species because I mean the technical limitation I'm talking about is whatever the best technique available. That is what I'm telling. Whatever the best technique is available, people could not identify all the different kind of species which are formed because of oxidative stress and all. But lipid signaling, what you are talking about, this raft, because these tumor cells, they have altered signaling, so they have different rafts on their surface, so they may have definitely different composition of the lipid as well.
Yeah, just to add, like lipidomics being done in many other cell system and tissues also. And that as Dr. Anand has already described, maybe some technical limitation, but nowadays the mass spec based ultra sensitive mass spec system are able to tell you the lipid composition, types of lipids between the two cells. So the comparison can be done very easily.
Yeah, Yeah, understanding is much better as compared to, because of the technical advancement, understanding is much better. But based on the interaction, I mean, with the people, lipid biologists, they still say that we still have to identify so many lipid species, which we are finding limitation that probably with the technical advancement we'll be able to identify more.
Yeah. Thank you, Anand. The second question is from Shikha. So she is asking that in the acquired radio-resistant cancer cells, what should we expect the DNA damage to be more as compared to normal cancer cells, or the hyperactive repair machinery?
When we see, I mean, in the control itself, when we see the gamma H2X, let's say, I mean, assessing any DNA damage, you will see the gamma H2X foci is already there in the a cancer cell as compared to normal cell. I mean, the number of foci even without radiation also. So, of course, the repair is much better. I mean, because they have the shortest pathways. Dr. Sir will tell in detail that the NHEJ, which is a short pathway, they have even alternate NHEJ, which is even shorter. I mean, which facilitates the repair much faster. So they have definitely better repair machines, but they have, I mean, sublethal damage is also always there in the cancer cells, because oxidative stress is always high. And as I said, as I showed the evidences that oxidative stress not only directly to the DNA, because the protein which are interacting to the DNA, they get oxidized and then they transfer their oxidation state to the DNA, and in that condition, single strand break, nick formation, and so many things may take place.
Yeah, thank you, Anand. I think it clarifies the question. However, again, Sangeeta ma'am's question related to flippase and the scramblase, which are there on the membrane, and how does the structural dynamics of a cell membrane distributes across lipid membrane when it's exposed to radiation. So the question is, the flippase or the scramblase, they are there on the membrane, and upon the radiation exposure, how this been changed, and thereby the structural dynamics of a cell membrane is being changed. That's the question.
Yeah, I don't know exactly in terms of this these proteins, but like as all the proteins, these proteins are also formed of the same 20 amino acids, and they are susceptible to, I mean, these many of these amino acids are susceptible. So so definitely, like once these are transmembrane proteins. So transmembrane protein may form adduct with the lipid because of the lipid oxidation, if not the protein oxidation, but they themselves also get oxidized and then they form. So the overall, I mean, message signaling may get affected. If they are channels, the channel, I mean, the transportation of the ions and then solutes, whatever important solutes, that may also get affected. So these, if all these transmembrane proteins, they I mean, easily get affected because of the oxidative stress.
Yeah, and just to add again that flippase and scramblase inhibitors are already there in place, because for many other diseases, they are also being tested. So in the radiation condition, if you can add the inhibitor and check whether it is further affecting the activity of flippase or the scramblase.
Yeah, I saw some animal studies were going on this particular area. Yeah. And it's quite interesting to know, actually, because if that happens, then can we kind of stabilize the normal cell membrane? Yes. That would be a real good, I mean, that's the way we can do, because even the bystander cell is getting affected. And so how do we kind of do that?
Because these are players on the membrane dynamics, these scramblase, the flippase, they are the main players.
Yeah.
And you can save them, maybe that will protect the bystander cells. True.
Yeah.
Thank you, ma'am. That was a very nice question. Thanks, Anand. So now I'm moving to the next question, Antara. So she wants to know whether you have checked the effect of radiation on the gut flora upon exposure to radiation, meaning what is the impact of radiation on gut flora?
We have not worked, I mean, we don't work with these microbiological experiments, but people have done. I mean, there are a lot of studies where people have done, and then they found that mainly anaerobes, I mean, there is change from aerobes to anaerobes, and the different kind of gut flora. There is, people have observed the changes.
Dr. Anand, here I can add it because we do a lot. I mean, I'm working on the colorectal gut microbiome. Why I'm saying is, as such, the gut is changed from normal flora. Now, given radiation, the main question would be post-radiation, after a particular period, say down 3 months or 6 months, because these patients do have a lot of GI trouble after the radiation. So how long, or does it at all come back to near normalcy? That's a main question to be asked, because if the disease is still persisting, and especially a GI tract cancer, does it at all come back, or it continuously lead the patient to have secondary infection because of this? So here are multiple questions that need, because anyway the gut is changed because of the cancer. Now, on top of it, you have given a radiation, which is a generalized kind of, it's not targeting only even if you give IMRT or whatever you give, they're not targeting specifically the cancer cells. So you have further changes. And a lot of mice studies have been conducted where they give, pre and probiotics for this very reason, that stabilizing the normal flora or recolonizing the normal flora. So the studies have been done on mice level, of course, and I think there are a couple of clinical trials on this particular area also. But it's some studies say the difficulty is microbiome is too big a area, the thousands, millions of bacterial colonization that happens. So some study will say some particular population, species or genera. Another study will say another. So it's still very kind of ambiguous. And exactly whether it improves or not, multiple factorial reasons. So we still need a lot of work to do on this. If animal studies are done, it would be really good. I know Dr. Vibha and all of you handle animal work a lot. So this area could be really explored a bit more. And this could lead to another therapeutic option out, what should be given, what should be the dosage, and whether it can be given before treatment, post-treatment, at what interval, so that the normal flora is somewhat restored. And then that makes it a lot of things is the normal chemotherapy given thereafter could be, the tumor cells could be more susceptible to them.
Yeah, I agree. Thanks, Dr. Sangeeta. Now, I think there are no any further question. Or Vaishnavi, okay, there's another question, sorry. So
Sridevi Pathak.
Okay. Sridevi Pathak. So her question is, the extent of damage to normal cells and cancer cell will be the same or different at a particular dose? So, yeah.
Yeah, I mean, see, yeah, there are different factors which are responsible, as you said. Like if you are exposing them in the, petri dish, let's say, so keeping in the same environment, then definitely the chances is very high that the extent of damage will be same if you observe at 0. I mean, of course, there are several factors which are responsible, so may not be very different, but chances is very high that they will be same. But the processing, that is what I showed in one of the slides, that the processing these resistant and the sensitive cells, they have different, and because of that it may have different. But in case of tumor, let's say, as I said, like tumors have hypoxia, so then as compared to normal tissue, the tumors show some resistance towards radiation.
Yeah. So Anand, the next question is from Vaishnavi Vishwas, our new student of CMM. So she wants to ask you that in radio-resistant cancer cells, the damage is less and repair is fast. How these cancer cells get selective advantage than the normal cells?
So they are evolved, as I said, like DNA repair is these proteins big because they have, like sometimes this sublethal level of damage or some lethal level of oxidative stress is responsible for continuous expression of these repair machinery in their cell system, right? So because they are continuously expressing, and a normal cell, once after getting a damage, it has to activate that system. But here in a cancer cell, that system is already continuously activated. So that is what they get the advantage of the continuously active system, and they are continuously fighting with that kind of thing. And they are selectively evolved also. I mean, you can see that, that a cancer cell is the evolved cell from our own cell, normal cell. So they have all those machinery to fight with this kind of extracellular, I mean, stress given from outside, like radiation.
Yeah, thanks, Anand. Again, one more question from Vandana, that lipid peroxidation occurs in cancer cells that are hypoxic condition when they are exposed with radiation. So in that case, the question basically is that the conditions, they undergo cell death only due to the DNA damage or lipid or the protein peroxidation. Whether the hypoxic condition has anything to do or that kind of information.
Yeah, hypoxia will make them resistant. I mean, the damage will not be that high. And you know, as I said, that due to hypoxia, HIF1 alpha will produce. HIF1 alpha is a transcription factor which is responsible for giving a protection to the cell. Right?
Recording paused.
Pardon, ma'am?
No, please, Anant, you carry on. There was some issue on the recording paused. So we wanted to record your question answer session also.
Okay. So because of the HIF1 alpha overexpression in hypoxia, the entire repair machinery, I mean defense machinery, will be activated. So once these machinery will be activated, what will happen? It will not let easily the cell to survive. I mean, cell to die easily.
Yes. So of course, the that means the bottom line is the hypoxic condition do play a role.
Yeah,
okay.
Play a role. Yeah.
So then we have another question from Vikas Maurya, one of our PhD student again. So he wants to know that, hi sir, as you said, in radio-resistant bacteria, proteins are the factor to protect it from radiation. Have you checked the genome damage in this? Means bystander effect probably. So he also sees genomic instability or genome damage. That's what he wants to know.
Yeah, so that is not done in my lab. I mean, that is a well-renowned group, Mike G. Daly's group. They conducted those studies. So they have shown, I mean, as I said, like in the beginning, that they took a sensitive bacteria, they took a resistant bacteria, and they exposed to equal amount of radiation. Then and at 0 time, they found the damage caused is equally same in both the bacteria, but the processing is different because the damage to the protein was less in the resistant bacteria. So the damage was equal. So the damage was equal, but the processing was different.
Very true. So I think, Kanan, right now we don't have other question, but it was a real wonderful.
Can I ask
yes. So one of our faculty, Dr. Rawat, he wants to ask. Please go ahead, Dr. Rawat.
Thank you, sir. First of all, very nice presentation, and
thank you, sir.
We'll learn a lot today. So my question is regarding the radiation and stem cell aging. Could it possible that radiation induce the stem cell aging or accelerate the stem cell aging?
So can you repeat it?
So could it possible that the radiation induce stem cell aging?
Aging.
Stem cell aging means that if we give radiation or any kind of ionization radiation in age of 24 or 28 or 30, that if you take the stem cell, after 1 year, after a few months, that we see that those stem cells now have already aging phenomena, or that it induce or promote the stem cell aging.
Sir, actually, radiation actually is known to accelerate the aging. So, and you have seen, I mean, it is predominantly the oxidative stress which is responsible for accelerated aging. So even similarly, it has been observed even in the stem cells also that their proliferation ability is compromised, and then they because of this oxidative stress also, you would see the aging phenomena in them also, in the stem cells.
Okay. Second question is regarding this GPX4 or GPX genes. So what do you think that these GPX genes, do you think they play a role in cancer initiation, or they are, let's say, playing a role in cancer maintenance, let's say? So at what stage this GPX protein, is contributing in?
GPX means glutathione peroxidase proteins.
Correct. Yeah.
Okay.
GPX4, GPX.
Yeah. So I think they have more role in management also, maintenance only, than initiation. I mean, this is what my knowledge says, that they have major role in maintenance.
Thanks, Anand. Thanks, Dr. Rawat. So some of our students have again asked a question, so I think we should also be taking their questions. So Pragya, you have raised hand, I think.
Yes, ma'am.
Yeah, please ask. Yeah.
Sir, you have discussed about the chromosomal damage that happened due to radiation exposure. And we can also tell the amount, we can also measure the radiation dose that person was exposed to. So how that actually can be measured?
See what happens, like after radiation exposure, you take the blood cell of, so suppose that some person got exposed at in the morning today, and anytime, I mean today or tomorrow, you can take the blood, and then or after a few days also. So that is why these assays are called gold standard, because once these dicentrics are formed, they will not go anywhere. So you take out the blood, you proliferate the lymphocytes, and then I mean, there is a process, there is a protocol of 2 days, and you go through it, and then you form a slide. And in that slide, I mean, you can, in a normal compound microscope, you can see, and then these chromosomal spreads, which are formed of every cell, you can identify in every cell how many number of these dicentrics are formed. So you can then go back, there is a curve, and you can go back and check that if these many per cell, these many average dicentrics are formed, then it corresponds to what amount of radiation dose. So that will tell the absorbed dose of radiation to any person.
Yeah.
Thank you.
Thank you, Anand. And just the last question by Dr. Kasi Shankar Venugopal, that blood components such as granulocytes irradiated to prevent transfusion-associated graft versus host disease, what are the parameters need to be monitored in this condition, where you are exposing the granulocytes and then to prevent the transfusion-associated graft versus host disease? So what are the parameters which one should monitor to do this? Yeah.
I think I am not I don't have much idea about it, and I am not a competent person to answer that question.
Yeah, but I think these information might be there.
Right. Yeah. I mean, in bone marrow transplantation, this is what done basically to suppress the immune system. The person get exposed to radiation completely. I mean, the bone marrow, and you suppress the immune system, and then the bone marrow BMT is done. So that is a routine process. But how to irradiate the blood to suppress some of the things that I don't know.
Yeah. Yes. Thank you, Anand. The level of questions and the level of response we have got is really telling us that this talk was really very, very informative and very and your experience with radiation biology helped all of our students a lot. So to the student, I want to say that there is a reaction column. So you can really give a big round of applause to our Anand sir for such a wonderful talk. And I personally want to thank him for such a informative one. Thank you, ma'am.
Thank you,
so
ma'am.
there's a reaction box. Please put the reaction on that. Thank you very much.
Thank you.
So we have our second chairperson with us right now, Professor Sangeeta Chaudhary from Gangaram Institute for Postgraduate Medical Education and Research, Sir Gangaram Hospital, New Delhi. Professor Sangeeta Chaudhary has a research interest in immunology and molecular medicine. We request the chairpersons to take hold of the session.
So our next presentation would be from
Dr. Sangeeta Chaudhary.
Yeah, I'm here.
If you want to introduce, it would be good.
I'm actually sorry for this. I missed the speakers which you had sent. I'm so sorry about this.
Always a pleasure to introduce my own supervisor, Dr. Dwarkanath. I am so attached to him. So
yes, Dr. Dwarkanath actually doesn't need, but yes, we would still like to introduce him. So if you may please go ahead. He's your teacher. I'm really sorry about this. So.
No, not an issue. Thank you. So it's because as already Dr. Sangeeta has said that Dr. Dwarkanath doesn't need any introduction, but he is such a great scientist, I should say, and such a great mentor, because he has mentored so many students and the scientists in his life that we all are so much awe of him, that his knowledge, his experience, his vast experience on the radiation biology per se related to this conference. So I don't want to take much time onto his previous career profile and all, because we all know that he has served in MAS DRDO specific to radiation biology division more than 20 years. While serving in MAS, he was also been guiding factor to other radiation related environmental issue, to very well-known news from the Delhi University, where they found some radiation exposure near to the campus, and he was in that committee to guide to the government and also to the environmentalists how we should solve those problems. But as a scientist, his research interest is to understand the mechanism-based approach for radiation countermeasures, cancer therapy, and also he was some way pioneer towards the use of 2-deoxy-D-glucose as an adjunct therapy for cancer treatment. He has published more than 250 papers, and you all can see in online that such a depth mechanisms he has defined, what is the applied radiation biology one should look towards. So that kind of information, if you really want to gather, all publication will reflect the same. And basically, he also served to China, Shanghai, to create a group in on radiation biology and to guide them to develop a good institution along with the new upcoming technologies related to radiation biology. But now he is back to India and again serving as a mentor to other institutes and to all of us. So with this information, I would like to invite Professor Dwarkanath to give the presentation and to enlighten us with all this radiation biology information. Thank you very much.
Thank you so much.
Thank you, Shailaja and Dr. Sangeeta, for that very generous, embarrassing, and a little frightening introduction. And I hope
you have more than that, which is a lot more not said.
All right. This morning, I will cover certain aspects of cellular response to radiation damage, which is somewhat historic in nature, one, and therefore probably could be very rudimentary to many of the learned colleagues who might be a part of this program. So I request them to put up with it. And for the students, I guess it will probably be an exposure. Radiation biology being such a vast interdisciplinary field, any of these topics will only be a small window opener. There is so much that one needs to look on the other side of the window. I'll be more than happy to discuss even after this program, if anybody so wishes. And the third comment I want to make is Dr. Anand's lecture, my own dear colleague back in MAS, probably has set the stage where we now move towards understanding the response of cells as an unit and not go down, except when we come to start discussing how do these cells die, what is the basic death triggering factor on different mechanisms. We'll come to that later, but then assuming that we have a reasonably good understanding that due to a number of processes that are elicited inside the cell following exposure of cells to radiation, it ultimately culminates in a few responses. One of them happens to be the cell death. And in tumor biology or tumor radiotherapy, the focus is always to achieve local tumor control and also avoid disease dissemination, both proximal as well as distal. What these jargons look very probably a little confusing. The idea is to be able to kill all the tumor cells within the volume that we identify is the volume where the tumor is located, and also see that it doesn't go elsewhere and start growing and create problem, or doesn't drift through and start growing and therefore come back. So the whole idea is how radiation has so far been successful or has had limited success, and what are they due to. Now, from this point of view, a cell which is dead and removed from the site, if it is in the culture, it will disintegrate into debris. If it's in the body, if it is dead and non-functional, it generates, as Anant very elaborately discussed, it generates a systemic response as dying cell sends out signals through various chemical signaling mechanisms. One of them he mentioned, damage-associated molecular patterns, as well as few other things, sends out signals so that the scavengers of the system or the body can come take it away. If that is done, and if it is done for all these cells that are dying in the tumor, and if your radiation killed lot many cells and all of them are efficiently removed, you will then imagine over a period of time what was looking like a tennis ball of a tumor mass will start looking after some time as a lemon, much later as a grape, and much later probably like a mustard. And hopefully the fond hope is you will not even have a mustard sometime later, but that doesn't happen in entirety in reality. This prelude is to make the point, therefore, if this death of the tumor cells and the response that it elicited in getting it eliminated is coined as the radiobiologically induced cell death or radiobiological death, it's good enough if it literally kills the cell, that means cell is non-existing, totally dysfunctional, or it is unable to grow and make its colonies. If it did not, then eventually it will probably stay as a tissue that wouldn't grow much and cause disturbance in the proximity as well as distally. That means you contained the tumor in a given volume, that is cytostatic effect. You did not allow the tumor to grow further. Or much better, in the first paradigm we said, we killed the cells and removed it, and therefore we do not have a tumor in the site that it originally was. And both of these put together will qualify to be called a radiobiological cell death, and both of them have in them the inability of cells to form colonies. Either they're simply not existing, dead, and therefore there's no question of colony, or they still exist physically, but they're functionally, physiologically, here the physiological function of a tumor cell is to divide. That is knocked off. That is eliminated. But these two put together is a radiobiological cell death. With this prelude, let me just share the screen and start the discussion. Let me do this. Okay. Is the slide visible now?
Yes, yes, it is visible.
And I am audible reasonably
yes,
well?
very good.
Okay,
Very good.
good enough. Okay, so this is going to be cellular response to radiation damage or biological damage. And number two, I'll have to keep this slide and a couple of slides because there will be different audience. All this understanding has come from my tenure at several places, and most importantly, initiated, sustained, and grown by my guide and mentor, Professor GIAN, and I'm always indebted to him. I have actually taken the liberty of taking material from across the internet, not recklessly violating the IPR, but it's only for the teaching and non-commercial purposes, and I guess that should be okay. So we go to cellular effects. I need to move this away. Okay. Now what we're going to discuss in the next an hour and a quarter is, if you irradiate a cell, what are the consequences? You could have immediate death, delayed death, and they may lose the ability to divide, as this is what I call as loss of clonogenic ability, unable to form colonies, loss of clonogenic potential. Or the cell may survive, but unable to function physiologically as competently as it was before irradiation. The third one could be that it has alterations in its genetic material, it is surviving, but now in principle has acquired an ability to function slightly modified, related to the modification in the information in its genome. These are all the consequences of late effect. A loss of function and transformation is not what we are going to discuss in this session. I'm going to confine myself to the various aspects of loss of clonogenicity in this step. All right. Now, if Puck and Marcus, close to about 65, 70 years, published the first breakthrough in our ability to answer this question. If I had 1,000 tumor cells picked up from a patient's tumor on hand, and I want to ask a question, how do these cells respond to radiation? Till Puck and Marcus came up with this, we didn't have an idea. But people were already, time immemorial, people were plating bacteria to look at the colony, to look at the resistance to drugs, and so on and so forth. Puck and Marcus did the same thing for the tumor cells and found that tumor cells on a suitable surface can also grow like bacteria. Of course, depending upon the nature of the cells that you want to grow, you need to give different micromilieu, surface, so on and so forth. Ignoring all this, they came up with the same method of assessing. If I put 1,000 cells that were undisturbed on 1,000 cells or any number of cells that were challenged with radiation, and gave them all the things that they need to be able to stay, survive, and grow. When they grow exponentially. 1 becomes 2, 4, 2 to the power of n, and therefore, since they grow in proximity, I should be able to see colonies. And when the number becomes sufficiently large, I can see with the naked eye. Therefore, it's called macrocolony, which is similar to the bacterial cultures and things you grow. Now, several aspects of this is involved. One is, even if you took same number of cells from different tumors, or if you had cultivated these tumor cells in culture and maintained them, the cause of alterations in very many signaling processes related to the proliferation, as we do know that unlike the untransformed or the non-immortalized cells, the tumor cells are transformed in such a way they don't care so much for pampering, and therefore continue to grow with minimal supportive system like the nutrition, growth, this basal growth factor, so on and so forth. That's something that we not discuss about. And therefore, if you put them, they will grow. If you put a lot of cells, due to intercellular interaction, they form a sheet and grow as a monolayer. If you put too few numbers per unit area, then they are dispersed. And if they are still able to survive as an individual without interaction with others, and be able to also not only survive, but also be able to function. The function or the main business of cancer cells is to grow, keep growing, and cause disturbance from the mass that they build. So the bottom line is that their ability to grow. And when they grow, as I said, since they grow in proximity, you can recognize them as a colony. Now, if you did this with cells from different tumors, or the same cell from different cultures or at different ages in the culture, and if you put 100 cells, most often than not, you will not get 100 colonies after a period of time that you gave for them to make colonies after multiplying several times. And that's due to a number of reasons that includes part of the thing that Anant discussed, which relates to the internal stress related to the basic metabolism, and therefore the inability to grow or even to survive. So that's something that we need to keep in mind. And when these cells are irradiated, many more will also be not able to form colonies. What we are interested in is how many cells made colonies after we irradiated as compared to cells which were not irradiated. Because there is a great deal of variation in the number of colonies made under a variety of conditions, which is contextual dependent, we need to know, if we did not do anything, what is the number of colonies. And that we call as the inherent clonogenic potential, but in a slang usage in radiobiology, it is called the plating efficiency. And this is a cartoon, you can keep looking at the cartoon, but I want to focus on. So this typical inherent basal clonogenic ability or the plating efficiency is typically the number of colonies that you got, that you could count after giving certain time, divided by the number of cells that you plated. As a practice, this is always reported as a percentage, and therefore you multiply it with 100, right? When we say visual colonies, which are called the macrocolonies, by a thumb rule, it's about 50, 60 cells. 2 to the power of 5 is 32. 6 is 64, and anything more is. So depending upon the type of cells that you work with, sometimes you might probably have to wait a little longer because the 2 cells are too small and colonies you are not able to recognize. Leaving all this, 6 plus or minus something, or 7 plus or minus few generations. And therefore, that number of generations into the doubling time is what you should be patient enough to start expecting colonies. So all these things are experimental details. When you did all that and got colonies that you'll see in the cartoon, you can start counting the colonies. And if you know what number you plated, you'll get depleting efficiency. Now you can ask a question. I plated and irradiated, or I irradiated the cells, came back and plated, and I want to know how many cells survived a given dose of radiation that I gave. And that's calculated by the ratio of plating efficiency. In other words, you're normalizing the plating efficiency of the irradiated cells to the plating efficiency of the unirradiated cells. That's all what you need to do with all this background to get this surviving fraction. If you did this with several doses, then you can get a dose response. And the dose response, when you plot as a function of dose, the number of survivors, the fraction of cells that are surviving, typically looks like what you see on the left. This will preclude you from deriving some of the information, which are quantitative information that would be helpful later. We'll see how it's going to be helpful. All I needed to know was, how are they killed? Why should I be worrying? We'll defer this question and come to this later. As I said, the whole area of radiation biology was driven by the radiobiological cell death, which was the focus in tumor radiobiology or in tumor radiotherapy. The question, therefore, not spelled anywhere, is if I have a tumor and I have had some number of ways to assess the tumor and say, this is a tumor of this type, and this tumor in this patient has about 10 to the power 9 cells. The question is, and this is one information. On the other information, I'm told that radiation can kill tumor cells, and therefore I can contain it or get it off just by irradiating. Now, the next question is, how much dose should I give? It depends, of course, on how much. If I know the dose response, if I give how many dose, how many cells are killed, and then if I know how many cells are there, then I can go ahead and give, okay, if a gray kills 1,000 cells, I'm going to give this much. In other words, if you know the number of cells, you would like to give a dose that kills n plus 1 cell, or 1 n times 10, at least a decade more, so that you are not going to be left with a single cell that can bounce back. Why not a single cells that can bounce back? Because our earlier experience said that if I put these cells, even in isolation, they can grow and form the colony, which is synonymous with if you treated and left one cell in the tumor not killed, that would regrow because it doesn't care about its neighbors. As long as it lived, it grows and goes. The colony is synonymous to the regrowth of the tumor. And therefore, you would like to ask a question, how much dose should I give? Now, for that, you need to derive a lot of information that will be helpful. If you transform this dose response curve from a linear plot to a semi-logarithmic plot, where you plot the surviving fraction, the fraction of survivors on the logarithmic scale, and then you can look at two things. First and the foremost thing that you will find is a small incremental dose doesn't kill linearly, but it kills logarithmically. That's first thing to be noted. And then it will also let you, for practical utility, it will also let you look at the kill over several decades, 10 to the minus n, minus 1, 10, that is 0.1, 0.01, so on and so forth. If you had 10 to the 8 cells, you will imagine on this curve, this is only representative curve. So you might probably have to give enormous amount of dose, because this is up to, here it is shown as 10 to the minus 3, right? So if you had 10 to the 8 cells, then you will imagine that probably you'll have to go another 5 decades, maybe another 30, 40, great. So the next question, they keep this in mind. I know the tumor and I have to give this dose because I know the dose response curve. Now, if you did this, if you extrapolated this, oh, I have the tumor and I give this dose, you're going to get into trouble. The trouble is our inability to spare the normal tissue and cells en route when we want to irradiate the tumor cells. That's where we need to start focusing on normal tissue cells. By and large, if they are in any of the organs which are terminally differentiated, you wouldn't have so many proliferating cells. Whereas the tumor, which is an island in the organ, is an island of proliferating cells. So you will now start thinking, what are the differences? What are the similarities between the response of a non-proliferating and a proliferating cells? We'll keep that on one hand. We know the total dose to be given, and we know that there are issues in giving this dose in one go. Now, how do we go about it? Much of answers to much of these questions will also be discussed in greater detail, I think, by the faculty who talks about clinical radiotherapy, dose fractionation, so on and so forth. But I'll only set the stage from the radiobiological point of view. This cartoon again in the top shows you have to wait for some time. Now, if you plotted this on the semi-logarithmic plot, it let you, as I said, in our ability to handle that question, it let us derive certain parameters which will tell us when we need to deal with different tumors. The first question is, how do different tumors differ in their response to radiation-induced cell death? And we now have, from the previous slides, we now have a way to generate the surviving fraction values as a function of dose. But I want to know the radiosensitivity of cell type A, B, tumor A, tumor B, so on and so forth. I need some parameters to qualify them. So this is derived from the dose response curve, which will tell you, I'm sorry, I need to just take this. Kavya, I'm in a lecture until 2. Please do not call me. Thank you. So the classical dose response curve, which is on the left side, shows that these are all for the mammalian cells. If I have to refer to other than mammalian cells somewhere down the lecture in one slide also, I'll refer to. Otherwise, all the discussions refer to the mammalian cells and their tumor cells. Now, this dose response curve lets you look at several things. What it tells you is at lower doses, the incremental dose does not kill proportionately the cells. However, when you go beyond, then there is a proportional increase in the cell kill, and this is by decay. For every incremental dose by a unit, there is a decayed increase in the cell kill. This is the first thing we note. As we move up the curve that is towards lower dose, you will see up to certain dose, this slope is shallower and shallower. These are the two things that we need to keep in mind. While most of the cells do respond, tumor cells do respond like this, the different types of the responses that we encounter as a function of dose is a little more complicated. And one of them is shown in the right side, which is continuously curving. And one is completely single slope linear. And the other one, the third one is a combination of the two. And typically, it's the blue line that you most often than not get as a response when you do the dose response curve for the surviving fraction. The classical radiobiological parameters are what's the incremental cell kill in the exponential portion that is shown by that shaded portion where I put D0, and the shoulder up to a dose where there is minimal cell death, incremental cell death, as I increase the dose. This have some phenomenon underlying, mechanisms underlying that we will evolve as we start discussing. And now, therefore, the question is, I said that there are certain things that are contributing to this. What are the factors then that contribute to the kind of dose response curve that we see? It depends upon the quality of radiation. I discussed about the LET yesterday, linear energy transfer, and Mahalingam also discussed. Now, the total dose that you give, if it is broken up fraction, the same 10 gray on the left, the previous slide that we saw on the left side, if I now give all these doses in different fractions, I give, let's say, 1 gray as 0.5, 0.5, 2 as 1 and 1, or 0.5, 0.5, 0.5, so on and so forth. Each of these doses, I fraction and I give with the time gap in between what sort of a response I get. And why do I want to do that? We'll come to that one. When I come, we can combine the two. then the rate at which the dose is delivered influences this response. Of course, most importantly than anything else, the intrinsic radiosensitivity. That's what we are mainly interested in when I am wanting to use all this knowledge for designing a radiotherapy regimen for a given tumor. The cell age and the other one, number 4 and 6 are very important. Of course, LET is important, but if I have a choice between high LET and low LET radiation, that becomes important, but I will do allude to that as I go along. Oxygen is extremely important. There was some discussion about hypoxic tumors being resistant to radiation. Therefore, the presence of oxygen is an important determinant, suggesting that oxygen has a role to play in the radiation-induced cell death. What is it and how do I quantify it? We'll come to that as we go along. Now, if I just look at this dose response curve across the different species, Anant was talking about the bacteria and different types of bacteria. Now, if I put the dose response curve for the virus, the phage, the yeast, the bacteria, and the mammalian cells, you will find the mammalian cells is at the greatest disadvantage. They're extremely sensitive as compared to any of the microorganisms. In other words, the microorganisms require large amount of dose to get inactivated, or radiation-induced inactivation, synonymous with radiobiological cell death, I was discussing. So the inactivating dose are much higher, orders of magnitude higher when we want to inactivate microorganisms. And this is one of the reasons as to why in the irradiation, in the sterilization of very many products using radiation, we use very high doses, because there the focus is sterilization. Sterilization means getting rid of the microorganisms. And that this is the reason as to why it is required to be a higher dose. Now, I mentioned that age of the cells is important. Age within the culture, within a batch culture, I guess everybody is well aware of a batch culture. You have a growth curve to begin with, going to the plateau. It takes some days. Now, in this whole cell cycle, for every cycle, the cell goes from a gap phase that it always rests and gets into the synthesis, organizes itself, and divides. Now, one can ask a question. If the age here is the age between the gap phase first and the second gap phase, it's almost ready to divide. And if I have a way of synchronizing these cells or sorting them out at different ages, which are synonymous with different cell cycle phases or physiological status, is the dose response similar or dissimilar? The answer to that question is no. The M and the G2 phase, the further in the growth curve, is more sensitive. The cell in the S phase is least sensitive or somewhat resistant, and the G1 is somewhere in between. Now, interestingly, there was also a statement: proliferating cells are more sensitive to radiation-induced cell death as compared to non-proliferating cells. And the important event in cell doubling is DNA synthesis. If a cell has to double, or a proliferating cell, the main business is to synthesize and double all its content, organize itself, and divide. If DNA synthesis is an important part of the cell proliferation or the cell doubling, and if S phase is the most resistant, relatively, of course, other phase, and one would wonder, is this at variance with the statement that proliferating cells are more sensitive to radiation, while S phase cells are relatively resistant as compared to the gap phases where the cell is not dividing? We'll keep this question and address this later. Now, as I said, LIT is an important contributing factor. Higher the linear energy transfer, higher is the level of cell death. As you can see here, X-ray, which is a low LET radiation, is relatively less effective as compared to the neutrons and the alpha rays. I think I'll defer all the discussions because I'm just blind. And it appears that there are optimal LET values where you get maximum cell death, and beyond that, the increase in LET doesn't add much, or due to a number of underlying radiochemical mechanisms, but matched with the biology of the cell, which is the responsible factor for the radiation-induced cell death. It appears that it matches the distribution as a function of LET, matches with the size of the double helix within the chromatin, and therefore more number of events in the DNA happens at an optimal LET value, and therefore you get more cell death. We'll defer this discussion. If there is a question, I'll address this later. Now, one way to quantify that radiation type A is more effective than radiation type B, or always the reference is the low LET and widely used photon radiation, which is X-ray photon or the gamma ray photon. And it's therefore customary to report the relative biological efficiency or effectiveness with reference to the X-ray. And that's typically arrived at as the ratio of doses that it will elicit the same biological response or the endpoint response. And the current discussion we have is radiation-induced cell death or loss of clonogenic ability or the decrease in surviving fraction. But it is also true, the biological effect could be anything. You can calculate the relative biological effectiveness with biological effect being the induction of stem phenotype, the induction of EMT, induction of immune response. It is possible. But for the current discussion, we are discussing about radiation-induced cell death. And it is typically a ratio of the doses between the X-ray and the test dose or the test type of radiation. As we saw in the previous slide, alpha and neutron being more effective, this ratio will always be more than 1 if the test dose, the test radiation is more effective because of the high LET. This is an example from our own work. Mohit generated this data some time ago for 3 glioma cell lines. You will see our focus actually, don't get distracted by that. Just look at the pair of orange or red lines vis-a-vis the black lines. The orange ones are the photon radiation and the black ones are the carbon ion radiation. Clearly, all 3 of them show that the dose response is steeper for carbon ion radiation as compared to the photon radiation. However, you will also notice that the gap between the photon pairs and the carbon pairs is not identical. This is not even the dose response for the photon individually or for the carbon. This is only to show, this is reiterating what I said earlier. There is always a heterogeneity in the dose response, and that's exactly what we want to know in dealing with the designing of radiotherapy protocol. And typically you go ahead and calculate the RBE values, and you will see for these 3 cell lines, we had RBE between photon and carbon vary by almost a factor of 2, 1.5 in one of the cells, to about close to 3 in the other one. Now this, because you're seeing this picture, I want to say, I want to mention, this is somewhat Dr. Anand was talking about. When you stimulate the cells for glycolysis, when you reprogram them for enhanced glycolysis, you will find that these cells become relatively resistant as compared to the normal glycolytic status. And this is true for the photon radiation. Unfortunately, it doesn't, or not fortunately, so interestingly, it is not the case for carbon ion irradiation. This is not to say that this is the gospel. This could vary when we query, let's say, hundreds of tumor cell lines. But at least for these three types of cell lines that we queried, the stimulation of glycolysis didn't influence so much the response to carbonylation. In other words, the RBE probably marginally varied based on the changes only in the photon response. As oxygen influences the radiation response, there are two aspects of the role of oxygen in determining the radiation-induced cell death. One is the underlying radiation chemistry. The other one is the associated biological / metabolic changes. Both of them have a role to contribute. The relative contribution, of course, varies in a context-dependent manner. Notwithstanding all those details, as you increase the oxygen tension, the response becomes more and more sensitive. Conversely, more hypoxic the environment is, more resistant the cells are to induce cell kill. And these are all data not in the tumor, inside the tumor, where there is a gradient of oxygen. These are all experiments done with cultured cells, which have been kept under regulated levels of oxygen. You'll see in the first picture, as you move to the hypoxic region, the dose response moves to the right. And I think that's probably enough. And this is precisely because oxygen serves as an intermediate in amplifying the indirect effect because most of the nascent, frank reactive species that are generated through radiolysis of water are short-lived and have limited diffusion distance. They cannot attack too many macromolecules, let alone DNA, of course. But if you have oxygen, which accepts these electrons from the reactive species frankly generated and becomes different types of oxygen reactive species, it has a longer half-life and therefore a larger diffusion distance. The probability of interacting and therefore damaging macromolecules is higher. And that's the underlying chemistry. I said there are two components. One is the chemistry. The chemistry part is actually this. And therefore, one can look at the influence of oxygen even in vivo. And then you will see that if the median oxygen in a given tumor is lower, then the tumor control is not very good. If it is high, then it's better. That's why hypoxic tumors are relatively difficult to deal with. Since we are at this discussion, I just will only mention, because of the hypoxia and HIF-mediated signaling triggered, there is reprogramming of metabolism. So the hypoxia-related metabolic alterations and therefore the associated signaling contributes equally, if not more, than the underlying radiochemical contributions to the resistance, relative resistance due to lack of oxygen or hypoxia. Now, the relative biological effectiveness, which is the effectivity of different LET radiations, and the influence of oxygen actually are inversely related. The lower the LET, more is the OER, suggesting that high LET radiations do not care so much about the oxygen in and around in causing the biological effect, whereas the low LET radiations do. And Anand did mention that between the direct and indirect effect, where in the indirect effect, the radiolysis, the radical generation is the dominant factor. In case of high LET radiation, much of the damage that is caused to the vital macromolecules is through the direct interaction of traversing energy with the macromolecules. And therefore, oxygen has little impact in determining the outcome, as you can see in the top quadrant. And the second cartoon is the same. We don't have to discuss. Next, we come to this. I'm sorry, this slide should have been 3 slides before. This is nothing but the summarized information from the curve where I showed the response of synchronized G1 cells, early S cells, late S cells, G2 cells, and M cells. This typically shows the same. I'm sorry about that. We also said that the dose rate is an important determinant. Now, if you irradiate to the same level of dose, but different dose rates, the dose response curve changes as shown in the right-hand quadrant. The left one is the paradigm or the experimental design. The acute high dose rate has the sharpest dose response. That means elicits a strong response. Now, if you keep increasing the, if you keep reducing the dose rate, that means you will need more time to put in the same damage as compared to the acute high dose rate, which is the leftmost. We are comparing for the same dose at different dose rates. Now, if you reduce the dose rate, you will find that the dose response becomes shallower and shallower. That means cells are able to survive better than the same damage put in a short interval of time. And this is because of the repair that is occurring during irradiation, and the cells are able to survive better. Now, if you go below a certain dose rate, then you will see that other phenomenon starts contributing that we will come to a little later. Keep this in mind. As we reduce the dose rate, the survival increases as compared to high dose rate. However, when we reduce the dose rates enormously, then as compared to the reduced dose rate, the cells start responding a little more sensitively than the dose rate where it starts reversing. These are the cartoon in the left bottom is only showing the high dose rate response and low dose rate response spread amongst several mammalian cell lines. I think this is several years ago, but now there should be data from maybe multi-folds number of cell lines that are clearly showing there is a difference in the dose response at different dose rates. There are also 2 relevant phenomenological considerations relevant to the radiobiological cell death or the tumor response to radiation in RT, mainly because of the conditions. What are they? The tumor is a growing island, as I said, in an ocean of relatively less number of growing cells around, which is the normal tissue in which the tumor is growing. Of course, the number of cells in the normal tissue that are in cycle varies from tissue to tissue. Much larger in the marrow, which is a tissue, to a large extent in the GI, the epithelial cells are turning over, and like this, so on and so forth. Minimal in the CNS tissue, and in between comes your kidney, liver, so on and so forth. So that's the kind of paradigm or scenario. And the tumor is a growing cell population within this. And when you irradiate both. And in our ability to design the delivery of radiation as a therapeutic tool, we want to derive from the knowledge that we have gained that increasing dose will kill, and dose rate will influence, oxygen influence. We have not. We put all that on in designing. We are confronted with the following, that still it doesn't let me give the dose that is required to sterilize the n number of cells in one go, because my normal tissue cells en route would be severely compromised. Tumor would probably be contained before that the patient may not be necessarily good condition, if not even alive. So how do I give the radiation? Now, these are some of the initial observations that form the basis for fractionation. Now, one is called the potentially lethal damage repair. We will be visiting this a little later. I'll be talking about potentially lethal damage. There probably Karsali will mention, since it is relevant here, I'll go. So radiation induces lesions that causes the cell death. If not all lesions are potentially lethal, that means the moment the lesions are formed, that leads to cell death. But they're having a potential to be lethal, but they could be manipulated. The question is, under what conditions they could be manipulated, how they get manipulated within the tumor, how they get manipulated in the normal tissue around, or relatively non-proliferating tissue around, and so on and so forth. Now, as an extrapolated paradigm, if you took a growing tumor which grows in a Gomperzian way, in other words, modified exponential way, an initial high growth rate followed by a plateauing, which is similar to the batch culture as well. There are less number of growing cells as you go to larger tumor volume, and more number of growing cells in the smaller tumor volume. Now, which is in a way somewhat similar to growing tumor cells amidst the non-growing normal tissue around. Within the tumor, you now have small tumors, large number of growing cells, large tumors, less number of growing cells. Now, if you irradiate both of these and just observe them, you're not going to do anything, just irradiate and observe them. Now, when you want to query, if you irradiated and ask the question, how many cells in the tumor I killed? In other words, how effective my radiation was. What you could do is irradiate the tumor, take the tumor out and plate them. The moment you plate them in the culture is completely supportive. And oxygen is available in plenty because they're in culture, and you have put all the nutrition, so they're ready to go start proliferating, right? Now, if you did that with a solid tumor, but now you do irradiate the tumor, immediately put them in culture. In this, conversely, you irradiate, wait for a while, wait for a little longer, wait for much longer, take the tumor out and plate them. Now, if you did that and then looked at the survival, the relative survival, what you will notice is in the small size tumor, you will see, in the small and the big, you will see the longer you wait, more number of cells will survive, or the number of cells that you killed reduces as the time gap increases, that you will see on the left side. However, the degree to which this happens is much pronounced in the solid tumor, suggesting if you had more number of non-proliferating cells, and if you waited for longer, they survived even better for the same duration in a population which were actively growing. Now, we could recapitulate this in culture, irradiating the exponentially growing phase cells vis-a-vis plateau phase cells. When the cells have reached the plateau, you can do this dose response, you will come up with the same. Now, these are because some factor that is linked with the less ability of cells to proliferate, cells to go through the cell cycle, help them recover from the radiation damage. The longer the time you gave, the better the recovery they made. Keep this in mind, okay? The other one is more important and the critical underlying feeding factor for the dose fractionation. Now, once we finish, we come back and answer, how do I give this radiation, total dose of radiation, and sterilize the tumor? The other one is, you took these cells, these examples are in vitro, which should also be true in vivo. Each of these doses I mentioned earlier, when I first discussed the dose response curve, that they're in the lowest dose, there was shallow response, and as we increased, the response was high. And if I said, if you split the dose into different fractions and gave the total number of dose in 1 fraction, 2 fraction, multiple fractions, and plated them and asked a question, what would happen to the dose response curve? Typically, these experiments are done by plating them already and giving them different doses by fractionating. Now, if you did that, and in the left side, you will see, if you incubate them between the 2 doses at a lower temperature, 30, these are mammalian cells, 37 degrees, they keep growing very well. At reduced temperature, relatively temperature attenuated, lower temperature attenuated, lower rate of progression through the cell cycle. You will see as the time gap increases between the 2 doses at lower suboptimal growth conditions, the surviving gets better. Now you do the same thing. At the same temperature, the cell is happily growing. That means at 37, these are all growing cell population, because this is in culture. You now irradiate them at different time gap. You are fractionating with a time gap, and the time gap is shown on the x-axis. The time gap between 2 fractions is varied, let's say 1, 2, 4, so on and so forth. You will now see that it is a complicated complicated response. Initially, the survival gets better. The cells are able to survive better. Now, if you waited for a little longer, that benefit is totally reversed. However, it is not worse than in one single dose, that is 0. But the survival becomes lower than the best is shown here at 2 hours. This is for CHO cells. Only look at the phenomenology and don't bother about the quantitative values of dose and the time gap. Okay. So in other words, a growing cell population between 2 fractionation responds differently, where there is a better survival followed by a sensitive phase, and again a better survival. A sensitive, relatively resistant to sensitive to resistant. Whereas always in the suboptimal growth conditions, the time gap always leads to better survival. Now the right one formed the basis for the dose fraction, which is more clearly exemplified in the bottom picture. And if you did the same thing with low LET radiation and high LET radiation, as we said, the high LET radiation deposits energy very closely, makes very difficult, complicated lesions that the cell is not able to repair and recover, and therefore damage induction is almost the major determinant of cell death. Whereas with low LET radiation, damage is induced, but the repair has a major say in their survival. Now, if you did this splitting of the dose for the high LET and low LET radiation, you will find that the gap between high LET radiation here, neutron, and the low LET radiation X-ray is larger in the fractionated dose. The neutron dose almost remained the same. There is in the left one, the neutron dose, which is a straight line, suggesting it's a exponential dose. In the right one, the fractionation, there is slightly a bad artist drawing a straight line. That's almost it's a straight line. Whereas X-ray, the slope is way, way different than the X-ray slope for the single dose. So the bottom line is that in growing cell population, by splitting the dose, you will elicit a response and catch a place where the cells actually respond more sensitively. And this does not happen with low LET radiation to the same extent as the low LET, not with the high LET radiation as the low LET radiation. Now put all these things together and ask a question. With all this information, how do I design my radiotherapy? So that forms the basis of fractionation, which I think the clinical radiobiologists will discuss, but I only will make the following. Therefore, by fractionating the total dose that you need to give for a tumor which contains n number of cells, by giving that much dose, which will sterilize n times or 10 times n cells, when you know what is the dose that will kill an order of cell, that means bring down the surviving fraction from 1 to 0.1, 0.1 to 0.01, 0.01 to 0.01, so on and so forth. A decade kill is what we will know. If I know how many cells I have, if I have 10 to the power of 8 decade cells, and I know that every 3 gray will kill a decade, then I know I have to give 11 times 333 gray. Or if it is 5, 11 times 555. Now this 55, since I cannot give it in one go due to enormous normal tissue damage in and around, I want to fractionate, and I have the rationale for fractionating. And that is, in growing cell population, it will elicit a sensitive response in between. Now, if I can match my time between 2 fractions and the dose per fraction in such a way that the differential, that this does not happen so pronouncedly in non-proliferating cells, which I said is the ocean of normal cells around, and happens in the growing island, which is the tumor cells where there are a lot of proliferating cells. If I did this, what I'm going to do, I'm going to elicit a differential response in this island. I'll give a radiation, I'll give the next dose of radiation when I elicit relatively a more sensitive response. And I told you that there is a phenomenon linked to this, which I'll come a little later. But in order not to carry the inquisitiveness, it's related to the radiation-induced perturbation in the cell cycle. Radiation synchronizes cells, retards the growth of cells in different phases of the cell cycle, G2M being the most, and that's the most sensitive phase. Therefore, you give a dose today, maybe come back after several hours, maybe tomorrow, when many of the cells have accumulated in G2. Give the second fraction. Now you are irradiating a population which is even more sensitive than the first. Whereas such a thing doesn't happen in a non-proliferating population, which is your normal cells. And that is the basis of dose fractionation. To be able to give, and coupled with this, we also saw that the lower the dose, better is the repair. So you give more time for the normal tissue to repair and make the tumor cells come to sensitive phase and get the second dose, succumb more, and this cycle continues. There are more other contributing factors, which are called the 4 Rs or the 5 Rs. I guess your clinical faculty will discuss that. Now we will come to a little bit of classical. As I said, it's a little classical radiobiology that I'm going to run through. It's only to give a flair, because radiobiology goes without mention to target theory is incomplete. Now, you should also understand these theories put forward by Madam Lee, was much before our knowledge about the double-stranded nature of the DNA molecule. We didn't even, the DNA helical structure was not discovered. These are all in the 30s and 40s. All we knew is you irradiate cells, the cells don't survive, right? So Madam Lee suggested, and her co-workers, why do cells get killed following exposure to radiation? She suggested that radiation are nothing but photon bullets. And when it hits a sensitive target in the cell is killed. It's something like taking you go to the fair and there's a game. There are a lot of balloons put, and he gives you for 10 rupees some bullets, and he asks you to fire. The number of bullets that you can manage to hit will give you a prize. In an exactly similar fashion, the radiation is the bullet that's in your hand. The cells are there. You keep firing. If you hit the target, then you kill that cell. Your probability of hitting a target which will kill the cell increases. Watch my words. Your probability of hitting the target increases as you increase the dose. And that is why as you increase the dose, more and more cells get killed. That's the bottom line. Is this really true? As I told you, this is all pre-DNA double helix area. The data that we have, which continues to hold till today, is if it is really true, then smaller the target, harder it should be for you to hit, isn't it? If the same balloon was very tiny once and you were asked to hit with they're all spaced around, you'll find it very difficult. You need more number of bullets. You'll say, no, for the 10 rupees, I can't be doing with 10 bullets, but I will probably need 1,000 bullets. Exactly similar fashion. And we also saw the dose response curve vis-a-vis mammalian cells, all the microorganisms starting from bacteria to yeast, the virus. And what's the difference between mammalian cells and this? Now we know that the nucleus is different. Now this has more DNA, so on and so forth. But certainly, yes, the nucleus, the size. But at the time when the target theory was enunciated, the sizes, each of these entities are smaller. So smaller the entity, lesser are the probability of inactivating, and therefore higher the dose that you get, higher the dose that you require. That is precisely what you will see, that as you go to a lower sizes, you will require higher dose to inactivate. Now, when we come to discussing with this backdrop, the dose response curve, the mammalian cells, which differ from one another, both in terms of their slope, as well as the shouldered region, so on and so forth. Same is applicable. The dose levels are different though, but you will still, there are mammalian cells. We did see some examples, especially the tumors and the normal cells of the hematopoietic and their lineage are extremely sensitive. There are quite a few sensitive lines. So there are cells which show a single dose response relationship or the purely exponential. There are also dose response curve where you see a shoulder and then followed by an exponential kill. And this is understood by proposing that there are multi-hits that has to happen, the n number of targets. So there is not only one target, there are more than one target, but there are multiple targets. If you hit all of them, then the cell dies. In the first, there is a sensitive target. If you hit, the cell dies. And that changes as a function of dose in an exponential manner. Therefore, there is only one single slope. To understand the non-simple exponential or the shouldered response, the proposal, the mathematical proposal is that there are more than one target. When you inactivate all of them, then the cell death occurs. Your probability of inactivating all of them increases as you increase the dose. And therefore, at lower doses, you inactivate probably a fraction of these targets, and then therefore you have progressively less cell death. And there are a number of theoretical models which have been proposed. As I said, single hit, single target, multiple, single hit, and the two components, that there is a linear component and a non-linear component. And the non-linear component is suggestive of that there probably are two events that are occurring within. When they are interacting, then their contribution starts increasing. The probability of two events occurring in proximity will increase with the dose, and therefore at higher doses, you have more cell death. And the linear quadratic model precisely look at this, resolves the linear component and the quadratic component. And at a given dose, the contribution from the linear component towards the cell death and the quadratic component. Quadratic component means an initial lesser cell death and a much higher cell death at a higher doses. That's the quadratic equation, right? So when both of them contribute to the same level, the alpha, these are referred to as alpha and beta in the equation that I don't know if I had shown earlier. No, maybe coming next. Okay, but this is something that I'll not deal very much in detail unless warranted later, because the clinical radiobiologists will deal with this, because this forms the basis for designing and in sparing normal tissues which have different dose responses based on the fraction of proliferating cells with the tissue damage linked to cell death as an endpoint. There is also a model which is linked to, which was born out of the observations of the potential lethal damage recovery, suggesting that the dose response curve is as it is, which is a shoulder dose response, because not all lesions are lethal. Some of them are lethal, some of them are potentially lethal. But as the dose increases, the fraction of lethal to potentially lethal damage increases, more and more lethal damage. And therefore, a lethal, potentially lethal model suggests that the nonlinear dose response on an exponential phase. Of course, the non-simple exponential dose response curve come from the relative ratios of lethal to potentially lethal damages that are induced. And one of the later models formed the beginning of the focus on the ability, the emphasis for looking at the response of cells as a major contributing for the cell survival rather than the disease. All these models that we have discussed so far, before I come to repair saturation, only suggested that you cause the damage, you inflict the damage, the cell dies. But the repair saturation model, from the data accumulated by this time, paid attention to the fact that as the dose increases, the repair system gets saturated, and therefore the cells will be left with more and more unrepaired damage to long time intervals, and the death occurs on account of that, suggesting that it's not only the damage induction or not only the hitting of the targets, but it is actually what happens of the target after the targets are hit, or what happens after the damage is inflicted. Repair restitution, be it DNA damage, be it protein damage, as Anant was discussing, put together, because the cellular response to radiation damage is nothing but an enzymatic reaction where the radiation generates more and more substrates. Whether it's a DNA damage, protein damage, lipid damage, it generates substrates. The repair system is a set of biochemical reactions, works in an orchestrated manner, and therefore, the more the substrate you generate by different doses, you will saturate the repair system, and the lesions have to wait, or they never get repaired, and therefore death occurs. So the repair saturation model suggests that it is because of saturation of repair at higher doses, the cell death starts increasing with the function of dose. This brings us to the discussion of what is involved in the repair of damage caused by radiation. Dr. Weber, how much time we have?
Yeah, I was thinking that if you can stop in 5-6 minutes, then we can discuss. Or if you want to go ahead, you can go.
No, 1 second. 1 second. Let me.
No. How many are left?
No. What I will do. Oh, no. I thought because we had squeezed, I had actually thought that we will come somewhere here and stop and ask ourselves a question. Because Anant already discussed in detail about the lipid and protein, he wanted me to discuss the DNA damage. Now, cellular response. Cellular response is the DDR and other response, right?
Yeah.
So what I will do probably is stop at DNA damage.
Okay.
Next 3 slides. That's all, 3 slides. And keep the actual response after showing one cartoon and say that this is what I'm going to discuss next. and which will probably not require much time because the other things I'm going to run through, which is actually various DNA repair pathways, which is all one cartoon per slide, and which will probably not require so much because the things that I discussed required a little bit of speaking. The rest probably are more cartoon driven. So let me complete 4 more slides and stop.
Okay. Okay.
All right.
Okay.
So we have come to the stage where the dose response curve, which facilitated us in designing a radiotherapy protocol based on all the knowledge that led us to fractionation based on the radiobiological phenomenon, brought us to the stage that in deciding the dose, in deciding the radiation-induced cell death as a response or no death occurring is related to the repair systems. Now, very many studies which looked at labeled protein, labeled lipid, and delivering the same amount of damage, and our ability to irradiate using microbeam to different levels. If this is the cell surface level, you can deposit radiation here, you can deposit radiation here, you can deposit radiation. In other words, if I go from top to bottom, I can deposit bulk of the radiation at the cell membrane level, top, in between, bulk in cytoplasm, and then change it further and bulk in the nucleus. All these studies put together on the one hand, and radiation dose response for cell inactivation or the cell death, and absorption of radiation and causes of various DNA damage have suggested very strongly, while damage to other macromolecules do contribute to the radiation-induced cell death, damage caused to the DNA is principally responsible for radiation-induced cell death, more so the radiation-induced strand break, and much more the radiation-induced double-strand break is the major determinant of radiation-induced cell death. This is also partly absorbed in the repair saturation model. All you need to do is the repair of DNA damage. It should be replaced with the damage at large. So therefore, it's been a study of radiation-induced DNA damage and cellular response to DNA damage has occupied a central role of classical as well as modern day radiobiology to a very large extent. So it's therefore prudent to just spend a little time. This is only the cartoon in the top is only to show the direct indirect effect. We don't have to worry, we have a number of them discussed. There are innumerable lesions that ionizing radiation can cause, and each of these different physical and chemical agents cause these type of damage predominantly, but ionizing radiation is one which can cause all these damages. However, the relative proportion of different types of these damages vary. It varies both as a function of the environment. It varies to a certain extent among different cells, but by and large, the induction of DNA damage is composed of innumerable type or a plethora of DNA damage, starting from base loss, base modification, typically oxidation damage, deamination, strand breakage, as I said, single and double, chemical modification to different moieties, intra and interstrand crosslinks in the DNA, crosslink of the DNA with the protein, the associated chromatin protein, both histone and non-histone protein, and a number of things. Most of the non-strand break lesions come from the oxidation products. Anant discussed about the radiation-induced lipid damage and protein damage. This cartoon again is only recapitulation of the different type of lesions and the contribution to these different types of damage from both direct and indirect effect. The cluster of damages are much more from the high LET radiation. In other words, the damage in a given vicinity in the genome or in the DNA is much more dense and complicated for the cell to handle when irradiated with highly radiation, simply because of the high density of events that occurring in a small time, in a small linear distance, because of the high linear energy transfer. But in case of low LET radiations, it's spread all over. The distribution of lesions across the genome, across the DNA, is far simpler than the cluster damage. Now, the radiation-induced DNA damage, particularly, as I said, strand breaks are the most important and vital ones, is not a simple single event. Actually, the breakage of the phosphodiester band, which marks the formation of the strand break, happens from more than one pathways. I think I should share the slide. Is it now a little bigger? Yeah. Okay. Now, do not worry so much about what is contained in the cartoon, and even less read that. It's only sufficient to pay attention to what I'm going to say. So radiation, the double-stranded DNA and the helix has the backbone, the sugar backbone, and different bases, and the linking of these bases to the sugar backbone. Now, radiation can ionize either directly or indirectly each of these constituent moieties of the DNA, both on the sugar as well as in the nitrogenous bases, and also break the phosphodiester bond. However, much of the radiation-induced strand break do not happen by the direct attack of either the radicals that are generated through the indirect mode, that is generation of offload of radicals, nor is it through the direct deposition of energy in the DNA that it readily cuts the phosphodiester bond and makes the break. Now, much of it happens from the generation of various oxygen and nitrogen centered radicals in the constituent moieties of the DNA, and their interaction thereof, depending on the proximity, which is again linked to the helical structure, the pitch at the different grooves, and that leads to the weakening of the phosphodiester bond, resulting in the strand break. There are a number of models. The most valued contribution in this area of the radiochemical basis of radiation-induced strand break formation came from a stalwart called von Sontag in Germany, who did pioneering work by irradiating isolated nucleotides, nucleosides, and trinucleotides, small stretches of DNA, oligobases, and so on and so forth. And then bacterial DNA, salmon sperm DNA, the mammalian DNA, on all sorts of work, he and his co-workers, and looking at it from the various radicals that are generated when they irradiate each of these constituents in isolation and then in association in small stretches of DNA and large stretches of DNA. If anybody is interested in the mechanisms underlying strand break induction through the generation of various centered radicals, I recommend reading von Sontag's book, which was published way back in 1990. Now, predominantly DNA damage. Of course, the non-DNA damage also contributes to the radiation into cell death. And as we have now reasonably accepted that it's not that the damage induction that's the sole determinant of the radiation into cell death or any response. It's actually how do cells respond to the damage that is caused is an important or a critical determinant of the fate of the cell. Either dies how, repairs, bounces back, or survives with compromised functioning, survives with altered functioning. So therefore, it's prudent to discuss the cellular responses to radiation-induced damage. While I do put both DNA and non-DNA damage induced both by photon as well as photon as a representative for low-LT radiation and particle for high-LT radiation, elicits both DNA and non-DNA damage, which sets into motion a lot of cellular responses, which are nothing but a set of orchestrated signaling pathways that come into picture, which are both spatially as well as temporally organized, that leads either to the recovery, repair and recovery of cells, or death. A death can happen in more than one pathway, this probably I'll discuss, and the whole thing can be summarized as there are pro-death promoting signaling pathways. Both of these will compete for the lesion. So here is a scenario where you have a bait there and two teams will compete. One team is tug of war. One team will try to pull the cell towards death with the lesion as the pivotal point, and the other one towards the survival with the lesion as the pivotal point. It is easily said as a story, but indeed, this is what happens in a hitherto not completely understood manner. There are a number of players, as I said, which are transcriptionally regulated. There are radiation-induced genes that come into picture whose products play a role, but much of it immediately after irradiation, it is the existing machinery predominantly driven by a lot of proteins, which undergo a post-translational modification proportional or in relation to the damage that is caused, which belong to the kinases, phosphatases, oxidolases, as well as the metabolic flux and the redox state, which join together, drive, as I show, on both sides. And on the one side, the ATM, ATR, and other P53 different products, they elicit all these responses: the repair, the division delay, autophagy, apoptosis, stem phenotype, senescence, and the bystander effect. Whereas the survival and an awful lot of other proteins, other regulators facilitate proliferation and also immune modulation. At the tumor level, it is facilitative, but radiation can also cause immune modulation that could be detrimental. I guess that probably, yeah. So this is another cartoon with a little more information, not this by no means exhaustive, but only to give a flair of that there are huge players. So this could lead either to a mitotic death, which is linked to the mitosis for the death to occur, or the mitotic death has two connotations. Mitotically death, that means the cells are unable to carry on the mitosis, that they're unable to grow, as I said, is the static effect, or the death occurs following mitosis after irradiation. So this is ensuing from the damage, the repair, leading to residual damage, manifests as cytogenetic damage in the pre-mitotic cells in the chromosome, post-mitotic in the daughter cells, which are recognizable, which are form the basis for the dosimetry as well. And then it leads to secondary apoptosis and leads to loss of clonogenicity. But these lesions can also elicit a primary death response in terms of apoptosis, necroptosis, which is a form of regulated necrosis, autophagy, which is both survival facilitating and death facilitating, and leads to senescence and phenotype. So these responses collectively determine the outcome that is listed at the bottom, and we will discuss more in detail later. So I'll come to DNA repair pathways in the next session, maybe about 15 minutes, and then go to the next topic. I'll stop here, Dr. Viva, and if people are still not hungry, I'll take questions.
I think we have the question. Now we have a lunch, sir.
All right.
So we have a question answer, and then we will go.
All right.
Yeah. So Professor,
I'll check in. Yeah, that was a very elaborate lecture. Dr. Dwarkanath, you didn't say anything about the abscopal effect.
This is coming. radiation induced by
it's in the
standard
next
effect
session?
and upscopal. Unfortunately, it will come later. I will be discussing both upscopal effect, but I will show a cartoon so that in case you're not there in that
No,
session.
I'll be there. I'll be there. It's okay. I just thought that since you were giving the background of everything, so fine, that will be next. So
I think here it is. Here it is. Can you see this side?
Yeah.
Yeah, the right one is actually the example of the upscopal effect, which is suggesting of the radiation-induced immune modulation. But never mind. I will discuss, but not so much in detail because I do not know the clinical radiobiology person how much and what he will discuss. So
then in that case,
I'll certainly touch.
Okay, sure. I'll start with some of the questions that were put up in the chat. So this was, I had only asked about it. What is the long-term immune modulatory effect of radiotherapy?
Long-term immune modulatory, in terms of what? There are two paradigms. A whole body or a partial body exposure vis-a-vis focal irradiation of the tumor.
Focal irradiation, if you say to it, then.
So focal irradiation of the tumor.
Because I was talking from the point of TME, that is a tumor microenvironment. So
yes,
we can say about the focal.
Yeah. When you talk in terms of long-term effects, if you're asking a question to elicit a response, whether the immune regulation in the system would change or the immune regulation in the tumor microenvironment, in the sentinel and the proximal nodes, as well as the spleen change permanently or only show transient changes following focal irradiation of the tumor. Is that your question, right?
Yeah, both transient as well as if there is any permanent.
The permanent changes in the immune response, if any, is only inferred, but probably not unequivocally demonstrated with particular paradigms. Right. Why do I say what I say? This is from you graft a tumor, irradiate, and you also, let's say, grew out the other side, the classical experiment of the upscopal effect. Yeah. And then look at the responses from the constituent immune cells at the spleen and this, and the nodes, and in the tumor microenvironment, looking at the secretions and the population, so on and so forth. Now, if you did this, left for a long time, you come back and graft the tumor again, the same tumor again. But the difference in the tumor take under those conditions will give you an indirect answer to how did you alter the immune system at the systemic level, because you graft not near the irradiated tumor, but maybe in other area where the systemic host response to the implanted tumor comes into picture. So such studies do show equivocal results, but therefore for me to say, no, there won't be any change whatsoever in the spleen, in the node on a long-term basis is probably far-fetched. But to say, no, the immune imprinting is altered once you focally irradiate a tumor would also probably be not prudent. I think these studies do show that the tumor take is different, but is it different irrespective of the primary lesion where you irradiated? And some of these studies, the limitations are not all of them orthotopic tumors which were irradiated and challenged again in the orthotopic. As you were rightly mentioning, it is the tumor microenvironment for that particular type of tumor in its particular host is more relevant when we talk about the immune profile in the TME, although the spleen probably wouldn't matter much. The nodes, and particularly the TME, would be more influenced by the nature of the grafting. And some of these studies that I am aware of are not with the orthotopic tumors. Probably, I'm afraid I will not be able to very decisively answer this question. That's my limitation.
Well, I mean, there are theories that are being hypothesized that this is the reason that post-radiation, you need to have a chemo that would probably target these newer immune, kind of modulated immune regulation that happens. So radiation alone is the reason, probably never complete therapy. Can we say that way?
Okay, let me put the same thing in it. Those therapies which have the potential to also modulate the host defense, particularly the immune system, vis-a-vis only radiation, or vis-a-vis those drugs which are relatively less efficient in targeting the immune system or modifying the immune system, have shown that an agent that has the ability to modify the immune system, either alone or in combination, has always been more effective in not only achieving local tumor control, probably also in reducing the regrowth. This is an observation. If you want to interpret that as the chemo enhance the immune competence as compared to the absence of chemo, is probably interpretation. For example, the cyclophosphamide at low doses,
right,
and the mTOR modifiers, which actually target the Tregs, particularly Tregs, which is immune suppressive in nature, have shown that the tumor response follows either the Treg suppressive regimen or the Treg not interfering regimen. These are some of the results that are currently available. But these results are also by no means very long term.
Right. Yeah.
By no means long term.
Yes, definitely. Okay.
Professor Vibha, I'm Dr. Prashant.
Yeah.
Can I make a small comment on this?
Yes, please, Dr. Giridhar. Yes, please.
Yes.
So in the clinical setting, we don't see consistent effect of radiation on the immune system. So we have done a series of meta-analysis, individual tumor site wise, to see whether there is a consistent correlation that happens between radiation and the effect on the immune system. It is kind of heterogeneous. Some studies actually show that there is an effect where anti-tumor immunity is stimulated.
Yeah.
In certain other cases, it is not so.
Yeah.
So I think the intratumoral as well as the intertumoral heterogeneity in the tumor microenvironment is still not completely elucidated, and therefore we cannot give a consistent statement that radiation will have this kind of effect on the immune system.
I think that that answers even one of my own questions, which I had posted, that immune cells vary in their radiosensitivity. So how do we target, those resistant cells?
So in the tumor microenvironment, unfortunately, we have a series of immune cells that are there: M2 macrophages, M1 macrophages,
right,
T helper cells, cytotoxic T cells. So when you give a dose of radiation, all of these get affected. But the point is, individual tumors have individual ratios of these cells, and therefore, the effect of radiation also varies.
Yeah.
Second thing, there have been some phase 3 clinical trials that have proven that if you combine radiation with immunotherapy, it does improve at least disease-free survival. We have recently the Pacific trial where post radiation we have added durvalumab, and that has improved kind of disease-free survival in carcinoma lung. But there have been trials that have failed in head and neck cancers, multiple trials have failed to improve survival with immunotherapy. So
are
it's
these
not been
checkpoint
consistent.
inhibitors, doctor?
Sorry?
Have they checked the checkpoint inhibitors here?
Yes.
Yeah.
So
Okay.
the Pacific trial was basically radiation in an N2, that is locally advanced carcinoma lung, non-small cell carcinoma lung. Post radiation, you start with durvalumab. So durvalumab is a checkpoint inhibitor, and that is continued till progression of disease. So there have been a good disease-free survival benefit with that. So that has happened. But in head and neck cancers, when they combine pembrolizumab along with radiation, that has not happened. So it's been similarly in cervical cancer, nivolumab has failed. So there have been some good instances where immunotherapy, at least the available immunotherapy till now, have worked, and in many cases they have failed. So we are still searching for the answer as to how we can utilize radiation to the best effect in modulating the immune system.
Yeah. One way would be over here, since you said about, the same drug not being effective in different cancers. So obviously they are against PD-L1 or CTLA-4 or something like that. Yes. So the way is to induce not induce, bring around expression of these and somehow in the radiation therapy or something, if that can be done. In the earlier study, Dr. Anand did mention about the structural dynamics changing. So I don't know if anybody has done that kind of studies.
So to that point, ma'am. So what we have with us is a series of drugs which are checkpoint inhibitors, say PD-1, PD-L1, CTLA-4,
PD-1 and PD-L1.
Yeah. We also have CTLA-4 inhibitors. We also have OX40 inhibitors.
Yes.
So all these things primarily act on a very small part of the immune action.
Exactly.
So what happens in the tumor cells is that even the neoantigens that are produced actually get suppressed because of mutations either in the MHCs or in the the proteins that would actually have to push the MHCs so that they get presented to the antigen presenting cells.
Yeah.
So for example, just for an example, in head and neck squamous cell carcinoma, we know for a fact that 1/3 of all tumors have suppressed neoantigen production presentation. And therefore, if there is no antigen and we are just blocking the PD-L1, the immune action is not going to happen. So that part also needs a lot of research, how to bring about neoantigen presentation and all that.
That's more like you have the hot and cold tumors.
Exactly.
So that's the major concern over there. Right. Yeah.
Right.
Okay. Dr. Dwarka, there's a question that has been sent. He's asking why few cancer cell types, for example, HSC3 or SAS, are unable to form colonies in clonogenic assay. And the ability of the cancer cell type to form colonies different from cell lines with male or female origin. Is there a difference? Or why don't they form the same?
The second question, honestly, I do not know. I've never paid attention to, but the HeLa cells are the ones meant to just, for having said that, HeLa cells probably form colonies, even if you don't want, if you're walking in the lab, they'll form a colony behind you. That's the same. So to say that the cells from the female
cell line,
but I do not know. If he's talking about ovarian cancer cell lines, there are a number of ovarian cancer cell lines. But to the first part of his question, that why do some when we say that these tumor cells are transformed in such a way that their anchorage dependencies for stimulation adhering is much less. There are still tumors, as I said, in a context-dependent manner. There are still some tumor cells which do not. For them, you have to give appropriate environment like agar or any of the sponge, chick embryo sponge, so that you provide them a structure. That's why organoids are more preferred, and they typically mimic the 3D and the microenvironment, since you can now generate with multiple cellular component, three-dimensional cultures are much better. That's something that I did not discuss because I was just taking the phenomenological approach. We have done work with organ cultures I've done with spheroid culture, or I've not done any work with organoids, but my colleagues back in Shanghai are continuing that we initiated. But three-dimensional spheroid responses, we have done ourselves work compared to the monolayer cultures are very different due to a number of reasons, partly integrin signaling, FAK signaling, and so on and so forth. So depending upon the kind of transformation that the cell has undergone, you may probably have to provide these what so-called the appropriate matrix for them to lodge and form colonies.
Okay, thank you. So there's one more question which says, optimum LET leads to, I think Dr. Vibha herself, optimum LET leads to maximum cancer cell death. Is this the same case in normal cells? And how the higher dose of LET decreases the efficacy?
Okay. In case of extrapolating this information, firstly, as I said, this optimal LET is just a proposition. It happens that at that LET, the spacing of the lesion in the distance-wise matches with the double strand. Assuming that double strand DNA is the critical target, which we believe are, there is more probability of the lesions induced on both strands of the DNA besides in and around, making the cluster very effective. But in case of normal tissue and the utilization of this information for designing therapy, it does not come from delivering equal dose to tumor and normal. With the LET, especially particle radiation, what you would deploy them is to exploit their ability to deposit much less outside the defined volume because of the Bragg peak, or when of course you have a solid tumor, Dr. Hemant will appreciate, but you need to spread out the Bragg peak by using different energies, but you still hope to deposit maximum to the tumor, minimum if not nothing to the normal, and therefore the relative abundance of the lesions in the normal is expected to be less. If you don't do that, if you induce the same lesion, of course, it will have the same consequence. Now, even with LET radiation, even if you did generate so much, if you use a higher LET, of course, you will come up with the same response. But with the low LET, the repair and recovery, because of the repair at least linked to the progression through the cell cycle, is what is exploited in the fractionation. Between particle and photon radiation, it's actually the physical as well as the ballistic properties which is targeted more, because while we do believe because of the nature of lesions that the high LET radiations generate, it will induce more cell death, but the differential is not derived only from the differences in the biological response, but as of now, more from the ballistic and physical properties of the high LET radiation.
Okay. And this is, I was just asking when you mentioned that different temperature, like temperature dependence, so it just thought, the thought just occurred, is this, like you said, lower temperature? Well, our body inside would have one singular temperature, 37. How can we achieve this as a therapeutic option? This is
proof of concept work with the cell lines. As I told, this proof of concept cell lines. But there it is not the use of temperature as an interventional agent. This is to show no matter how the progression through the cell cycle is retarded. The classical one is the liquid holding. Even in culture, we don't use temperature. We do liquid holding, but you could also, in CHO cells, this is a good system. You can just put them in room temperature, irradiate, put it in room temperature, come back tomorrow, put them back in the incubator. For ease, those studies have been done. But the bottom line is to show suboptimal growth conditions facilitate recovery from radiation-induced damage leading to death.
Okay. Okay.
Yeah.
I think there's one last question where Deepak Kushwa, I think, he says that, could you please explain a little more on why suboptimal conditions increase chances of survival of cancer cells? Will this be true for both solid and liquid cancers? I mean, leukemia, I'm sure he means that.
This would be not linked to solid cancers or liquid cancers. Okay. Now, in case of liquid cancers, they're moving all over, right? Now, if you isolated these cells from the liquid cancers and query them in culture and generated single cells from solid tumor, queried them in culture by generating suboptimal growth conditions, the response in vitro wouldn't be any different. However, you need to find out what sort of an endpoint you will look at. And in case of liquid cancers, you can still do that, probably use an appropriate culture to derive the readout. The cell death readout needs to be ascertained, but the phenomenon of proliferation-linked fixation of damage leading to enhanced cell death or suboptimal growth condition related decrease in the damage fixation leading to better survival wouldn't be very different between solid and liquid cancers. Maybe Dr. Prashant had something to say. Phenomenologically, I wouldn't think that they're going to be any different.
I think Dr. Prashant.
All right. Could you repeat the question once, please? Because I had some connectivity. Yeah.
Oh, okay. Just a minute. Yeah, the question that was asked was to explain more on why suboptimal conditions increase chances of survival of cancer cells, and is this true for both solid and leukemias?
Oh, okay. I probably answered only the latter. The first one is the first one probably I didn't address at all. When we say proliferation-linked fixation, typically, if a lesion is not completely repaired and the cell moves from G1 to S, it will amplify that. And the probability of susceptibility to an amplified damage is more than what it was when only the lesion before amplification. Same is true when a cell divides with lesion before completely repairing or rectifying. Then you amplify the damage, cross it onto the daughter cells. When that happens successively, there is increasing level of damage that elicits a response, and which is going to be more amplified than the lesion, which was to begin with. By holding it under suboptimal growth conditions, not letting it amplify going through the S phase or amplify going through the M phase, you enhance the chances of better survival. This is insofar as why does it happen. The second part, I think we already discussed, and I think that's where Dr. Prashant wanted to add something.
Yeah.
Between solid and liquid cancers. Yeah.
So regarding suboptimal survival conditions, let me just give two examples. One is a tumor cell which is quite large, such that a part of the tumor is hypoxic, a part of tumor is oxic.
Right.
So the part of the tumor that is hypoxic is expected to be radioresistant. The reason is because oxygen does not reach there. So for radiation, once it hits it, the oxygen fixation doesn't happen. And therefore, in a suboptimal situation in the microenvironment, that is, the tumor is hypoxic, the tumor is expected to survive more. And we do see these things in cervical cancers. That is, cervical cancer which appears kind of proliferative, responds quite quickly to radiation, while a cervical cancer that clinically has mainly necrotic tissue, either in the pathology or on clinical examination, seems to be quite resistant to radiation. The second situation is when you create a suboptimal condition by giving some chemotherapy or so. That is, externally you induce a suboptimal condition for the tumor to survive. Now here, the situation differs based on the drug that you're giving. Like for example, if you give paclitaxel, it arrests the tumor cells into G2M phase. And therefore, if you deliver radiation, say 4 to 6 hours after you're given the chemotherapy drug, there is more cell death that is happening.
Yeah, that is sensitization.
So the creation of suboptimal condition again will affect the tumor control either way. It may be better in some cases, it may be worse in other cases.
I think he was talking about he was talking about the suboptimal growth conditions that facilitate recurrence. You're talking about exploiting the cell synchronization either chemically or even that's also true between two fractions where you push them to the sensitive phase. He was asking about when I was discussing PLDR. I was discussing PLDR. I think to that he was asking the question.
So in the clinical scenario, we don't see much of effect of PLDR.
Yeah,
so PLDR is mainly a cell line or a in vitro experiment.
It's a phenomenological proof of concept experiment to demonstrate.
So it doesn't have clinical relevance for routine patients.
It doesn't in one limited sense, but I gave the example of as you were telling, Dr. Prashant, a small tumor, same fibrosarcoma at a smaller size, fibrosarcoma at a larger size, you irradiate them, make single cells and plate them in culture. You are right, half of the answer is right because your query is still in vitro.
Yes, sir.
But the whole entire radiobiology, cellular radiobiology basis is that what you observe here is actually to some extent happens there, of course, in limited sense.
Yes, sir. I absolutely agree with that point. That it is lab research that has led to
question from that point of view.
Yes. So, I totally agree that lab research is what has led to any clinical usefulness of radiation.
To say that our ability to observe and then exploit in vivo probably is limited, but these phenomena do contribute to the response. To show that, I was giving some examples both from in vitro and in vivo irradiation, of course, in vitro query.
Yeah, I think that's all the question out here.
I need to move because I have another commitment, Dr. Vibha.
Yeah.
I'm already late.
Yeah, so Dr. Chaudhary, you want to close it?
Yeah, I need to because I had a commitment at 1:30 and I'm off by 10 minutes.
So fine. So
we can close this particular lecture session.
Okay, so fine. Now I can see other hands also, but we can talk later. Dr. Dwarkanath will be there. So I would like to thank Dr. Dwarkanath and Anand both for this session, and Dr. Sangeeta Chaudhary for wonderful, I would say, to initiate wonderful discussion. Dr. Prashant Giridhar to add to it. And I would really thank Dr. Sangeeta Chaudhary, whom
thank you so much.
We never met each other, and we never met each other, and she agreed, and it was really fruitful. So thank you very much. Can I ask Dr. Prashant that can we come back? You can leave, Dr. Dwarkanath.
Thank you so much. I would probably join towards the end of the day, if at all. Okay.
Yeah. 4 o'clock. Because
maybe around that.
Prasanna will be happy to see you. Yeah.
Oh, I'm sure. Yes.
Yeah, you have to join.
No. Yeah, I have to. Correct. You are right. Otherwise, I would be short.
And you should. So, Dr. Prashant, can you give us permission that if we start at 2:10, would you mind?
Sure, sir. Absolutely.
Because people have to have lunch and they must be.
Absolutely. No issues.
Yeah. Okay. So we will start the next session at 2:10. And once again, thanks to everybody. Thanks to the participant for such an active participation, and the people who are registered participants, please try to say you are being followed by the GIAN Kharagpur, not by me. So they are writing that these are the registered participants. We didn't see them, this, that. So please try to attend it, and it is good for you also because every lecture is connecting to the other lecture. So if you don't hear one lecture, you cannot connect it to the other lecture. So with all these things, we will assemble back sharp 2:10, and we will start the lecture. Dr. Prashant, you sent me the lecture, so I should upload, or you will be uploading?
I do have the presentation with me. That was just sent in as a backup if by chance there is.
Yeah. So you can do it.
Yeah.
Fine. So we'll see each other at 2:10. Okay.
Yeah, sure.
Yeah, we are logged but we'll see later. So I would
Vibha,
like to thank Dr. Shalja also. Yeah.
Yeah, Dr. Vibha, I'll also take leave immediately and I'll join back again later.
Okay, thank you.
Yeah.
Thanks a lot, ma'am. So let's be in touch.
This was wonderful. Yeah.
Okay, thank you. Okay. Yes, Dr. Mishra, Vibha here. You are muted, sir.
Good afternoon, Vibha. Good afternoon. Thank you.
Yeah. So, sir, we have actually spilled over a little bit. So we will be starting at 2:10.
So it will start by 2 o'clock.
That's what I'm saying. We will start at 2:10. Although the speaker of the next say session is with us, Dr. Prashant, but we have got up to 1:30 because of the question answer session that is spilled over. Dwarkanath's talk was there. So we just announced that we'll do it at 2:10.
Okay, no problem.
Yeah.
I'm there.
Okay, okay. Thank you. So now everybody started coming in BARC in person?
Yeah, we are working for quite some time. For last, I think 6-7 months. We did not stop in third wave, so-called.
Yeah. No, we also, I didn't stop, but we just. So Dr. Tulkar.
Hello.
Yeah, I'm here.
Good afternoon.
Yeah, sir, welcome. So today you have along with you Professor Harisharan Mishra from BARC.
The program says Dr. Rakesh Kumar Tyagi.
Yeah, Rakesh Tyagi spoke, but today he had something to do, so he did yesterday. So Professor Harisharan Mishra is a molecular biologist. He's here. He has already joined us. So, and the two speakers are there, Dr. Prashant Giridhar and Pataji Prasanna, and we sent you the speaker's profile.
Yes,
and,
I got it. It's in front of me. And Dr. Prashant is actually my old colleague.
Oh, good. Then you introduce him. Yeah. Great. So it's a kind of and the only thing, because we spilled over the last session, so we requested to start at 2:10.
Right. I'll be there. I'm there.
Okay. Okay. So you're welcome, and thanks a lot for accepting it.
Thank you for the honor.
So yeah, so that's there. Okay. And we have planned it in such a manner that we have almost 15 to 20 minutes for asking questions and everything.
Right.
Because a lot of questions are coming. So questions will be in the chat box.
Okay.
Yeah.
Okay.
Vibha, this question answer also we have to manage, or you have somebody who will handle it?
No, sir, you have to manage. If you have any problem, I can manage. But there are not so many questions. Questions are there, so many, but they are in chat box.
Oh, okay.
So from chat box, you can read and do it. And if you have any problem, we can manage. That's not a problem.
So we can direct the question to speaker also, in case, because they are.
Yeah. It is for speaker only. You have to just read it from there.
Sure.
You have to read and ask from the speak. It is for a speaker only.
Yes.
Mm-hmm. So, Dr. Tulka, how long you are in AIMS?
I am for last 29 years. I'm here.
Oh.
It's 30th year.
Okay. So in same building, IRCH
for 3-4 years, I was a senior resident in main building.
Hmm.
And since 95, I am here in IRCH.
Okay. So are you involved in research also, or just clinic work?
Yeah, as part of our job.
Yeah.
Seeing research and patient care. It's a multi-pronged like that.
Actually, I collaborate. We have started a collaboration with Dr. Ritu Gupta.
Right.
But I know Dr. Pratik and Dr. Ritu Gupta, and a couple of people from very long. But, so what have you been doing exactly as a in the
I'm a radiologist basically. So my main work is patient care. So we have a lot of patients are there, and there's pressure from the large crowd. So our research gets somewhat compromised. We don't get too much time for research.
I know.
In years I was alone in the department. Now fortunately I have my juniors. So it's basically
that is the problem in AIMS. Like
exactly
so much of patient load that you are not supposed to do that. You are not able to do it.
Yes. Still there are long waiting for patients.
Yeah, I agree. I'll be just back in within 10 minutes.
No problem. I am here.
Pragya?
Hello, everyone. I hope
So,
we are back with all the energy to move on to our next session.
Just a minute, Pragya.
Professor Sanjay Thulkar, Head of the
Pragya.
Radiology Department at Institute Rotary Cancer Hospital, AIMS, New Delhi, and Professor Hari Sharan Mishra, Head of the Molecular Biology Division,
Pragya.
Homi Bhabha National Institute, Park, Mumbai, to chair the session. Professor Sanjay Thulkar has his area of interest in cancer radiology, specifically breast and prostate imaging, as well as non-vascular interventional radiology. Professor Hari Sharan Mishra has his expertise on molecular biology, genetics, and cell signaling. I request the respected chairpersons to take hold of the session.
hello, am I audible?
Yes, you are audible. Sanjay, you can pick up
hello,
the
everyone.
first talk. Yeah, how are you?
Fine, thank you, sir. How are you? And
very good, dear.
And I thank you, Professor Vibha Tandon, for the honor. And at the outset of the session, with the permission of my co-chair, Dr. Mishra, it's my privilege to introduce Dr. Prashant Giridhar. Hello, Prashant, can you hear me?
Absolutely, sir. How are you, sir?
Fine, fine. It's good to see you here. It's
yes,
a good pleasure. And it's my privilege to introduce one of my old colleague, Dr. Prashant Giridhar. He is a young radiation oncologist. Currently, he is assistant professor at Tata Memorial Center, Radiation Oncology Department at Varanasi. He is actually clinical radiation oncologist, radiotherapist, and he did his graduation from JIPMER, Pondicherry, and post-graduation from our hospital, AIMS, New Delhi. He also held the position of medical doctor, senior resident at the Department of Radiation Oncology, AIMS, and so he has vast experience in treating the patient actually. And he's also research associate at NCI AIMS. So early in his career, he has more than 57 publications in reputed journals with a lot of citations, and that is really creditable. So over to you, Dr. Prashant. And Dr. Prashant will be speaking on tumor radiobiology, radiobiological basis for fractional radiotherapy, and 5 R's of radiotherapy for tumor, microenvironment, inflammation, and immune responses.
Thank you, sir.
Prashant, you may start.
Thank you, sir. Thank you, sir.
Pragya?
Yeah.
Fine.
Could you enable share screening, because I think
have you made everybody co-host these?
Yes, ma'am. They are the co-host.
Dr. Prashant, your share screen must be green.
Yes. Now I'm able.
Yeah, we can see. You have to go to the full screen mode.
Yeah. So now can you see the screen, ma'am?
Yeah, yes, we can see, but can you come out of this? I don't know. There are two. I think in some.
Next slide, you can turn it off if possible.
Yeah. There is some I don't know how it happens, but it happens.
Not sure how this is happening.
Yeah, now you can go to full screen. Yes, here.
Actually, what happens is that you have to stop sharing, and again you have to go and do the full screen share, because several laptops, the latest one, have this only, the way you are sharing. You again go back to the share screen.
Yeah, I've gone there, ma'am. I think.
And then you have to say entire screen.
Yeah. Now can you see the screen, ma'am?
No. No. And then you have to share your slide.
Okay.
Share a screen. Yeah. Again, the same thing. Okay, you go to the slideshow.
Still the same, right? The next slide is also being displayed there.
Yeah, there are options. I don't know how to do it. May from anybody knows these
Hello.
guys?
Hello.
Yeah.
Yeah, sir, please escape कीजिए एक बार.
Escape close कर दूं?
नहीं escape करिए. Escape.
Okay.
Escape the one.
And then go slideshow.
Slideshow.
Yes, sir. Orange bar. Slideshow.
Fourth one. Here. Insert design, transition animations, then slideshow.
Next to it.
Next.
Insert
animation.
1 minute. Yeah. Okay.
From beginning.
From beginning. No, but still that second thing is still showing.
Display setting you can change.
Now how is it?
Yeah,
Is it okay now?
done. Okay. Great.
Right. So I'll be talking initially about fractionated radiotherapy. That is the biological basis for fractionated radiotherapy. And then we'll move on to basics of the tumor microenvironment and its clinical implications. So to understand fractionated radiotherapy, we need to learn initially the 5 R's of radiobiology. So the 5 R's of radiobiology, as shown in this picture, includes repair, repopulation, reoxygenation, intrinsic radiosensitivity, and redistribution. So before we move on to individual points in the 5 R's of radiobiology, initially we'll have some discussion about historical perspective of evolution of radiobiology, the 5 R's, the clinical implications, and then basics of a cancer cell and basics on tumor microenvironment. So as we all know, Roentgen initially discovered X-rays. Then in 1896, Becquerel discovered radioactivity. Then the Curies discovered polonium and radium. And then Marie Curie coined the term radioactivity. So it was in the late 1890s and the early 1900s that radiation oncology as a field started. Radiobiology is the science dealing with the study of action of ionizing radiation on living things. So the principal aim of radiotherapy in clinical practice is to deliver the maximum possible dose to the target while sparing the adjacent normal tissues. So what is fractionation? Fractionation is delivering the total dose in small fractions. And in early experiments, this fractionation, that is delivering small fractions of small doses, showed to improve therapeutic ratio. So the history of fractionation begins with the ram story. This was done by some of the French scientists. So ram, that is male sheep. So the farmers were worried that the population of ram was one, increasing, and second is that the female sheep used to get pregnant, and therefore they could not utilize the wool from the sheep. So the French scientists found that they could not sterilize the male sheep by giving a single dose of radiation without extensive damage to the skin. Then what they did was, just as an experiment, they divided the same dose into multiple days and then gave the radiation to the ram. What they found was the sheep was sterilized, but the extensive skin damage that was seen was not found. And therefore, the 4 R's of radiobiology was born. So late in 1970s, Withers reviewed the radiobiological basis of fractionated radiotherapy. And he provided initially the 4 R's of radiotherapy, that is repair or recovery of sublethal damage, reassortment or redistribution of cells in cell cycle, repopulation or proliferation between fractions of radiation, and the 4th one is reoxygenation. And the 5th R was coined later on, that is inherent radiosensitivity of individual tissues. So I think this part has been already discussed in previous sessions, that the ionizing radiation can act by 2 ways. One is by directly ionizing, the other one is indirectly ionizing. So in clinical sense, when we use X-rays or gamma rays or electrons, most of the biological damage that happens in tissues is by indirect ionization. In fact, 70% of damage actually happens by indirect action and 30% acts by direct action. So indirectly ionizing means that the gamma rays or X-rays enter the body, they produce free radicals. These free radicals then damage the DNA. So when there is radiation given, there is a large number of lesions that develop in the DNA. Most of these are repaired successfully by the cell. So if you give a dose of radiation, what you see is about 1,000 base damage, about 1,000 single strand breaks, and about 40 double strand breaks. Now, these base damages and single strand breaks are non-lethal damage and therefore will get repaired. So what we need is a double strand break that would then lead to the death of a cell. So just for operational classification, radiation damage is therefore divided into lethal, sublethal, and potentially lethal. So lethal damage is that kind of damage that would lead to direct cell death. Therefore, it is irreparable. Sublethal damage is a damage that is repaired in hours if a second dose is given later on. Potential lethal damage is there will be cell death under ordinary circumstances, but these can be modified by changing the environmental conditions. So in the clinical sense, we see lethal damage, we see sublethal damage. We don't see much of potentially lethal damage. So first R. So we would start with repair. So when a DNA is damaged, either in the tumor cell or in the normal tissue can be early reacting tissue or a late reacting tissue. There can be single strand and double strand breaks, and therefore we have certain repair mechanisms for individual things. So if it is a single strand repair, it can be a methyl transferase based repair, it can be a base excision repair, it can be a nucleotide excision repair or a mismatch repair. And if there is a double strand repair mechanism, it is by non-homologous end-joining or homologous recombination repair. So I won't be going into the details of these repair mechanisms because we then veer off from the topic. So what is this concept of sublethal damage repair? So if you give a dose of radiation now, and then you wait for a few hours and then give another dose of radiation, some amount of sublethal damage gets repaired. That is, some of the DNA breaks get repaired. Now, this repair occurs both in cancer cells as well as in normal tissues. So, what is the clinical implication of it? So, what we know is that cancer cells in general have less recovery or repair capacity, while the normal tissues in our body have a higher repair capacity. And therefore, if you give a dose of radiation and then wait for some time and then give another dose of radiation, it is expected that more normal cells, that is normal tissues in the body, will get repaired compared to tumor cells. Therefore, more tumor cells are expected to die. So considering this concept of sublethal damage repair difference between normal and tumor cells, we have one aspect of fractionation. So fractionation would create a bigger difference between a repair in a normal tissue versus repair in a cancer cell. So by splitting radiation dose into small parts, cells are allowed to repair sublethal damage. The amount of damage that is repaired depends on the ability of the cell to recognize the damage and activate repair pathways or cell cycle arrest. Now, what we know is malignant cells have suppressed these pathways, often through mutation or inhibition of p53, preventing them from undergoing efficient repair. While normal tissue cells in the body, they have intact repair pathways and therefore repair the sublethal damage by the time the next fractionation is delivered. And that is why in clinical sense, usually radiation is given with a 24-hour gap. And when you're practicing something called as hyperfractionation, you give at least a 6-hour gap between two radiation fraction doses. So 6 hours is the minimal time that is required for repair to occur. So that is called as the repair half-life. So interfraction interval and the repair half-life is an important consideration when fractionating radiotherapy. Some tissues, notably the spinal cord, appear to have a slow repair mechanism with a half-life of about 4 hours. And therefore, we usually split at least by 6 hours between 2 radiation fractionations. If the dose rate is sufficiently low, repair may be able to take place during the radiotherapy treatment. This considerably reduces cell death due to sublethal damage. So potentially lethal damage repair, again, this is something that happens mostly in vitro, where you incubate, say, a cell line in a balanced salt solution instead of full growth medium for several hours after radiation. Then you expect the cells to die. So this is not something that happens in a physiological situation, and therefore it does not have much of clinical use. So this is about the first R, that is sublethal damage repair. So before we move on to the next R, a basic description about what a cell survival curve is. I'm sure this has been explained in the previous lecture as well. So it basically describes the relationship between surviving fraction of cells and the absorbed dose. Cell survival as a function of radiation dose is graphically represented by plotting the surviving fraction on a logarithmic scale on the ordinate against the dose on a linear scale on the abscissa. So there has been a talk about high LET and low LET radiation. So the type of radiation also influences the shape of the cell survival curve. Densely ionizing radiations exhibit a cell survival curve that is directly exponential to the function of dose. For sparsely ionizing radiation like X-rays, gamma rays, the curves show an initial slope followed by a shoulder region and then become nearly straight at higher doses. So this curve must have been already shown. So a typical X-ray would show a curve which is shown above orange, and a typical neutron or a densely ionizing radiation would have a straight line. That is an exponential kill. Same thing again. So if a radiation dose is given in a series of equal fractions, which is separated by time interval that allows complete sublethal damage repair, the effective dose survival curve becomes an exponential function of dose. So if you can look at this curve, this is when a single fraction is given. So what happens is there is an initial shoulder region, and then there is an exponential region. Now, if you divide the dose into smaller doses, so instead of giving 12 gray in a single fraction, where this would be the kill that is happening, the survival fraction comes down drastically. If you give the same 12 gray divided into, say, 12 fractions, what happens is the shoulder region gets repeated again and again, and therefore the surviving fraction increases. Now, this surviving fraction, when we speak in terms of normal tissues. The cancer cells do not have this kind of repair capability, and therefore they would die at the same rate as previously, or at least close to the same rate as previously. And therefore, to save the normal tissue, as you can see, this would be the effective survival curve when fractionation is done. The second R of radiobiology is redistribution or reassortment. So the concept of different phases of a cell cycle was introduced by Howard in 1953. So a single cell cycle in a proliferating cell would be 18 to 24 hours in time, out of which there are 2 gap phases, that is G2 phase and a G1 phase. And you have a synthetic phase, that is S phase, and then you have got a mitotic phase, M phase. So it is the mitotic phase that where actual cell division happens. Then in the gap phase, there is cell growth, there is duplication of organelles, and there is preparation that happens for DNA replication. And in the synthesis phase, DNA synthesis happens. And in the G2 phase, again, for the cell prepares itself for cell division. So this cell cycle goes on and on again. Now the cell cycle, that is movement from one phase to another, is governed with in the presence of cyclins and cyclin-dependent kinases. Now, different phases require different cyclins. As you can see, cyclin D here, cyclin E here, cyclin A and B here, and similarly cyclin-dependent kinases. So these things would stop or push the cell into the next phase of the cell cycle. Warren Sinclair did some experiments to see whether the phase of the cell cycle actually affects the survival curve. Now, what was seen was that the cells that are present in the mitotic phase are the most radiosensitive, while the cells that are present in the late synthetic phase are the least radiosensitive. So as you can see, there is a gradation. So if you have an entire group of tumor or entire group of cell line that is completely in mitotic phase, you'd achieve a higher cell kill with the same dose versus an entire cell group or a tumor cell mass that is only sitting in the late sensitive phase. But in reality, what is present is that a tumor has cells which are in different phases. Some of them would be in mitotic phase, some of them would be in S phase, some of them would be in G1 phase, and therefore we wouldn't have a similar cell kill because of this. So why is this happening, that the late S phase is relatively radio-resistant? This is because there is a presence of a sister chromatid which favors repair. While in the G2M phase, there is no time for repair. The cell proceeds to mitosis and therefore it encounters a mitotic catastrophe. So, following a radiation dose, the cells in the G2 and M phase are expected to be killed more. Cells in the S phase are expected to be killed less. So, cell growth kinetics that are present in individual tumors that would differ from an individual set of tumor versus the other would determine what percentage of population will be in each phase of the cell cycle. So how do we utilize this, this heterogeneity of tumor cells in different phases of cells? So that is where the concept of reassortment or redistribution comes. So we give a dose of radiation now, and then we wait for say another 18 hours or 24 hours. That is a single day. So what happens is, because of the radiation dose today, a large amount of cells in the G2M phase die. A percentage of cells in S phase do not die. And by the time you irradiate these cells again tomorrow, some of these cells in the S phase have moved into G2M phase, and therefore they get killed better. So instead of giving a 10-gray dose single shot today, if you divide them into 5-gray today and 5-gray tomorrow, what happens is we expect a higher cell kill because of this concept of reassortment or redistribution of cells into the next cell cycle phase. Reassortment occurs more quickly in tumors. Again, reassortment favors the survival of normal rate responding tissues. Now, what we need to know again here is we are again looking at difference between how the tumor acts versus a normal tissue. So normal tissue, say a spinal cord or a muscle or a nerve, they do not have a very fast proliferation capacity. That is, they don't keep on moving from a mitotic phase into the gap phase, into the S phase, into the next gap phase on a regular basis. While tumor cells are proliferating all the time. And therefore, it is expected that tumor cells move from one phase to another quickly, and therefore, they would move into a radiosensitive phase more often compared to a late responding normal tissue, and therefore, a reassortment would provide a therapeutic gain. So, the general conclusions from this is that cells more sensitive to radiation are close to M phase, resistant to S phase. They're usually sensitive in the G2 phase. But we do see some exceptions to these rules in certain cell lines. So how do we utilize this in radiation therapy? So basically it is used in two ways. One is by just fractionation, as I've already explained. The other way is using certain drugs which push and stop these cells into a particular phase. The most common drug that is used is a chemotherapy, that is paclitaxel. Now, paclitaxel is known to stop or arrest the cells in G2M phase. So what happens is, if you give a dose of paclitaxel a few hours before your radiation dose every day, what happens is the cells get synchronized into G2M phase by the time you give radiation. And therefore, this will kill cancer cells more. So this is how you use redistribution or reassortment for therapeutic gain in clinical radiation therapy. The third R is repopulation. So the two R's that we have seen till now, that is sublethal damage repair and the reassortment or redistribution. Both of these things have helped in therapeutic gain. That is more cancer cell kill and less normal tissue kill. The third R, repopulation, is the opposite. So repopulation. So whenever you damage or cause cell death, there is an increased rate of cell proliferation that happens. This is called as repopulation. More commonly, we call it accelerated repopulation. So this repopulation occurs in two places. One is early responding normal tissues, that is tissues which are normally which are present in your body but which have a high proliferation level, like the skin or the epithelial tissues that are present in your gut. So these things also have repopulation. That is, you give a radiation dose to the skin or to the mucosa of the gut, they will start proliferating again. Now that is essential to reduce your side effects, but the same repopulation also happens in tumors. In fact, it has been seen that 3 to 4 weeks after you have started radiation, the repopulation happens in an accelerated rate, and therefore you'll have to give probably a higher dose to control the same tumor. So when does it start and what is the repopulation rate in different tissues are two important aspects of this question. Now, repopulation occurs in different speeds depending on your tissue. So the early responding tissues, that is the stem cells, the GI, that is gastrointestinal tract epithelium, the mucosa that is present in the mouth or the skin, they start repopulation at 4 weeks. So if you increase the treatment time over this amount, that is you finish the treatment course over 6 weeks instead of 4 weeks, you may reduce toxicity in these tissues. But there are also late responding tissues, that is your kidneys, your blood vessel endothelium, or your nerves. These things start repopulation much later. Therefore, prolonging a treatment course is not expected to reduce toxicity in these tissues. Now, tumor cells also have an accelerated repopulation, and they start usually at 4 weeks after your radiation. Therefore, we have to strike a balance wherein you don't prolong the treatment duration too much so as to allow accelerated repopulation in the tumor cells to cause progression, and you do not want to shorten the treatment course to such an extent that the patient becomes too sick because of the acute side effects of radiation. So there has to be balance that has to be maintained. The point that has to be noted is this accelerated repopulation occurs both in tumor cells as well as in normal tissues of the body, and both of them start around 4 weeks from the start of radiation. So this accelerated repopulation is seen that it is mediated through radiation-induced receptor activation as well as cellular growth after the radiation exposure starts. So this is just a graph which is showing different tissues, having repopulation. So as you can see, these are experiments on mouse primarily. So mouse jejunum, that is the epithelium of the gut, they start repopulating at 7 days post radiation. Here you can see the human skin that is actually starting at 28 days, around 28 days, so around 4 weeks. What you can see here is that the liver and the kidneys, the spinal cord, these are all late or slow proliferating tissues. These things start having repopulation maybe 8 weeks post the start of radiation. So by that time, the repopulation starts in them, you have already finished the course of radiation. So the current standard treatment durations try to confer benefit by allowing regeneration of acute responding tissues, reducing toxicity. There have been attempts to deliver radiation much more quickly, that is, finish off treatment within 4 weeks, within 3 weeks instead of 7 weeks. They have led to severe and dose-limiting toxicities. We do use some amount of growth factors, GCSF most commonly, to protect tissues from radiation injury, but most of them have not actually worked. So, this repopulation has been, in clinical sense, shown to affect tumor control in head and neck squamous cell carcinoma and cervical cancer. Local control is reduced by 0.5% for each day that the overall treatment time is prolonged. So in clinical sense, we have certain durations that are fixed for completing treatments. For example, in cervical cancer, we need to finish the entire course of radiation treatment within 8 weeks. So say because of toxicity, the patient has developed, you prolong this 8 weeks to 9 weeks. What happens is the tumor control, because you've extended by 7 more days, the local control of tumor is reduced by 3.5%. So that would continue if you prolong it further. So maintenance of this treatment time is essential, but at the same time, toxicity also has to be reduced. Right. So clinical implications, as already explained, it is for differential sparing of normal tissues and tumors. So the next R, that is the fourth R, is reoxygenation. Now, has the concept of oxygen fixation been explained already in the previous lecture? Okay. So we'll move on to the fourth R, that is sensitivity of radiation with oxygen. So sensitivity to radiation increases with the presence of oxygen. So this is just a photograph showing the indirect action of radiation. So a photon hits a water molecule, releases an electron, it produces a hydroxyl molecule, and then it produces a free radical. Now this free radical goes and damages the DNA. Now this damage that has happened to the DNA is fixed or made constant by attachment, by the presence of oxygen. So if oxygen is present, the DNA damage gets fixed and therefore the cell dies. In the absence of oxygen, there is more likelihood that the damage gets repaired. So therefore, if there is hypoxia in tumors, that is reduced oxygen amount in the tumors, then it is expected that the DNA damage in tumors can get repaired. So hypoxia occurs in tumors by 2 ways. One is acute hypoxia and the other one is chronic hypoxia. So we'll look at chronic hypoxia first. So because that is more consistent. So this is the blood vessel through which blood flows. So you have got the arterial end and then you've got the venous end, that is just one vessel where the blood is flowing. And then you've got the tumor cells all around. Now what has been seen in experiments is that the oxygen diffuses to a thickness of 70 micrometers from where the blood vessel is present. So if the tumor's size or the distance from the blood vessel to the last tumor cell is more than 70 micrometers, it is expected that tumor cells are hypoxic and some of them are anoxic. So if you can see, these are areas where oxygen is reaching. Now these white areas are regions where oxygen is not reaching completely, little bit less. And the black areas are regions where there is no oxygen. So the black areas are regions where there is no oxygen, therefore the cells die. But the white regions are the regions where oxygen concentration is not so low that the cell dies, but it is low enough to cause radiation resistance. So this is the rim that is going to survive after a dose of radiation is given, because these are cells that have no oxygen and therefore they die. These are cells that have oxygen, and therefore radiation does have sensitivity and therefore kills the cells. And this rim is what is going to cause resistance. The other thing that can happen is acute hypoxia. Now, the cancer growth that happens also includes growth along, there are blood vessels that are also getting formed as the cancer grows. But these blood vessels, as I'll be explaining later on, have leaky endothelium. And the endothelium is not formed in a proper way. So it is what we see is that some of these cancer vessels get occluded suddenly. That is, you see it today, you take a scan or you see it in the microscope, it is allowing blood to flow through, and in a few hours it is getting closed. So this transient occlusion of blood vessels can cause acute hypoxia. So what happens because of this? Because of, as explained, malformed structure, some places at some time points there is temporary closure and therefore hypoxia, and therefore the time when this happens, if you deliver radiation, because of hypoxia, there can be radiation resistance. So there can be two types of radiation resistance because of hypoxia. One, chronic, because of the distance from the blood vessel of the tumor cells, and second is because of temporary occlusion of blood vessels. So this leads to radiation resistance. This is again just a picture that is showing that this is the blood vessel. Now this is chronic hypoxia, as explained. This part has oxygen, this part has less oxygen, this part has no oxygen. So this part dies after radiation, this part remains after radiation. And this is acute hypoxia, that is suddenly the vessel has closed down, and therefore this part, if you deliver radiation at this point, is going to be resistant. So how do we utilize this problem for our benefit? So as you have seen that tumors have a combination of hypoxic as well as normoxic cells. Therefore, you give radiation now, what happens is the cells that are radiosensitive because of oxygen presence die, and therefore the tumor starts shrinking. So in this picture, as you can see, if this part shrinks or dies, and then it is eaten away by macrophages and taken away. Now this rim of tumor tissue that was hypoxic becomes closer to the blood vessel, and therefore this now would become oxic and therefore sensitive to radiation. So again, when you fractionate the radiation, instead of giving it a single dose high fraction, the normal the cells which have oxygen die first, and then the cells that have less oxygen become oxygenated and then die when the next fraction of radiation is delivered. Similarly, acute hypoxia also gets help because this vessel closure is transient. So this fractionation may be, the vessel is closed, by the next fraction this vessel opens up and therefore the cells die again. So this is just again explaining the same thing, that if there is a fast component and a slow component, this fast component is acute hypoxia, occurs within hours. The slow component, chronic hypoxia, happens within days. So this reoxygenation helps us to limit most of the radiation, radiation doses to 60 to 70 gray for killing most of the tumors. So what are the mechanisms by which reoxygenation happens? One, by opening the blood vessels. Second is by reduced respiration of lethally damaged cells. That is the anoxic cells, they don't use any oxygen. The third thing is that the dead cells get resorbed and the distance from the capillaries, that is the blood vessels to tumor cells, increases after a few days. So to see this mathematically, there is a concept called the oxygen enhancement ratio. So oxygen enhancement ratio is the ratio of radiation doses in hypoxic versus aerated condition to get the same biological effect. To kill the same number of cancer cells, how much radiation is required in hypoxic condition versus aerated condition. So for X-rays or gamma rays, it is about 2.5. In other words, more than twice the number of cancer cells would be dead if there was oxygen compared to if there was no oxygen. So how do we use this in the clinical sense? One, as I already said, you use it by fractionating. But apart from that, you also have certain things. That is hyperbaric oxygen. Second is carbogen and nicotinamide. Third is maintaining hemoglobin levels. And fourth one is hypoxic cell sensitizers. So out of these four, the greatest clinical evidence is available for number 3 and number 4. So in cervical cancers, any patient comes in with a low hemoglobin, it is necessary that you maintain the hemoglobin level before you start radiation. That is by blood transfusion or other means. That improves cancer control. Hypoxic cell sensitizers in head and neck cancers, nimerazole, which is a nitroimidazole, which mimics oxygen action in hypoxic regions, has been shown to improve survival when you combine with radiation. Number 1 and number 2 have not have a great utilization as of today. Hyperbaric oxygen has been shown to reduce radiation toxicity, but has not been shown to improve cancer control as of today. So to summarize the 4 R's and the fractionated radiotherapy, repair, that is sublethal damage repair, or in other words, the difference between the sublethal damage repair capacity of cancer cells versus normal cells, reassortment, that is redistribution along the cell cycle, and reoxygenation that happens in cancer cells all benefit by fractionation. That is, you divide the total cancer dose into multiple fractions. These 3 things would help. Repopulation is the negative aspect of fractionation by radiation, but this repopulation is essential for reducing acute toxicity in normal cells. Repair occurs in both normal cells and tumor cells. Reassortment, mostly tumor cells, but in some normal cells as well. Reoxygenation, only in tumor cells. Repopulation, mostly in tumor cells, but also in early responding tissues, that is epithelium and skin. So, as we have seen, these are the 4 R's. The next R that we are going to look at is intrinsic radiosensitivity. So, we should not assume that all tumors, all different types of cancers are going to get killed by the same dose of radiation. So each tumor has, or each cell has, a different radiosensitivity. So in this picture, you can just see the difference in radiosensitivity in the normal cells. So lymphocytes are the most radiosensitive cell that is present in the human body, and then we have in the decreasing gradation, blood cells, epithelial cells. So the muscle cells and the nerve cells are quite resistant. In 1906, just when the science of radiobiology was developing, scientists Bergeni and Tribondu, they found that the sensitivity of radiation was most if the cells were rapidly dividing and they were undifferentiated. So, sorry. So then came the law of Bergeni and Tribondu. What they said was undifferentiated cells and cells that are proliferating fast, these are killed more by radiation. So this is also seen in the clinical picture, that we see undifferentiated carcinomas responding better to radiation rather than things like verrucous carcinoma, well-differentiated carcinoma, sometimes being a little resistant to radiation. So this is the law that was stated: the radiosensitivity of a tissue is directly proportional to its reproductive capacity and inversely proportional to its degree of differentiation. So the cause of radiosensitivity, different hypotheses have been given. One is high metabolism of tumor cells is probably a factor in radiosensitivity. Increased or unstable vascularity probably leads to rapid growth and therefore more sensitivity. So multiple theories have been put forward. Now, different tumor types have been divided into 5 categories based on decreasing sensitivity. So category A includes lymphoma, myeloma, neuroblastoma, and category B, different tumors. Now category E includes the radio-resistant tumors, that is melanoma, osteosarcoma, and renal cell carcinoma. So even in clinical sense, we have minimal, as radiation oncologists, we have less role in tumors like melanoma, osteosarcoma, or renal cell carcinoma, but we have a larger role to play in cervical cancer, small cell cancers, or squamous cell cancers. So apart from this classification, there have also been some other classification. Now, this is just for theoretical explanation. There is a Rubin and Casaret's classification of cellular populations based on their reproductive and functional characteristics in various cell lines. They have actually divided into 5 groups. One is the vegetative intermitotic cells, that is rapidly proliferating cells. Now, this would include the stem cells, this would include tumor cells as well. And then the second group is differentiating intermitotic cells. They are having some differentiation, but they are still rapidly proliferating. The third one is multipotential connective tissue cells. The fourth one is reverting post-mitotic cells, and the fifth one is fixed post-mitotic cell. So the vegetative intermitotic cell is highly proliferating, lowest differentiation, but the fixed post-mitotic cell is well differentiated and proliferating much less. Therefore, we come to the final synopsis regarding fractionation. So fractionation basically exploits the difference in recovery rate between normal, acute, and late reactive tissues and the tumors. The 3 things, repair, reassortment, reoxygenation, help us in improving cancer control. The 4th one, repopulation, is utilized to reduce toxicity, and then intrinsic radiosensitivity helps us to define which tumors would respond to radiation and which tumors will not respond to radiation. So then coming to the clinical part of it, so we have seen the radiobiological basis of fractionation. Now we, in the clinical sense, what do we do? So one thing, the classification we have is the types of fractionation. The commonest fractionation that we use is called as conventional fractionation. So here, the tumor is given 1.82 gray per fraction. It is usually 1 fraction per day and 5 days a week. So you give radiation at this dose for 5 days a week, and then you give 2 days off, and then you continue for 5 days onwards. So different tumors have different, because radiosensitivity is different, the tumor location is different, the toxicity profiles are different. So different tumors would have different total doses being delivered, but the doses are given in this conventional manner, that is 2 gray per day for 5 days a week. And now, what we have seen is, what we know is that the fractionations can be changed to play with the 5 R's of radiobiology. So one we do is hyperfractionation. What we do here is, we reduce the dose per fraction. Instead of giving 2 gray, it is about 1.2 or 1.4 gray per fraction. And then more than 1 fraction is given per day. So here, if you see, if you give, say, 2 fractions per day, it is usually at a gap of minimum of 6 hours. That is because we have seen that the sublethal damage repair would require at least 6 hours to happen. So to give normal tissues the time to repair themselves, even though we are giving 2 fractions per day, there's a gap of 6 hours given. And apart from lower dose per fraction, more than 1 fraction per day, the total dose is slightly increased. Now, what do we expect from this? We expect a lesser late toxicity. This is because late toxicity, that is toxicity that happens years later, is based on dose per fraction. So here we have reduced the dose per fraction. Slightly higher acute toxicity because the weekly dose rate, the rate at which we are giving the dose, is slightly higher. And then we expect better tumor control because the total dose is higher. The second thing that we do is called acceleration. Now, in the acceleration, we have different types of acceleration, but let us discuss the simplest type, in that the dose per fraction is kept the same. The fraction per day is 1, and the number of fractions per week is increased to more than 5. In some trials, it's been 6, some trials even 7. That is daily you continue to give radiation. So in this, what we achieve is that the overall treatment time is reduced. So instead of giving 5 days per week, if you go 7 days per week, instead of 7 weeks, probably in 6 weeks or 5 weeks, we are finishing the treatment. Now, radiobiologically speaking, this is going to help in us overcoming the accelerated repopulation that the tumor is going to start. But with acceleration, you have higher acute toxicity because you're giving more dose in a weekly basis. The late toxicity is going to be the same because the dose per fraction is not changed, and a higher tumor control is expected because the repopulation is expected to be overcome. The third thing, the difference from conventional fractionation is hypofractionation. Now, hypofractionation, in this, the dose per fraction is increased. Now, if you increase the dose per fraction, if we go back to the initial cell survival curve, just to so we are using X-rays, and therefore there is a shoulder region and then there is an exponential region. So if you increase the dose per fraction, what you expect is a much higher kill compared to just dividing them into smaller dose per fractions. So using that, by hypofractionation, we are expecting a higher tumor kill. But when you increase the dose per fraction, the late toxicity is also expected to be higher. So this hypofractionation is used in only those places where you'll be able to control doses in normal tissues. So clinical sense, it is usually used in prostate cancers and breast cancers. Anyways, so besides this play with fractionation, what other ways can we use this 4 hours or 5 hours to improve tumor kill? One is hypoxic sensitization, as we did see, nimorozole. Second, chemotherapy, as we did see, paclitaxel arrests tumor cells in G2M phase, and therefore it causes synchronization and therefore better kill. Cisplatin, it creates interstrand links in DNA, so the DNA damage that has happened becomes fixed. And then third one is by maintaining oxygen levels, that is maintaining hemoglobin before starting radiation. The fourth one, that is combining immunotherapy along with radiation, is something that is coming up. We have some data coming up in the last 2-3 years which have proven that combining immunotherapy may help. So we have seen fractionation, we have seen the hours of radiobiology, but what we need to understand is that all these things appear very very simple on if you look at it, and you would expect that 70 gray, if given to a tumor, is expected to kill the tumor. But what we see in actual clinical practice is the same size tumor, same patient parameters, we treat with the same kind of radiation. The tumor behaves differently, the patient behaves differently. Some patients get cured of cancer, other patients don't get cured of cancers. So there is a heterogeneity in the response that is seen with cancer control. Now this has to be explained further. Now this cannot be explained by what we have seen previously. So that is why we need to know a little bit of what a cancer cell is, a little bit of tumor microenvironment, because this is probably the future in lab research as well as clinical research. So a cancer cell is typically seen to have uncontrolled cellular proliferation, unbalanced cell division, loss of apoptosis, loss of certain special functions. It is seen to have abnormal angiogenesis, that is vessel wall production, vessel wall structure. It is seen to invade tissues. It is shown to metastasize, that is go from one area of the body to another area of the body. Tumor cells have a high glycolytic rate. They have a very minimal Krebs cycle that is happening in them. And most tumor cells also show some form of hypoxia. So what do they have? Because of all these things, they have sustained proliferative capacity signaling, they evade growth suppressors, they have invasion metastasis, they have replicative immortality in certain sense, they have more angiogenesis, new blood vessels getting formed, quickly formed, and they resist cell death. And some of the emerging hallmarks of cancer are deregulated cellular energetics, immune evasion, and tumor-induced inflammation and genomic instability. So genomic instability and mutation means there is an aberrant DNA or DNA structure that is seen in almost all cancer cells, and they acquire a abnormal number of chromosomes. This leads to production. Sometimes this mutation leads to production of neoantigens, that is neoantigens that are not seen in the human body. If you look at the cancer cell in a microscope, difference is seen. In the cancer cell, instead of a single and small nucleus, you've got multiple and large nucleoli and nuclei. You see chromatin clumps in the nucleus. You can see dark staining in the nucleus and an irregular nuclear border. You have nuclei that has become irregular and beginning to fold. Certain specific nuclear matrix proteins are absent, and perinuclear compartment is present. So in the microscope, cancer cell looks different compared to normal cell. The cancer cell also has a dysregulated cellular energetic. That is, it exhibits in general a defective Krebs cycle and derives little or no energy from it. It derives almost all the energy from glycolysis and derives most of the energy in the absence of oxygen. Now, this is the dangerous part, that even in the presence of hypoxia, the tumor cell will get its energy, will continue proliferation. There are some differences in the mitochondria. That is, there is mitochondrial DNA mutations as well. So, in the mitochondria, you have got some molecules that are upregulated and downregulated, which actually maintain homeostasis, that is, promote apoptosis. But in the cancer cell, we see there is downregulation of ROS, BAX, there is upregulation of BCL2 and SOD, catalase, etc. And therefore, again, mitochondrial function is also mutated. The plasma membrane is defective. So you see in the tumor cells, there is change in the plasma membrane fluidity. That is, the covering that is present around the cell has defective opening and closure that is happening. And therefore, the presentation of antigens in the cancer cell is also defective. So instead of having antigens on the top of the cell membrane, the cancer cells have antigens within them. Therefore, the immune cells many times don't recognize them. And therefore, the cancer cell, this defective plasma membrane leads to immune evasion as well. And the second thing that it can cause is that the changes in plasma membrane leads to better motility and ability to infiltrate basement membranes and cause metastasis. Now, this is about individual cancer cell differences. That is, difference between a cancer cell and a tumor and a normal cell. There is something called as the tumor microenvironment. That is, there is an ecosystem of a series of a combination of cells that are present around the tumor. Now, the radiation that you deliver to the tumor is not only going to affect the cancer cell per se by all these factors that have been explained before, but they're also going to affect the cells that are present in the tumor microenvironment. So how these cells get affected is still an area of active research. There is a huge amount of heterogeneity in individual tumors regarding this tumor microenvironment, and therefore we do not have a single answer to this question as to how radiation affects these cells. So TME basically is a functional ecosystem of tumor and stromal elements that interact through signaling molecules. Now, it includes stroma. The stroma includes mesenchymal cells, vascular, and immune cells. So in the tumor microenvironment, you have these things. One, neoplastic cells. Neoplastic cells includes the cancer cells, and it also includes cancer-associated fibroblast. Then it includes immune cells, lymphocytes, and macrophages. Then you've got the resident cells, that is the fibroblasts, the endothelial cells. They form the baseline. They form the base in which these cells reside. Then you've got certain secreted factors, cytokines, matrix altering enzymes, mostly matrix metalloproteinases, VEGF, vascular endothelial growth factor, fibroblast growth factor, PDGF, TGF, etc. And within this tumor microenvironment, you have got certain features. There is hypoxia, there is low pH, that is acidic conditions, and there is hypoglycemia. So the cancer, the tumor microenvironment has certain predominant cells. I'll just touch some of them. One is the cancer-associated fibroblast. It is the predominant cell type in stroma. It is responsible for structural architecture of the extracellular matrix. It produces multiple proteins, collagen, periostein. It has been seen that it promotes cancer proliferation and infiltration by producing certain factors. Similarly, it promotes angiogenesis as well. And there is growing evidence that it leads to epithelial mesenchymal transition. Now, this is something that is essential for tumors to become metastatic, that is, move from one place in the body to another place in the body. So the cancer-associated fibroblasts are a very important part of tumor microenvironment, which we on whom we have to assess the effect of radiation as well. The next cells that are important are the immune cells. Now, tumor microenvironment is filled with a lot of immune cells. Now, sometimes some tumors don't have any immune cells. Now, those are called cold tumors. Some tumors have a lot of immune cells in them. They're called hot tumors. Now, there is heterogeneity in this. Even within a type of tumor, that say cervical squamous cell carcinoma, in one patient you may have a lot of immune cells, in the other patient you may not have immune cells. Now, these are all areas of active research where we do not know why these things happen, but we need to find out why these things happen. So there are cells which have anti-tumor effects in the tumor microenvironment, that is M1 macrophages, the T helper cells, the cytotoxic T cells, some of the antigen presenting cells, and natural killer cells. And there are some pro-tumor effects, that is, some immune cells will allow the tumor to survive and prevent immune action, that is, they will help in immune evasion. So those are the M2 macrophages, the regulatory T cells, and T helper cells. So in addition to these fully differentiated immune cells, you also have a multiple differentiated myeloid progenitors that are present in the tumor microenvironment. So macrophages, as we did see, white blood cells, there are two types, M1 and M2. Now, M1 macrophages help with immune surveillance. That is, these may actually help in killing tumors. The M2 macrophages, they suppress immune action against tumors. So the balance between M1 and M2, needs to be modified, or we should attempt to modify them so that you have more immune cell kill and you have less immune evasion. Then you've got T lymphocytes. Again, you've got two groups. One is the cytotoxic, the other one is the regulatory. Now, the cytotoxic T cells, they actually produce interferon gamma. They lead to apoptosis of the individual cancer cell. These cancer cells then help in producing antigens. Then these are presented to antigen presentation cells, and then leads to tumor killing. So CD8 cells, good cells, help us in cancer control. Regulatory T cells, bad cells, they help in proliferation of tumor cells. Then you've got antigen presenting cells. Now, these again have two types, MHC1 expressing and MHC2 expressing. I'm not going into the details. And you've got natural killer cells as well in the same stroma. These are innate immune cells. They are important in halting tumor progression. The next part is endothelium and pericytes. As I had initially told, tumor cells, when they grow, they produce a lot of blood vessels, but these blood vessels are defective. The endothelial cells usually have a pericyte covering around them, but in the tumor cell, the endothelial cells lack a pericyte covering. They have leaky tight junctions, and therefore there is leakage that is happening around the blood vessel. The pericyte endothelial cell, there is a signaling network which can contribute to tumor metastasis as well. And the last one is that you've got an extracellular matrix. The extracellular matrix, it is the scaffold on which these cells survive, the stromal cells, the cancer cells, immune cells. So these extracellular matrix also have a lot of cytokines that are present in them. These things also have the capacity to initiate and produce signal cascades within the TME. The extracellular matrix also houses the cancer stem cell, which are known to be more radioresistant, and they also have certain inflammatory components which help in metastasis. So as you can see, the tumor microenvironment has multiple things, and all these things seem to be stimulating tumor metastasis. So to summarize this, fractionated radiation therapy, along with radiation response modifiers, they have brought us a long way in cancer control. So we have understood how to use these 5 hours of radiation, radiobiology, and along with technological advancement in radiation machines and CT scans, we are able to deliver much higher doses of radiation to the tumor than we were able to maybe 40, 50 years ago, with minimal toxicity to the patients. But there is a large amount of intratumoral and intertumoral heterogeneity due to the tumor microenvironment, which is leading to different varying responses to radiation. So it is time for research into effects of radiation on the TME components and identify ways in the lab to modulate them with radiation or with any other drug, and then translate them into clinical validation. Thank you.
So, yeah, Professor Sanjay.
Thank you, Dr. Prashant. It was very nice lecture and really enjoyed it.
So, sir, a lot of questions in the text box. Should I read or you will read?
I think you can go ahead. I have seen the questions. I will intervene if required.
Okay. So, Dr. Sangeeta Chaudhary has asked a question that is there any difference? Sorry, just
between chemo and the radiotherapy toxicity.
Yeah,
that's what she means.
Yeah. So, is there she's asking, is there any difference between radiation-induced toxicity versus chemotoxicity?
Yeah, absolutely. So radiation therapy for all practical purposes is a local treatment. So you're going to have toxicity which is in the local site. So if you give radiation to a head and neck tumor, you're going to have toxicity in the head and neck region. That could be mucosal inflammation, there could be pain in the throat, there can be increased salivation or blackening of skin. So that is going to be the type of toxicity for radiation. For chemotherapy, because it is a drug that is injected into the blood in most cases, it is going to be an overall effect on the body. So patient is going to have hair loss, patient is going to have reduced blood counts, patient is going to have vomiting, patient is going to have fatigue. So the side effect profile is most of the times totally different.
Apart from the physical or as you mentioned, demographic details, what about innate or adaptive immunity involved in it, and is there anything that can be very clearly defined?
Right. So, are you speaking about the body's innate and immunity, or the tumor microenvironment?
Yeah.
Right. So we have data across tumor sites which show that if there is lymphopenia, that is reduced lymphocyte count in the body, the cancer control is poorer. So a patient having good lymphocyte counts, he's expected to control cancer better versus a patient who is having low lymphocyte counts. There are multiple hypotheses that we have generated for that, but clinically, this is what has been proven. That is, you have low lymphocyte counts, that is poorer immunity, poorer cancer control, better immunity, better cancer control. With radiation, not speaking about immunotherapy.
Right. Okay, and because ultimately even the resultant of radiotherapy is lymphocytopenia.
Not always, because if you are irradiating, say
many of the times, right?
Yeah, so if you're irradiating a child with craniospinal irradiation for medulloblastoma, so you're irradiating a huge amount of his bone marrow, his or her bone marrow. So in that case, yes, it will induce lymphopenia. But say you're irradiating an adult in, say, thoracic region for, say, lung cancer, that is not going to induce lymphopenia directly. So it is not going to, well, radiation will not induce lymphopenia in all cases, only in a few cases.
All right. Okay.
Professor, yeah. Go ahead.
No, I said thanks. Yeah.
Okay, so Dr. Harisharan Mishra has raised the hand. Sir, you can ask the question.
Yeah, so I think thank you, Prashant. It's very good coverage which you have done on different aspects of it. I just have two small concerns. I mean, curiosity question, because I'm not from the field.
Right.
So you said that this oxic condition of different cell population is something known, whether that differs, or let's say G1, G2, M, S, do they also have some mechanism to have different oxygen or redox regulation? Is something known, or?
I didn't get your question, sir.
Yeah,
so are you asking if this oxygen enhancement ratio would be different for different cells?
That's correct. Is something known that if you fractionate the population, let us say G1, G2, M separately, and try to
yes, so that is absolutely there. So the oxygen enhancement ratio is expected to be maximum for S phase cells. It is going to be least for the G2M phase cells. So already they are highly radiosensitive. Oxygen enhancement is going to be lesser.
No, Prashant, I think this is what I am trying to get the answer. Since they are highly radiosensitive, so it shouldn't be implicated that they have got a different redox. Whether they have different redox regulation, that is how they are radiosensitive. This is what I am looking for.
Okay, sir. Okay.
I'm looking other way around.
Okay, sir. Okay.
Yeah, and one more thing, you said that fractionation, the dose fractionation, fractionated dose which is given, is it effective because of DNA repair, or it's a cell population dynamics? What is the mechanism? How does it help with fractionated dose? Because you implicated that it's because some DNA repair kinetics something could be that's implicated. Is something has something been done that this fractionated dose is effective because of this cell cycle population dynamics? Because 6 hours.
Absolutely. Cell cycle population dynamics, of course, play a role. The sublethal damage repair is just one component of it. Okay. Of course, fractionation would help in the cell cycle dynamics would favor fractionation as well. So it is combination of both.
Thank you.
Okay, so there is another question which says that what do you mean by unstable vascularity? Is high vascularity leads to more radiosensitivity due to the availability of high oxygen levels? I think you answered, but she needs more clarification.
Yeah. So what we mean by leaky vessels is that these vessels are unstable. That is, these vessels may close down sometimes, these vessels may open up sometimes, and these vessels don't have the proper mechanisms for proper oxygen diffusion to occur. So you need to have proper vessel so that proper oxygen diffusion as well happens. And therefore, when you have a leaky vessel and not improperly formed vessel, the cancer cells are expected to have slightly higher chance of hypoxia. So, and therefore, more chance of radiation resistance.
Okay. So another question comes that you have mentioned tumor cells does not recover from radiation induced damage unlike normal cells. I don't think you mentioned it.
Yeah.
I think it's a little bit confused.
No, I didn't
let me complete. It's a half the question. Then she says, then why prolonged radiation greater than 5 weeks leads to repopulation of malignant tumor?
Right. So first part of the question, I didn't mention that there wouldn't be any repair. I just mentioned that the repair capacity of cancer cells is lesser compared to normal cells. So normal cells will repair better, cancer cells will repair, but they will repair much less. So that is one point. And second thing is that the repopulation that is happening, that is by release of certain inflammatory cytokines. Now, I don't think we should combine sublethal damage repair with repopulation. Those are mediated by completely different mechanisms.
Okay, and then there is another question. If the sensitivity to radiation increases with oxygen, a radiation dose to tumor will do more damage to the surrounding normal cells in the aerobic state. So, I don't know, you can address it, but...
Yeah, that's an interesting thing, but the...
Actually, I can, maybe I can say...
Yeah, please go ahead.
But see, it is not the case. See, when we are comparing, we are mostly comparing among the tumor cells only. So this question arises, but yes, we have been finding that all of us know that the normal tissue injury is there, normal cell injuries is there. And that has been happened that when we have increased the dose of the due to the say hypoxic condition to the tumor, then the normal cells get damages more. And there is a whole lot of calculation is there where it is clearly shown that what is the dose which is required to the tumor, and what is the beyond that, how much is the normal cell will be affected. So, but the kind of comparison you are asking, probably this kind of answer we don't have. Maybe we are not clear, we cannot clearly answer it. Or fine. So is there, Dr. Tulkar, can you add something to this question, or I don't know?
No, I think it's nicely elaborated by yourself and Dr. Prashant. So this damage to normal cells will occur anyway. That's why this Prashant has given some examples like head and neck tumor treated with radiation. They will have mucositis and that type of changes. So normal cells do get exposed, they get damaged. But that's what they do, given radiation, 3D conformal and sophisticated radiation technology, so as to altogether avoid radiation to normal structures and normal tissue and concentrate only on the tumor cells. So that's their continuous endeavor of the science of radiation oncology, how to prevent this damage by using different techniques and different type of radiation.
Yeah, and then we have a question here that herbal medicines having antioxidant properties are given orally to cancer patients undergoing radiotherapy. Is it going to hamper the cancer cells killing? What is your experience?
Yeah, so there is, so I would like to quote two evidences for that. One thing is that erythropoietin that was expected to stimulate production of hemoglobin, increase the blood oxygen levels. Erythropoietin has been used in a randomized clinical trial by the Tahanka group in head and neck squamous cell carcinomas along with radiation. And what it was found that this erythropoietin actually worsens the survival rather than improving the survival. So the use of these kind of drugs, erythropoietin or antioxidants, say vitamin E or vitamin C, in clinical setting, they are not translated into actual cancer control. There is evidence that combination of alpha tocopherol, that is vitamin E, supposedly acting as antioxidant, and pentoxifylline reduces radiation-induced fibrosis. That is, it is reducing some toxicity but no data to say that cancer control improves.
Here I can add something. If you remember, Prashant, long ago when Dr. Julka was there in the department in AIMS, there is some study using curcumin, that is haldi,
yes,
CA service patient. So there were some encouraging results, but I think it was not significant, so did not transfer into medicine. But it showed some promise. The haldi, the curcumin trial. You are aware of that.
Some herbal medicines, like even recently we had this uplat, papaya powder extract. So that was going on till 2016-17. So that helped in improving platelet counts in patients. But again, it was not a consistent result that we are seeing with these herbal products.
Shant, I can also add it. I mean, this question that somebody is curious on this aspect. Maybe if you highlight little bit that normal cells and cancer cells, they have got, or tumor cells, they have got different redox metabolism. And that is going to correlate with the effect of antioxidants. If something of that kind of information exists, maybe that will be the most appropriate answer.
Right, sir.
Although we know that, I mean, that is why we have not been able to become taken care of all cancers by radiation treatment, etc. Because the radiation or chemotherapy, they all will have some side effects. But the point here that the benefit versus effect that is what being viewed in radiotherapy and chemotherapy. I mean, if I understand. But the better answer can come to such kind of curiosity that if we explain the redox metabolism of the normal cells and tumor cells, if they are different, I'm not actually aware of it. But that will be the perhaps the better line of explanation.
Okay.
That though there will be effect, but effect can be different because these two different populations may have two different redox metabolism. I don't know. If
right, sir. Even I'm not aware of it. I'll definitely look it up. So.
Okay, and then the next question is, in conventional radiotherapy, 2-gray radiation is given 5 times a week. However, in vitro conditions, a single dose of 2-gray radiation is mostly practiced. How much effective a single dose of 2-gray radiation causes? And can we observe significant, you just answer half, then I'll. So she's asking that in conventional radiotherapy, 2-gray radiation is given 5 times a week. Is it true? It's not always true, no?
No, when you call it conventional fractionation, it is true. When you call it conventional fractionation, it is true. But not all tumors are irradiated like this. Say prostate cancers, breast cancers, we give a huge, much higher dose per fraction, finish it off in 5 days time. Newer techniques called SBRT.
Yeah, because this Eric Hall and all other say textbooks, they talk about the 2-gray as a correct.
2-gray is just the term for when we use conventional fractionation, it has to be 2-gray per fraction. But that is not the practice across tumor types. So 2-gray per fraction in the clinical sense, only 2-gray per fraction is not going to be useful for any tumor. 2-gray per fraction is useful only in one indication, that is it is indicated when a child of neuroblastoma has a huge liver because of the disease hepatomegaly, and only in that case, to relieve his pain, 2-gray helps. Apart from that, in just for tumor control in gross disease, 2-gray alone is not going to be useful. It has to be 2-gray per fraction over 4 weeks, over 6 weeks, over 7 weeks. That is what is usually used. But of course, when you're using a small cell line where you have a defined number of cells and you want to look at events that are happening within that cell, obviously a single fractionation dose is given in the lab.
So she's asking that if we give the 2-gray radiation to the patient and then we give it to the cell line, so in the patient, can we observe any significant changes so that we can
we would observe some changes.
Combine it with the in vitro results or something?
Yeah, so we would see some changes, but the point is, how do we detect those changes? So if you have a 4 centimeter tumor, you are given 2-gray per fraction. Of course, some part of that 4 centimeter tumor, say 0.001 centimeter of it, is dying. But which part of the tumor that is, how do you detect it? There is no way to detect it. So that is the problem. Now, if you have a mechanism by which you can detect that, if this is the small part that is dying, you take it out and you compare it to the cell line, it should correlate to an extent, but again, the tumor microenvironment factors would modulate it to an extent.
So again, she's asking, Pragya is there only, so because she worked on that. So she's asking that, is it the right strategy? Because it is written in several papers, so that's why she's asking. That is it a right strategy to give a single dose of 2-gray radiation to cells in vitro to observe the effect of potential radiosensitizer drug? So the kind of thing which we do in xenograft or in even in the xenograft or in the case of cells. So in both cases, actually, she's asking. Yeah.
No, it would be useful because that whichever sensitizer that you're going to use, most likely it is going to be given along with all the fractions of radiation. For example, the drug Nimerazole, I was using that in one of the trials. So in that case, the drug used to be given before every radiation fraction. So because every radiation fraction is only 2 gray per fraction, if you want to look at the effect of the drug in vitro, obviously you will have to do it with a 2 gray fraction along with the drug. So it makes absolute sense to do it in vitro.
Yeah. So as you.
So, sir, here I guess you are talking about 2 gray of radiation in multiple fractions. So I'm asking if in the in vitro condition, we usually irradiate the cells with a single dose of 2 gray radiation along with the drug treatment. So is it the right strategy to irradiate the cells with just the single dose of 2 gray?
I think so. Because if you're, if you have the entire tumor microenvironment there, instead of just having, say, a HEP2 cell or a HeLa cell, you just if you have just a homogeneous cancer cell line, giving multiple fractions along with the drug is probably not going to give you much of an additive benefit. But if you're growing, I mean, I have no experience in lab research. I'm just extending my statements here. But if you're able to reproduce the tumor microenvironment in your lab, that is, you have fibroblasts, you also have endothelial cells, you also have immune cells in the cancer cell line, then it makes sense if you're irradiating it multiple times and then seeing what the effect is. But if you have just the cancer cells, I think single fraction should make sense, and then go ahead with clinical trials after that.
Yeah, can I get into? I think this curiosity should be addressed. If you have such curiosity, why don't you do fractionated dose treatment under in vitro condition and try to see what will happen?
Yeah.
So you have very good question in your mind, and why don't you do the experiment to actually provide the evidence?
Right, sir. We can do this.
I think that kind of curiosity should be addressed. And I can tell you that in BRC, I was the member of the DC. One of the PhD, our officer has actually done such kind of study.
Yes, mute everybody.
Yeah.
Sir, you go ahead. Yeah, Professor Mishra, you go ahead. Yeah.
I think I had muted myself. So
no, not you. There was somebody who was on road, some participant is on road.
No, it got muted. Anyways, what I'm saying that such kind of study has been attempted in BRC, where one of our officers, actually he had done the fractionated dose in the cell line system. And he has seen some observation, and that he led to actually address the question at the genome organizations, etc. Maybe that would be something else to talk about. But what I am saying that it's a good question which you have in your mind, and such kind of curiosity questions should be addressed by doing the experiment in the lab. Do that and then try to see what are you going to get it.
Right, sir.
Okay, so Dr. Prashant, I have a question, and because you have worked on the Nimerazole, so can you please tell us that because when we tried something with the Nimerazole, so we could not get much results. So what kind of a mechanism you have observed with the nimrozole?
Yes. My trial was basically in cervical cancer, where I was giving nimrozole along with chemoradiation, that is standard treatment, compared with standard treatment. So the idea was that a part of our cervical cancer tumors are hypoxic, and therefore you give nimrozole, it is expected to sensitize even those tumors and improve survival. My recruitment could not be completed because I had shifted from AIMS to Tata. But with the 20-25 patients that we had seen, obviously you cannot make any comparative results out of it. But only thing I can say is that nimrozole was tolerated quite well. There was minimal toxicity in terms of severe nausea. That is all patients developed. Apart from that, with the 25 patients, you didn't have much of a difference in control rates. But Dahanka group has worked a lot on this, and they have definitely shown improved overall survival in head and neck squamous cell carcinoma. There have been trials which have directly compared chemoradiation, that is considered standard world over, with only nimrozole plus radiation. So they have achieved equivalent survival with both chemoradiation and nimrozole. So for a country like India, where chemotherapy is costly, people are poor, nimrozole is hardly 10 tablets cost you 50-60 rupees. So it should be researched further, particularly in.
I think there are some early results on nitroimidazole which talks about its toxicity, very high toxicity or something.
No, those are different nitroimidazoles. So if you use mesonidazole, so nitroimidazoles are two groups, 2 nitroimidazoles, 5 nitroimidazoles. So if you use mesonidazole, there is increased chance of neuropathy that is happening. But with nimrozole, you don't have neuropathy happening. You have only severe nausea and vomiting that happens, but nothing else. So nausea vomiting can be managed with drugs.
Okay, Dr. Sangeeta Chaudhary has a question that in vitro constructing TME will be challenging. So I think.
Yeah, I mean that
because you are not an experimentalist. So
comment on the previous.
Yeah, so it will be obviously quite a challenging thing, but people are trying to work on tumor microenvironment, Professor Chaudhary, because what people are trying to do that they are trying to make either the say PDX patient-derived xenograft, and they're trying to make the say orthotropic model of xenograft. Then they are trying to make 3D organoids and spheroids where they are only using say single cells or 10 cells to develop the different clonal spheroids from the one tumor, and then they are trying to say consider it that these are the different heterogeneous cells and they are having the different tumor microenvironment, and they are trying to treat with the them at the different radiation dose in the at the different say chemotherapeutic drugs. So a lot of I would say that the 3D organoid and this thing has started taken a lot of people are working in this area, and so a lot of work is being done in this area. And I would say that people are claiming also that they are able to find the difference there. But yes, at the clinical level, it was translated or not, that I can't comment actually. Okay, is there any other question? Because I don't see our next speaker here. He has to join from US. So, but he said he will join. So if anyone else want to have some question, you can ask. Professor Prasanna is here. Okay. Yeah. So fine. So Dr. Tilkar.
Yeah, I think I will hand over to my co-chair. Maybe he can introduce the speaker. Professor Misra?
Yeah. So, I would like to thank, but Dr. Prashant and Dr. Tulkar, I will request you that if you can stay for another lecture, it's nice. And it was really nice lecture by Dr. Prashant, and he has really very, being a clinician, he has put the cancer in a very simple manner and very nice manner, and very, with lot of clarity he has shown it. So I would just like to ask Dr. Prashant that you may just tell people that what is your area, because maybe certain people would like to collaborate with you. Some young students may also come to know about it.
Yeah, so presently I'm working on prostate cancers, but that is purely a clinical trial that we are working on. In the immediate future, our group is planning to work on signet ring cell carcinoma of rectum, where we are planning to look at effect of radiation and immune changes in the tumor microenvironment. So that we are trying to collaborate with our lab in-house in our institute. And we are also working on effect of we are doing a series of meta-analysis where we are finding out how individual doses to the spleen to different lymph organs are affecting the rates of lymphopenia and cancer control. So that is a series of meta-analysis that we are doing. So three things: signet ring cell cancer rectum, prostate cancer, and immune system and radiation. These are three things that we are working on right now.
Okay, so you have, is that in the Tata Memorial Cancer Center you have you have experimental labs also, because it's a new one, I think.
Yeah, so we have some labs. We have labs that can identify drug levels. We have labs for doing basic tumor microenvironment assessment, like immune cells, fibroblasts, and all that. But for further things, we sometimes send samples to Tata Memorial, Bombay. So our lab here has limited capacity, but we probably would expand in the future. And the other two are clinical trials that would not have any lab association. And the meta-analysis is done with some of my colleagues in the US. So that is just, it is a meta-analysis, so it's easy. You can sit at home and do it.
Okay. I think, Doctor, so that's nice, and maybe we can collaborate. And a lot of people, there are certain faculties in we here who are working in head and neck cancer, and there are other in School of Life Sciences also. We have a couple of faculties who are working, and we have Rana Pratap Singh. You may be knowing it. I don't know him, and so yeah, it's good if we can and if there are other students also here, they can. Dr. Prasanna joined, and then he said that I'm joining in 5 minutes. Maybe he had some problem. So he's coming, and
Prashant, can you exit your presentation now?
Yes, sir. Yes.
Yeah, good one.
No, you have exit. You have exit.
Has he joined or no?
No, he joined, and then he wrote a message that I think there was some problem, and he said
enough. He's still having 11 minutes to go, so not to worry. We can continue having more discussion. Let's know a little more about Dr. Thullar's work, what he has been doing actually, since we are meeting first time.
Yeah, great. That's
I am a clinical radiologist in the cancer hospital. So my main work is basically patient care and teaching to postgraduate students of radiodiagnosis, radiology students. My research was mainly in the different areas, breast cancer. I was fortunate to start few diagnostic procedures, basically clinical procedure like biopsy, stereotactic breast biopsy for small non-palpable breast cancer, And for prostate, I had some work like MRI spectroscopy and multiparametric diffusion weighted MRI. That work is still going on for early detection of prostate cancer and guided biopsies. So that was my work mainly in the area of breast cancer and prostate cancer.
More on the clinical aspect of it.
It's more primarily the clinical aspect.
Yeah, there has been one very spokesperson question. I thought of just putting it, we are doing a lot of monitoring of radiation in the environment. So that is one aspect which are concerns in different regions, particularly in BAE where I am belong to. Our column monazite survey kind of projects, etc. we have in Kerala. So there we monitor the level of radiation and its effect on the human population. So that is one aspect. The other aspect that we are giving radiation to even human beings, in patients, etc. So some spokesperson, they keeps on asking such question that on one hand you are so much concerned for millisieverts level of radiation. On other hand, you are giving great level of radiation to human being. So how should we address that question? This is more of the NGOs kind of question.
Like in clinical scenario, Prashant can address that better.
Yes, so for that we just have to explain in simpler terms the ALARA principle, as low as reasonably achievable. So for a general population, no dose should be received, but for a cancer patient, for a cure, the ALARA is the radiation dose that we give.
We should accept the risk, some side effects, but because ultimately it will be beneficial to the patient.
Correct. So how should we convince them that what we are doing is actually a meaningful? Both the aspects are very important to do that.
Yeah, general population means probably you are talking about that thorium-induced cancer background.
The people who are living in the low level of high background radiation, in Kerala.
Cosmic radiations, etc.
Right. So anyway, we got it. Now we have Dr. Prasanna around. Good morning, Dr. Prasanna.
Good morning. Can you all hear me?
Yeah, we can
hear you. Very good morning. It's early 6 o'clock.
It is about 6:30 here, 6:25. I think 9.5 hours difference.
Nice for coming here. And we have two chairs here, and Dr. Harisharan Mishra from BARC and Dr. Sanjay Tulkar. They'll be chairing the session, and so Dr. Harishankar Mishra will do the needful. He is a molecular biologist, and he is in BARC, and he's also attached to the Homi Bhabha Atomic Center. So he's one of the very senior molecular biologists with us.
Very pleased to meet you.
Yeah.
Virtually.
Thank you, Vibha. And at the outset, I would like to thank you for giving this opportunity. And first, I would like to welcome very early morning to Professor Prasanna for this session. And with a brief introduction about Dr. Prasanna, he did PhD from Mangalore University. Am I correct?
Yes, specifically Mangalore University from Manipal. Yes.
Yeah. And then he did postdoc from Armed Force Radiobiology Research Institute, Maryland. I think that must have been very perfect place to get to know a lot of radiobiology and all these things what's going on. And for last several years, he is a program director of SBIR DC, National Cancer Institute, USA. Professor Prasanna has published nearly 75 research papers in the area of radiation interaction with the matters. If I want to bring it again with all the areas and then biodosimetry. And today he has opted to speak on radiation-induced normal tissue injury. Beautiful. Normal tissue injury is actually more catching to us. And radio protection and mitigation to improve the outcomes. So with this, now I request Professor Prasanna to please take over and then deliberate. I request the people who are actually having questions, they all can keep recording it the way they did in last session. And then they can raise the hand at the end of it, and we will let them to actually interact with Professor Prasanna and with all of us. Thank you. Let's move, Professor Prasanna.
Yeah, thank you for that very kind introduction. A small change. I am at the National Cancer Institute that is in Maryland, which is part of National Institutes of Health, and specifically associated with the Radiation Research Program. And I do collaborate with the Small Business Innovative Research Development Center at NCI. Good morning and to you all, and well, it is good evening. Congratulations to Professor Vibha Tandon for putting this course in radiobiology under the GIAN initiative. Let us discuss today, without further ado, radiation-induced normal tissue injury and approaches of using radioprotectors and mitigators to improve the clinical outcome in the radiation therapy context. We all wish this was an in-person, face-to-face meeting rather than this virtual event. Nevertheless, given the circumstances we are in, I will make it as interactive as possible. If you raise your virtual hand, if somebody can monitor and point out, then given the earliest opportunity, please unmute yourself and turn your video on and ask your questions. If I ask a question, then raise your hand if you know the answer, and if you don't know that, if you want to guess the answer, and I will call out the names, then you turn on the video and unmute and answer. Let us see how much student participation is here. How much time do I have? An hour and a half? You are muted, Dr. Tandon.
Yeah. So 1 hour. You have 1.5 hours. 1.5 hours. Okay. Let's have a very interactive. I think you started very well. We'll be happy to have this kind of thing. And then so we'll have 1.5 hours. Am I correct, Vibha?
Yes, yes.
Okay, perfect. Yeah. So, I am a public servant, so there is no financial, there are some disclosures, there's no financial conflict of interest. And views expressed in this presentation are those of the author, that's me, and no endorsement by NCI, NIH, or HHS, or any other US government agency that has been given, implied, or inferred. And images are used in this presentation are obtained from the public domain and are for non-commercial educational use only. Having said that, this is the broad outline of my presentation today. I will provide a very quick background in about 10 slides or so. The goal is to discuss, as I mentioned earlier, radiation-induced normal tissue injury with certain specific examples. For the cancers of the head and neck, chemoradiotherapy is the standard of care in most cases. And one of the main adverse effects of this kind of treatment is this mucositis. We will discuss mucositis in this presentation. In the second part of the presentation, I will move on to radiation-induced lung injury. A specific example would be radiation-induced fibrosis, which can occur when radiotherapy involves irradiation of the upper thoracic region. Moving along, we will discuss a little bit about radiation-induced brain injury, cognitive impairment, as an example of radiation-induced late effect, then also an exciting and emerging area of senescence before summarizing this talk. We will also intermix these things with discussions on approaches to protect and mitigate some of these adverse events of the treatment as and when necessary in this particular sections. The entire talk will follow a theme of significance of these adverse events, how they occur, and what are the mechanisms, and what can be done by understanding these mechanisms. So with a brief background, Rifat Atun, not so long ago, published an excellent paper in Lancet Oncology Commission on worldwide cancer data, which is a specific project assigned to certain people, by the way. To reiterate the importance of radiotherapy globally, in 2012, according to this estimate, there were about 14.1 million new cases of cancers were identified, which is projected to go up to about 24.6 million by 2030. Out of this, approximately 7 million patients were treated with radiation therapy. The utilization rate is about 50%. So this utilization rate of about 50% is expected to remain at this level even by 2030. So let us look at the estimated number of new cases of cancers and deaths in the US to further emphasize the importance of radiation therapy. So radiation therapy is used to treat with the intent to cure in most cases, but it does not happen all the time. But if not, to palliate in the treatment of several organ sites. This data, do you see the table? Yeah, this data here is from the American Cancer Society. The data is a little bit old, but it still gives you an idea on the number of new cases and deaths. And the economic and social burden the disease has on the American society. In 2018, approximately well over 1.7 million new cancer cases were diagnosed, over 600,000 deaths. Cancer of the digestive system, lung, and breast and prostate were predominant. Radiation therapy is used in the treatment of cancers of vast majority of organ sites, as I mentioned. So significant technological advances in the dose delivery and its distribution within the tumor volume over the last decade has occurred, no doubt about it, and it has brought about some improvement in survival and quality of life outcomes, no doubt about it. This improved dose delivery and its distribution within the tumor volume has also improved the cure rate, both in terms of survival and quality of life, a point that's to be noted. But generally speaking, it has also decreased the incidence and severity of adverse effects for several types of cancers. We must acknowledge that fact. We must also realize, while acknowledging, that there is still a lot more room for improvement. So conventional radiation therapy is still being used, especially in low to middle income countries. And it is not likely to go away anytime soon with the replacement of LINAC and other treatment modalities. The distribution of dose within the tumor volume is very poor, which limits the intensity of therapy due to normal tissue tolerance very drastically. However, in LINAC, by and large, the radiation is delivered and distributed conforming to the shape of the tumor, or relatively speaking, more precisely. external beam protons that uses LINAC, for example, IMRT, can improve normal tissue sparing because you can deliver the dose from different angles, and in doing so, the organs specifically at risk can be spared, and you can thus minimize injury to the normal tissue, and also thereby reducing the adverse effects related to therapy. For example, in lung cancers, when you treat with IMRT, you can go around the heart, and which is an issue, that you can minimize the dose to the heart. And some of these things are not apparent immediately, as some of these effects are quite late, occurring quite late, and there are comorbidities also that compounds the issue. These advances now clearly allow a higher dose to be delivered to even the subvolumes of the tumor by so-called dose painting. In proton therapy, the dose distribution is different from photons and is relatively even more precise. It is important to understand the biological effects of protons, which is different compared to photons. Especially in proton therapy, it is the normal tissue injury which will limit the success of the treatment, especially in terms of the quality of life. Even if the dose delivery and its distribution is precisely within the tumor, that's not what really matters when it comes to the quality of life. It is the dose at the Bragg peak that can often, if you miss the tumor, you miss it in a big way, and that dose can be delivered to the normal tissue. So, and that will result in severe adverse effects might relate. Because of the higher relative biological effectiveness of proton, avoidance of injuries, normal tissue is crucial. In brachytherapy, the radioactive seeds are implanted within the tumor volume for continuous irradiation with the low integral dose. The choice of a given radiation delivery technology versus the other will depend on a variety of issues. That includes the organ site that is going to be treated and access to the treatment facility to the patient that has needed technology to effectively manage his or her cancer. For example, even this day and age, LINACs are not universally available, especially in certain villages and of low and middle-income countries, and patients may have to use the conventional radiation therapy. So, having said that, improving the efficacy of radiation therapy and post-treatment quality of life is essential to improve the treatment outcome. First, dose delivery and distribution. By the use of technology, as I explained in my previous slides, we have accomplished quite a bit in this arena, wherein today we have the ability to deliver dose more precisely and distribute it as needed within the tumor volume. Second, with the use of suitable biomarkers coupled to the technological advances in dose delivery and distribution, it may be possible in the near future a concept called personalized approach to radiation therapy use, personalized approach to radiation therapy, what is known as accurate precision radiation medicine. use of predictive modeling, for example, of the response of the tumor along with the surrounding normal tissue, it may be possible to refine as well as optimize treatment to a given patient as necessary. This is a new paradigm in radiation oncology that's called accurate precision radiation medicine. The third is with the use of radiation effect modulators. Either we could use radiation sensitizers on one hand to specifically sensitize the tumor without increasing the toxicities to the normal tissue, or use radiation protectors or radiation mitigators without protecting the tumors to improve the overall outcome to the patient. We will revisit these things in a couple of slides later. That is the meat of the talk. Therefore, it is reasonable to envision that the next big improvement in radiation therapy has to come from biological optimization of therapy, and there is some room in the technological aspects also. Having said that, an understanding of how radiation induces cell death is critical to understand normal tissue injury. Some time ago, when I was in India, I said the power of translational science depends on how well we understand the basic biology and basic science, and how well we answer the fundamental underlying questions in biology. In this case, it is radiation biology. How cells undergo cell death after exposure to radiation. I'm sure that you have heard from many speakers. In the context of radiation-induced normal tissue injury, understanding the mechanisms of radiation-induced cell death that leads to loss of homeostasis, leading to the loss of tissue function and its manifestation as radiation injury is very critical. Radiation can cause cell killing via multiple mechanisms, leading to different cell death outcomes. These include apoptosis, necrosis, autophagy, mitotic catastrophe, necrosis, senescence, and whatnot. So many genes are involved in radiation-induced cell death. Several genes now have been identified, and many molecular pathways of manifestation of these injuries are now better understood. I will get into some of these things, some of the specifics in a little bit later, as and when it is necessary, with certain specific examples later in the talk. Having said that, here are some of the genes involved in radiation-induced cellular injury tied to specific mechanisms of cell death. For example, in autophagy, you have P53, PARP1, and PI3K kinase, and AKT and mTOR. And for apoptosis, you have caspase-9, TNF, caspase-3, caspase-7, caspase-8, P53, BCL2, BAX, TGF-beta, and many others. It's important to understand the molecular pathways and networks involved in normal tissue injury. So, as I said, radiation exposure causes cell death and reduces, it results in reduced proliferation, and that causes loss of homeostasis and ultimately results in loss of tissue function. An acute effect in actively proliferating tissue, for example, in the oral mucosa, patients undergoing radiation therapy for the head and neck cancer can experience mucositis. This can occur acutely following radiation therapy of the head and neck cancer, head and neck area, starting in about a week to several weeks after radiation therapy. The effect can also be intermediate in relatively slowly proliferating tissues. One example would be fibrosis. That can occur in about 4 months to an year following treatment. During the treatment of upper thoracic region, for example, either breast cancer or even lung cancer. So it can also be late in post-mitotic tissues. The classical example is the patients who are undergoing radiation therapy for the brain tumors. They often experience cognitive injury. This can occur several months later after therapy to several years even. Radiation-induced secondary cancers is another example of late effect, which can take several years to manifest. There are two implications to this. First, some adverse effects could interfere with treatment, wherein it may not be possible to complete the full prescribed course due to the adverse effects. Most of the acute effects are seen in that context. And it will require cessation of radiation therapy or a temporary interruption in the treatment. Efficacy and therapeutic benefit both will be, and efficacy and therapeutic benefit will be compromised. The second implication to this is some of these adverse effects will affect very drastically the quality of life. The treatment is successful, but you have a compromised quality of life. In some instances, this will cause tremendous economic and social burden. For example, the pediatric brain tumor survivors is one example. So to summarize this portion of the talk, radiation therapy is playing a major role in cancer survivorship. Approximately 40% of the cancers cured today are treated with radiation. Number of estimated cancer cases will go up, but the number of patients treated with radiation therapy will likely to remain the same. With significant increase in the number of cancer survivors, addressing treatment-related toxicities will be crucial to predict, prevent, and mitigate. So the premise of using a radioprotector and mitigator is to improve the efficacy of quality of life. The probability of the tumor control is related to radiation dose. With increasing radiation dose, the TCP, the tumor control probability, will also go up. But mirroring this, you will also see an increase in normal tissue complication probabilities. So that limits the radiation dose that can be delivered to the tumor. With the use of a normal tissue protector or a mitigator, you can further increase the radiation dose in theory to the tumor without bringing in the adverse effects. So with that, I hope to have convinced you that there is a need for radioprotectors and mitigators, collectively referred to as radiation effect modulators. This is a bit of complex slide. I will focus on the process rather than getting into the nitty-gritty. This is a very generalized framework for developing radiation effect modulators. You have a lot of innovation that is happening in the academic and laboratories. But it's early on, it is important to figure out what is the problem that we are trying to address, what is the type of organ site that we are trying to go after. That will position ourselves the way how we go about developing this radiation protector or a mitigator. So you need to synthesize, you need to demonstrate the activity, and using cell lines and look at the clonogenic survival, look at the mechanisms of action, and some evidence of efficacy and normal tissue toxicology will also be necessary before we move into the clinical trials. Before that, we need to do the tumor growth delay in vivo and whatnot. And in the clinical trials, you assess the safety, then the efficacy, and then confirmatory trials. And finally, it will find use in the radiation oncology clinic. Any questions so far? And I see there are 3 people wanted to ask. Let's go.
Hello.
Yes, Sangeeta Chowdhury.
Yeah, yeah, Dr. Sangeeta, would you ask yourself?
Please unmute. I cannot hear you.
Okay, can you hear me, Dr.?
Yes, I can.
So she's, I'll ask a question. She's asking, is there any predictive biomarker to evaluate the efficacy of radiotherapy in patients, that is response to RT?
This is actually a very, like genetic profiling and cellular signature. Yeah, this is a very broad area. There is a lot of work going on in this area, and some of them are in the phase 2 trials with NCI funding, and I have updated some of our work, some of our funding, and what through a collaboration between academia and industry. Some of the efforts in a very recent paper in 2020. I don't think I have that listed in the further reading section. I can, if you send me an email, that's in the first slide, or get it from Dr. Tandon, and I will be very happy to send that paper to you. And it's a broad area, and that will be a talk by itself. But as I said in one of the slides, the specific cell death mechanisms and the molecular pathways, that gives us a lot of clues with respect to identifying the biomarkers. And it is not just a single biomarker that will give you the answers necessary for a variety of questions. What is the stability of the biomarker? Then how well it can predict the partial body, and then the normal tissue and the tumor response plus the normal tissue to gain the optimum therapeutic outcome. So it could be a panel of biomarkers. As you said, it could be a profiling involving multiple biomarkers. So yes, I will share that publication if you send me your email.
Yeah, sure. I will share it. So yeah, I can give her say email also. So you can carry on. There are no other.
No other questions. Yes. Thanks. Yeah. So with that background, now let us move on to some of the specific examples. One example is mucositis. Radiation, this is again a very large and complex area. I will not venture into all the complexities, rather provide you some quick information and point you to specific literature in the field for the reading at the end. So this is an acute adverse effect of radiotherapy and chemotherapy that includes erythema, xerostomia, and mucositis. The mucositis is inflammation and ulceration of the lining of the entire gastrointestinal tract. That is how it is viewed these days. And not just restricted to oral cavity anymore. You know, if you see the mucositis in the oral cavity, you can pretty much assume that it is entire GI tract is involved, even if the radiation is delivered only to the head and neck cancer area. That is how the Stonich model is alluding. That's what Stonich model is alluding to. So this occurs at an accumulated doses of about 10 gray and above, and quite intense, around 30 gray. And both radiotherapy and chemotherapy-induced mucositis. So in most cases, head and neck cancers are treated with chemoradiation therapy. So distinguishing between these two becomes very important. As one can see in the graph here, there's a subtle difference between the chemotherapy-induced and radiation therapy-induced mucositis. Here is the chemotherapy-induced is relatively fast. Yeah, the chemotherapy-induced mucositis peaks very rapidly, and compared to radiation therapy. Accelerated radiation therapy, you will see more severe mucositis compared to conventional radiation therapy. This supports the radiobiological underlying 4 or 5 hours of radiobiology. In both cases, the accelerated radiation therapy and conventional radiation therapy, severity of mucositis increases very gradually compared to chemotherapy. Steve Sonitz has done a lot of work in this area. So what are the risk factors? You can categorize these risk factors into patient-related and treatment-related. The patient-related risk factors include the tumor type, inter-individual variability, genetic polymorphism, age and gender, oral conditions, care and microenvironment, nutritional status, and of late, you see a lot of importance is being given to microbiota, and that is also obviously another emerging area that is very, very, there are a lot of opportunities in this area. Then there are treatment related, that is radiation dose, chemotherapy and drug, treatment schedule, and neutropenia can also cause, can exacerbate mucositis. So this is again, as I said, a model that is developed by Steve Sonitz. And this model describes events leading to different phases of mucositis. There are about 3 or 4 or 5 phases. There is initiation, then there's damage response, signal amplification, ulceration, and finally healing. And the initiation can cause damage response, and that leads to signal amplification, and signal amplification can further cause initiation due to chronic oxidative stress. And here, the tissue will make an attempt to heal, and during the process, you will see ulceration with the opportunistic bacterial colonization. And during that, you can also see additional pro-inflammatory cytokines are secreted. And there is robust infiltration of macrophages and neutrophils. And finally, you will see the halt of epithelial proliferation as a response to apoptosis and necrosis. There is an exhaustion. And eventually, you will see the healing. And there's somebody can you mute, hear a lot of background noise.
I muted,
okay.
I muted. There's
Thank you.
One person is getting him unmute himself. Okay. Yeah, I'm muted.
Thank you so much. Appreciate that. So the current treatment approaches for mucositis, there is no effective options for the prevention. The philosophy here is to prevent over mitigate, then mitigate over treatment. The majority of the treatments are post-treatment management and are palliative at this stage. And what has been historically being used is keratinocyte growth factor, that is palifermin, as an approach for management. And it is approved for stem cell transplant patients. It stimulates epithelial cell proliferation via NRF2 pathway, and it is anti-inflammatory, and it attenuates pro-inflammatory cytokines. So the non-pharmacological approaches are also used, such as benzidamine. It's non-steroidal, anti-inflammatory, anesthetic and analgesic. And some of the coating barriers like mouthwashes and oral cryotherapy, etc., are also used. Radioprotectors and mitigators are still under development. But the one concern here is when you use a radioprotector to deliver the dose that is fully prescribed, so that you can go to the full prescribed dose, you should not get the mucositis. The one issue is will the radioprotector protect the tumor, so you don't see that therapeutic benefit by using this radioprotector. So to summarize, it can occur along the entire GI tract, can limit the intensity of therapy and compromise post-treatment quality of life. Understanding the mechanisms of manifestation of mucositis may allow discovery of novel targets and the development of suitable intervention strategies. Currently, there are no effective options to treat mucositis or prevent from happening. So SOD mimetics, the GC4417, that's an experimental compound from Galera Therapeutics, and BMX001, that is biomimetics, Greenwood, Colorado, are being developed and show some promise in clinical trials. It is also, the more details on these things are also in my publication. I believe it is 2015, and there's one more publication that update came out in 2019 or 2020 by Zachary. So any questions so far on oral mucositis or mucositis? I see if somebody has a chat there. Oh, that is Dr. Tandon telling everybody to mute.
Yes, so so I just want to ask something. Like when we talk about the oral mucositis, so there are any ways to handle it, or this is going to be the one of the outcome of the oral cancers, or it is there any ways, or is there any successful treatment has come against the oral mucositis?
Yeah, this is a very good question, Dr. Tandon. I know and why you would ask that too. I think it is a big concern in India. There is no very effective drug as you can call as a radioprotector or a radiation mitigator. Whatever we have is still under development and still under clinical trial. And it takes time from if you follow the process and the GLP and then the in vitro clonogenic survival, and then the in vivo the tumor growth delay assays, and using appropriate oral mucositis model, for example, the Chinese hamster model or a rat model, and then you run the clinical trial. It takes demonstrate there's no tumor protection. It takes time. The clinical trials are also often, in most cases, the accrual can be slow, especially in US, and there are certain barriers. So there's nothing out there that I am aware of in the true context of a radioprotector or a mitigator, but everything is being managed with the palifamin or as I showed in my last slide, the mouthwashes and such analgesics and give a temporary relief. But if you look at the course of the development of oral mucositis and the healing and the halt of ulceration and the healing, that is much more delayed compared to the chemotherapy. So unfortunately, it is difficult for the patient who is undergoing the radiation therapy. And on one hand, you have the cancer burden the patient has to deal with, and on the other hand, the burden of treatment.
I have a small request to elaborate on a point, because this is being attended by a large number of younger colleagues also. You said that there are two arms. One is patient side, the other is a treatment side. In patient side, you mentioned microbiota. Would you like to elaborate how does microbiota can help the patient to overcome some of the injuries or negative effects of radiation?
This is a very...
Do you know why I brought this point? Because the majority of these 48 attendees today, they are very young people, have several years to go. Correct. So I just wanted you, if you can bring out some, if you highlights that how you think that this microbiota particularly can help to control the radiation injuries or maybe efficacy of radiotherapy, etc.
This is a very fertile and emerging area, very exciting area. This is, there are a lot of young students out there, and the context is a lot of students are in India. And there is a system in Indian system of medicines called Ayurveda, right? Everybody knows about it. And wherein you characterize a patient according to the phenotype. Where you have Vata, Pitta, and Kapha and whatnot, all these things are coming from the microbiota. There is some sort of understanding. People are getting into that kind of understanding. And with the microbiota, with modulating the microbiota, can you enhance the quality of life? Is it possible? Is the response to radiation of the tumor as well as the normal tissue dependent on the composition of microbiota? These are the questions that is unanswered. Now, I understand there is a clinical trial ongoing, especially in the context of head and neck cancer, wherein they have characterized the different microbiota. Then they trying to see what type of microbiota actually induce people who have what a microbiota can be related to radiation response, and then trying to figure out whether you can modify this microbiota with either with the prebiotics or probiotics or postbiotics. Then will it benefit the patient? This is a very, very fertile area. Thank you for bringing that up.
Yes, I want to actually highlight another very important thing, because I know I'm basically a microbial person. So see, when you say that treatment of 10 gray or 15 gray of gamma radiation, that is something dose which you highlighted in one of your slides for radiotherapy. Now, this is the dose of gamma radiation that will not be able to kill 100% of the microbes in the midgut or anywhere they are there. So this could be another area of research, in my opinion, if some of the beneficial microbes can be actually manipulated to produce some of the radioprotectors, antioxidants, and then if they can be fed to the human before they are actually irradiated for gamma, because 2 gray, 4 gray, 10 gray gamma radiation, it doesn't touch to microbes. They have ability to actually overcome. Of course, there are microbes that can tolerate in kilogray, but even majority like E. coli, which is the simplest microorganism, the D10 is 60 gray. So even by 60 gray exposure, the 10% of the cells remain viable. So this is another domain which actually radiation biologists may think of it. If they can club the microbial engineering or biotechnological approach to the radiotherapy, and by going to gut microbiota, microbiome analysis, etc. As you said very rightly, I agree. It's a very good domain, very flourishing domain of research, which actually people should be looking forward.
Yeah. So I have another 30 slides to go, and I will
please.
need about 35 to 40 minutes. Is that okay? If I went about 5 minutes?
Continue.
So that I can,
I think you have time.
Yes,
Okay.
you have
time.
Yeah.
Please go ahead now. We'll not interrupt you a lot.
Okay. But I wanted this to be a little more interactive, for the benefit of students. They understand the discussions, and even if I have to skip a section, that is fine. But I think it's important. Thanks for bringing that up. And let's move on to the radiation-induced lung injury. And thoracic radiation invariably induces damage to the lung, and you see immediately the pneumonitis, which is asymptomatic and opaque chest X-ray, and the acute response is about 2 to 6 months, and it is dose dependent. There is a linear relationship from about 5 gray to 30 gray. TGF beta 1, IL-1, IL-6 appear to be involved. And then you have the late response. Some of this fibrosis can manifest into, while some of this pneumonitis can resolve by itself, and some of them will manifest as fibrosis down the line. It is late response, months to years, and irreversible. So what are the determinants of RILF, radiation-induced lung fibrosis? Again, it is treatment-related and patient-related. In the treatment-related, you will see dose per fraction treatment schedule, use of chemotherapy, and there is a relationship between the dose and volume of the normal tissue irradiated. So in lung, what happens is if you give a very high dose to a small volume, that is less damaging compared to a low dose to a large volume. There is a linear relationship between 5 to 30 gray, as I mentioned earlier. So the genetics will play a role. The trauma or surgery in the irradiated site, then non-malignant systemic diseases such as diabetes and hypertension, cellular turnover rate, role of the tumor itself. The tumor also has a role in how the fibrosis will manifest. So not so long ago, we at the National Cancer Institute organized a workshop on radiation-induced fibrosis to highlight the importance of this normal tissue injury in the context of radiation therapy. And the objectives of this meeting were to highlight the significance in the global context and summarize the recent advances in our understanding of the mechanism of RAF and discuss the opportunities for pharmacological mitigation, intervention, and modulation of specific molecular pathways involved in the fibrosis. And also consider rational design for optimal clinical trials to mitigate and treat fibrosis. And outline key regulatory non-prescriptive framework for approval. One problem that you have is, not every patient who undergoes radiotherapy of the lung or the upper thoracic region will develop fibrosis, and it occurs in about 15% to 30% of the patients. So you have to have, how do you incorporate the quality of life outcomes to a clinical trial which predominantly focuses on the primary objective of patient survival. So these were some of the challenges. So the consensus was that there are several known and newly identified targets that can prevent or mitigate radiation-induced fibrosis in preclinical models. In addition, basic and translational research and focused clinical trials are needed to identify optimal agents and strategies for therapy. So having said that, let's look at the underlying mechanisms of pulmonary injury by radiation. This understanding is important for several reasons. The purpose of this slide is to provide a broad overview of this particular effect. This radiation-induced pulmonary injury has about three phases. There's an injury and inflammation and repair, and they're not very drastically separated. They are more congruent, gradual transition from one to another. And in the injury phase, it is due to mostly oxidative stress. You will see degradation of extracellular matrix. You will see apoptosis of pneumocytes, loss of barrier membrane function. Then you will see in the inflammation, the injury will lead to inflammation. You will see dysregulated inflammatory response. You will see a lot of leukocyte infiltration. You will also see cytokines, chemokines, and growth factors secreting. And you will see chronic hypoxia. So the inflammation can cause further injury, or inflammation, if it is pro-healing, you will see the repair also. So in the repair phase, you will see release of signaling cascade events and tissue remodeling and change in tissue composition. There will be vascular changes. Macrophages will start infiltrating. And you will see pro-inflammatory and pro-fibrogenic and pro-angiogenic cytokine secretion. And finally, fibrosis will settle in. One concept is, when I talk about senescence, it will be a little more clear that about this concept that I'm going to tell now, is the fibrosis is often seen as a failed attempt of a reparative regeneration of the lost functions of the tissue. What I mean by that is, you will see that there's an attempt by the tissue to restore tissue functions, but it is not happening. And fibrosis will settle in. So manifestation of fibrosis here, this slide actually further expands on the concept and help understand the molecular events that occur during the fibrosis. You will see in the normal lung, you will see the quiescent fibroblast, and that can get activated, and you call it an activated fibroblast because of stress, reactive oxygen species, and growth factors, particularly the TGF-beta, and hypoxia and cytokines. All these things will cause the quiescent fibroblast to get activated. And if in the immediate aftermath, if it is taken away, the activated fibroblast can go back to the quiescent stage. The characteristics here is extracellular matrix molecules. You will see type 1 collagen and fibronectin, etc., that characterize the quiescent fibroblast. So in the activated fibroblast, you will see the cytoskeletal remodeling, vimentin and smooth, alpha smooth slactin. And you will see the secondary phenotype, differential cross-linking of ECMs, fibronectin production, VEGF and TGF-beta, CCL5 and IL-6 that will characterize the activated fibroblast. So you will see the an activated fibroblast can further progress as a fibrosis-associated fibroblast, wherein you will see MMP1, MMP2, MMP3, and MMP9, and they acquire a phenotype called what is known as an enhanced secretory phenotype characterized by TNF-alpha, NF-kappa beta, NF-kappa B, and IL-6 and IL-8. So TGF-beta is seen as the master regulator of fibrosis. So ionizing radiation can directly act on TGF-beta, or ionizing radiation, such as radiation therapy, can act via reactive oxygen species and reactive RNS. And you will see, and it can also cause cell death. That is, cell death associated signals, the DAMPs, can also act on TGF-beta. So you will see the receptors TGF-beta 1, R1, and R2 in the cell membrane, and these can act further on SMAT signaling pathways, and the SMAT signaling pathways will play a role in the proliferating fibroblast and can act on quiescent fibroblast and differentiated fibroblast, and all these things will lead to ECM and collagen deposition and tissue remodeling. And this can again circle back to hypoxia and modulate TGF-beta, and that vicious cycle can continue. So to summarize, many organ sites are involved and can occur between 4 to 12 months and progress into several years. And can it limit the intensity of therapy, compromises post-treatment quality of life, and understanding the mechanism of manifestation of fibrosis may allow discovery of novel targets and allow development of suitable intervention strategies. Some important clinical trials to treat RIF include using ACE inhibitors, enalapril, lisinopril, etc. And Bio300, it's a nano formulation, Humanetics Corporation. We funded this also, NSAI funds some of the clinical trials, also some of these things. And they are still under clinical trials. So another example is radiation-induced brain injury as a late effect. Here, the important adverse effects of after conventional radiation therapy of brain tumors and head and neck cancer, you can see. This is the cancer type, the standard of care, glioblastoma, temozolomide, along with radiation therapy is used. For head and neck cancer, cetuximab is used. For unresectable, for resected, you have the cisplatin. And the mortality rate for GBM is 73.5 with a median survival of only 14.6 months. For head and neck cancers, the mortality is about 45% and median survival is about 4 years or so. And in these cases, you will see the treatment-related cognitive effects is about 50 to 90% combined. It may not be severe, but you will see the cognitive changes. So having said that, improvements in detection, treatment planning, standard of care, and personalization has been improving survival. But as survival improves, post-treatment quality of life becomes even more of a significant concern, and it has possible long-term effects. So radiation-induced brain injury occurs in 3 to 4 phases, and acutely you will see the edema, headache, drowsiness, which is although rare under current treatment conditions. You do see some of these things. The grading of that is a little bit lower, but you do see. The weeks to months after radiation therapy, you will see early delayed effects of brain injury, wherein transient demyelination, some of that can resolve, but the attention deficits, you will see shorter memory loss. Severity is not predictive of late effects. Some of these, even if some of these things are resolved, it appears that it is resolved, but it can come back. And years after, you will see the brain injury that occurs at higher doses, greater than 60 gray. Intellectual deterioration can occur even at lower doses of radiation. You will see vascular abnormalities, demyelination, white matter necrosis, memory loss, and includes hippocampus. Over the years, physical delivery of radiation to the target has tremendously improved. Depending on the tumor location, radiation can directly cause injury in white matter, hippocampus, or prefrontal cortex. Irradiation of white matter will lead to apoptosis, and depletion of cycling oligodendrocytes and demyelination of loss of white matter integrity. Hippocampus is in the modern radiotherapy of there's no whole brain radiotherapy, but in the whole brain radiotherapy is there with hippocampal sparing, where active proliferation of neuronal cells happen. So you need to avoid that region exactly for the same reason. So, which is of a lesser concern people try to avoid. It is very well known that the hippocampus has to be spared, and people do make an attempt, and you don't see it that much. But it is possible that sometimes erroneous treatment planning can lead to such an issue. If this happens, the loss of neuronal precursors that will cause inflammation and loss of vasculature. Damage to dendrites, altered signal receptor tracking will happen, failure to differentiation, and long-term neuronal loss. So the prefrontal cortex, the damage to this area, you will see white matter necrosis and neurons, tissue dysfunction, and all will cause lower glucose uptake, cognitive deficits, and cognitive decline. So there's been some efforts to treat brain injury. This is not a comprehensive list by any means, but you do see that ACE inhibitors have been used, NMDA receptor agonist, memantine, VEGF inhibitors such as bevacizumab and statins have been used. Although many agents show promise in preclinical studies, and some of them are under active investigation in clinical trials, there seems to be so far limited success. Novel approaches to reduce cognitive impairment is desperately needed. So a quick summary, radiation-induced brain injury is a classical example of important late adverse effect. Understanding the mechanisms and manifestation of radiation injury is critical to develop next generation radiation effect modulators and biomarkers to accelerate accurate precision radiation medicine, treatments that modify cellular damage and death processes. Inflammation, tissue regeneration, tissue remodeling, and senescence will help mitigate these adverse effects. Any questions so far? Hearing none. So let's move on to an exciting area called therapy-induced senescence. This is again an emerging area in the context of radiation therapy. So what is senescence? Senescence is a state in which cells are permanently growth arrested but are metabolically active and is a burden to tissue function. That's the simple definition, but the definition is highly debatable. So the premise here, and here you see a normal cell, and you will see there are two types of senescence. One is early acute and another one is chronic. I will come to that in a minute. The acute stress, such as reactive oxygen species, and all these things will cause something called secretory associated senescence phenotype. So in the chronic state, there is also the chronic stress coming from reactive oxygen species, again, will bring in SASPs. That's the late and chronic senescence. And the acute senescence is routinely overcome. The acute senescence causes tissue regeneration and helps to restore tissue fitness. And it is cleared by immune system, and there is a cell cycle arrest. And although it can be transient, it can progress into quasi-transient and quasi-permanent, and then allows tissue repair. And acute senescence is routinely overcome by immune clearance. So, but the chronic senescence, that's the tissue disruption and decrease in tissue and stem cell fitness. And there's a DNA damage due to oxidative stress, inflammation, etc. Cell growth, angiogenesis, and plasticity it will cause. So tumor genesis is promoted, tumor resistance and metastasis, and you will see tissue injury, and that will result in aging. So with that, the cancer treatments also now have been shown to induce cellular senescence both in cancer as well as in normal cells. Second, selective clearance of the senescent cells is beneficial and will likely to expand the health span of an individual. This is a new paradigm, although it's been known for years. How can we use this to enhance the quality of life of an individual? So last year, we at the National Cancer Institute organized another workshop on therapy-induced senescence involving the world experts, with the goal of understanding molecular pathways that govern senescence to generate novel insights into the discovery of novel anti-cancer agents and molecular biomarkers. This workshop discussions led to the identification of important knowledge gaps in the exploitation of cellular senescence, what is known as 1-2 punch cancer therapy. And I will explain more on that, where 1-2 punch cancer therapy in a nutshell consists of agents to induce tumor cell killing, such as radiation therapy or chemotherapy or CRT combined, and then subsequently use a senolytic to get rid of selectively the senescent cells, both in the tumor microenvironment as well as in the normal tissue environment. Such an approach will help prevent tumor recurrence, metastasis, and is expected to restore healthy lifespan of cancer patients. Having said that, cellular senescence is seen in aging, infectious diseases, in cancer, and is induced by cancer treatments. However, in order to use this concept of using 1-2 punch cancer therapy, it is important for the field to answer several important questions. That includes the role of senescence in the pathogenesis of a disease or an adverse event following cancer treatment. For example, pulmonary fibrosis after radiation treatment or cognitive injury after radiation treatment. Then we need to characterize the senescence phenotype, because senescence itself is very heterogeneous, with different senescent cells having different features in different tissues. So there is no, the field also suffers from lack of biomarkers. There are only limited number of biomarkers from which you are able to characterize a permanent growth arrest. And these cells are there, and you have only 3 or 4 biomarkers right now to be able to classify these things as senescent cells. And getting rid of these senescent cells with the help of these biomarkers, is not fully possible yet. So then you need to understand the mechanisms and the targets. These are the varied targets. And if we get rid of these, the so-called senescent cells, what other effects you will see? When you target these particular senescent cells, is it also possible that there might be some off-target effect? So, again, the field, as I mentioned, suffers from the lack of biomarkers. Nevertheless, understanding these fundamental questions will help further develop the fields of xenotherapeutics and perhaps even better anti-cancer agents, and possibly to adopt this approach in the personalization of radiation therapy or chemotherapy. For example, you may be able to modify the dose. You may be able to adjust the dose schedule and whatnot. So that is the problem here, and that is the process that is very active area, and the product, not so active area, but I think we are getting there. So this is again another a conceptual slide. You can look at the JNCI paper that came last year for more details, but this is the broad overview of the concept of 1-2 punch cancer therapy. This is a complex slide. Here's the legend. You will hear this is the normal cells, and here are the tumor cells, and you have a heterogeneous senescent cell population both in the normal tissue as well as in the tumor, and these are the various SAS factors. So this is the first punch here that is served by radiation and the chemotherapy or the targeted therapy. And then you will have a second punch. This first punch will bring in senescent cell heterogeneity, which is a dynamic, and you need the biomarkers, you need, these are plastic cells, you have SASPs and SCASPs and tumor versus normal tissue. You need to understand these things. And then you provide a second punch. And this second punch specifically targeted to eliminate the senescent cells, to prevent the tumor relapse, metastasis, and treatment. And therapy-induced late and acute and late effects can also be prevented by eliminating the senescent cells in normal tissues. So integration of 1-2 punch cancer therapy with personalized adaptive tumor therapy concept, is outlined here. This would require pre-treatment characterization of molecular profiling of tumor, the genetics, heterogeneity, imaging, and such, and identifying suitable biomarkers, both in the tumor as well as in the normal tissue. So then you have the primary therapy, such as the drugs, immune therapy, or radiation. That will be the first line of treatment. Then you will monitor the response and adaptation of the tumor, and you evaluate those things, both in the tumor, stroma, and the bystander tissues. For therapy-induced senescence, that's what you will evaluate, you will look for, which is a dynamic thing. And you dynamically adapt tumor targeting with drugs, immune, or radiation, and you provide the second punch. And this could be in days to weeks and then months, and you may require a second course or a third course. This is like after, say for example, after the CRT, after the chemo radiation therapy, then you pause for a week and bring in the second punch, the senolytic treatment, a course, similar to the antibiotic treatment. So this is a very novel concept, where the feasibility of these things have to be yet to be determined. So having said that, some ongoing efforts in xenotherapeutics include testing of natural compounds, targeted therapeutics, and protopic and PROTACs. So these are the quercetin and PI3 kinase inhibitor, one of the examples of natural compounds, the mechanism of action, and it is undergoing multiple clinical trials. Jim Kirkland is doing a lot of work in this in the Mayo Clinic. It is being used in Alzheimer's disease, diabetic kidney, idiopathic pulmonary fibrosis. If it works for IPF, can it work for RIF too, right? So curcumin analog, EF24, that promotes apoptosis, and apoptosis of senescent cells may be beneficial. That can be senolytic, and it's in the discovery and early preclinical trials. And cardiac glycosides, the ATPase inhibitors, it's also another discovery stage and drug repurposing. And you have the targeted therapeutics such as dasatinib and tyrosine kinase inhibitors. Clinical trials are ongoing. And navitoclax, ABT263, which is a BCL-XL inhibitor. It's a late stage preclinical development. Unfortunately, there's a lot of toxicities associated with this. And Dr. Dao-Hong's lab in University of Miami, is University of Florida in Jacksonville, is using an approach called PROTACs to reduce the off-target effects and toxicities of this BCL-XL inhibitor, navitoclax. So here, the whole idea is to attack therapy-induced senescence from various angles. So TIS is therapy-induced senescent cells is the target. So in order to do so, we will need several tools and approaches that include single cell and spatio-temporal analysis of senescent cells. We need biomarkers, we need a lot of imaging tools. We need to understand the tumor and we need appropriate models to understand the tumor and normal tissue injury, and xenotherapy needs to be developed, understand the SASPs and SCASPs. So, however, there are several pitfalls and challenges and opportunities. The discovery, experimental models, and whatnot. So with that, I think it is the time for me to summarize my talk, and we looked at the background. We also looked at radiation mucositis as an example of acute effect, and lung fibrosis as an intermediate effect of radiation therapy, and brain injury, and also the novel area, an emerging area of radiation-induced senescence, and we will summarize. So radiation-induced mucositis, fibrosis, and brain injury are examples of important adverse effects which will impact the quality of life of millions of cancer survivors. Therapy-induced senescence is an emerging area. Understanding the mechanisms and manifestation of radiation injury is critical to develop biomarkers and next-generation radiation effect modulators. However, there are many challenges to overcome, which are also opportunities to improve the outcomes of millions of cancer patients undergoing radiation therapy. So what are these historical challenges in the development of radiation effect modulators? So you have the radiation-induced normal tissue injury. The mechanisms are complex. You need to understand those. And long-term studies are necessary, especially to understand the late effects. And it suffers to some extent suitable models and some of the regulatory barriers too. Such as you're not able to keep an animal, a tumor-bearing animal, long enough to study, because of ICOC issues, long enough to study the radiation-induced cognitive injury in a mouse model. So differential effects of on the tumor and normal tissue still, after several decades of research, we don't have a true radioprotector that can differentially act, that can act as a sensitizer of the tumor and a protector of the normal tissue. And if there is one, and what is the mechanism of action? And in order to understand that, understanding that these mechanisms are very crucial, what makes a tumor, and what makes a normal tissue. So there are differences, and how can we exploit the differences? This has been an issue. So rigorous and valid preclinical data for translation is necessary, and our field actually lacks, to some extent, a very active collaboration between academia and industry. That's how the progress is happening elsewhere. So significant challenges in the translation of radiation modulators also include changing standard of care and changing the SOC. There is a constant change in the standard of care in the treatment of a given organ site. There is also, how do you change a standard of care and bring in a radiation effect modulator as a first line of treatment, wherein you need to get a confident data that will allow you to test a new standard of care that would replace an existing standard of care, which is radiation plus a radiation effect modulator without being another chemotherapy or some other second or third line of treatment. To what extent, how a patient can confidently sign off a consent form, or a physician can be so confident to treat a patient with what level of data is necessary to treat a patient with a radiation effect modulator and just radiation. How can the radiation be used to bring an immune response, to take care of the systemic disease burden? These are the issues. Some of these things are holding us back. Because we do have now, on the positive note, we do have stronger academic industry partnerships, relatively speaking, compared to the last decade or so. And significant advances have been made in developing protectors and mitigators, for example, mucositis and fibrosis and even brain injury, with the stronger academic partnerships. Some clinical trials are ongoing, at least I'm happy to see that. So with that, thank you. And I will leave this slide if anybody wants to note down some of these references as further reading. Any questions? I think I'm about 6 minutes over my allocated time.
Thank you, Pat, for nice talk. Now it's open for discussion. If people have questions, they can reach out to us.
I can see that Dr. Dwarkanath have a couple of questions.
Okay.
So he has, Dr.
Dr. Dwarkanath, so pleased to see you.
My pleasure, Prasanna, after a long time seeing you, hearing you, and also getting educated. As I always do periodically.
I don't know about that, educating you. But
good. So nice, a broad coverage, a very, very vast coverage. I hope people will appreciate that. And also nice to see Dr. Hari Mishra.
Yeah,
in a long time.
Smiling. I'm also smiling by seeing you. I wanted to say a few words.
It's been a
anyway,
long time.
Thank you.
Let's
go ahead.
Yes.
Let's go ahead with questions.
I had a couple of questions. The second one is actually to initiate a discussion rather than question. The first one, of course, I'll start off from the end when you talked about somewhat personalized approach in managing the therapy, targeting the treatment-induced senescence. Probably a couple of things that you mentioned, which included also identifying the induction of senescence and a marker for that, and to be able to see if that can be accomplished with the imaging or any other approach, and then to come up with appropriate interventional strategies to attenuate, if not completely eliminate, senescence turning into a path for recurrence and failure of therapy, if I understood that slide correctly.
Yeah.
My question is the following. For a tumor which is beneath, located in any of the internal organs, mainly not from the phenomenological or conceptual point of view, it looks very good. From the practical aspect linked to any approach and its inherent sensitivity, and therefore our ability to identify this in a non-invasive way. Some of the readouts in any of the accessible body tissue, for example, body fluid, probably will have minimal value. And when you come to imaging, I do not know at this point of time which imaging, unless you're suggesting some specific vector targeting the senescent phenotype tagged with a reporting radionuclide or fluorescent in humans is probably far away. To be able to do this, even if we did all this, the amount of senescent cells that a therapy would induce in a given volume of the irradiated area, even if the entire tumor is gone, how as it stands today and years to come, this will really be very highly achievable.
Yeah. You are actually making me really think. Okay. So what I'm hearing is, there are multiple layers to your question. This is, I've been thinking about this. How can we do this? Our biopsies. The tissue banks not going to work because the tumor is heterogeneous, and you are even if you do the gross biopsy, you're not going to get everything that you want because you are not still addressing the temporal heterogeneity there, the time course issues. You may be able to address, even with the gross biopsies, you may be able to address the spatial heterogeneity issues. You may be able to see with the limited amount of biomarkers, P16, P21, or some of the other things, the SAS, the so-called SAS, you may be able to see some of these things. Judy Campisi's lab is creating a tissue atlas for this purpose. That's one angle, just one angle, wherein can you look at the proteomics. The SASPs are nothing but that the proteome, the senescent associated secretory phenotype, the proteome. So you create a proteomic atlas, and to some extent, you may be able to come one layer of hurdle, but not fully, because you are now looking at, but still it will be heterogeneous. But what other things are necessary? In one of my slides you saw, you need to focus on biomarkers. You need to have the imaging. You said the non-invasive imaging is probably decades away, if not years. So that is another difficulty. But in my opinion, I think there is a low-hanging fruit is there. Maybe microRNAs, maybe, And if you are able to look at these microRNA changes in a quasi or semi-invasive way, looking at the blood, looking at looking at the tumor cells, circulating tumor cells or ctDNA from that may be another opportunity, and look at both normal tissue and a tumor. But the field is thinking about it and going in that direction.
Sure.
So I think there may be an opportunity, but it is challenging. I just totally agree with you, and I think you were multi-layered, multifaceted questions. We don't have answers to all of that.
No, I'm sorry about that, but the other thing I wanted to initiate, probably you could take questions and defer what I'm going to mention now to a later point for discussion. What would you like to dwell on? The precision radiation medicine and also precision oblique individualized radiation medicine in the backdrop of the existing comprehensive knowledge that we have accumulated or accrued, and also the upcoming and promising knowledge that would become available in the near future. I'm talking about next year, if not earlier. With that in the background, what do you think are the approaches, very practical, doable approaches, and commensurate success rate for the precision radiation medicine? This is, I know it's some sort of a general discussion, therefore I would even be okay if you defer this and take other pointed focused questions and then come back to discuss this. I'm done.
Dr. Tulkar, do you have any? I saw him unmuting himself.
Yeah, I think now I have unmuted myself.
Yeah. So
yes, we will come back to the precision one if there's any other question, otherwise, we will go on. Yeah.
If there are some pointed questions, that probably should receive it.
Especially from students.
Correct.
So, so if there was a question, but it was like sent to me only. Yeah, so there was a question there, like where
Maybe Viva, I can get into just a provoking question.
Okay, I don't see anything in the chat.
No, I somebody sent it to me. I'm just asking.
Okay.
I'm trying to paste it to everyone. Then you can always, Dr. Mishra, you can do it.
Yeah,
so somebody sent it to me that about the tumor growth delay. So they were asking that when you talk about the tumor growth delay, how to calculate it, and like what do you mean by this tumor growth delay? What kind of study should be done in the tumor growth delay? What is expected out of it?
There was a topic that would have covered this as an integral part of that lecture. Somebody was talking about tumor response. Yes, I'm also probably supposed to cover this. If it is
actually,
from the student,
he showed it in as the part of the radioprotected development. That's why it is question.
No. The reason I'm suggesting this is, if this is from any student cohort, let's defer this, because I'm going to be discussing this as an integral part of the lecture.
Excellent.
I think when I talk about.
Thank you for that. In essence, the radiation sensitive.
Oh, please, sorry.
Radio sensitive. Go ahead. Okay.
I think in my opinion, the same question can be addressed by different people. Probably will get different perspectives.
Sure, absolutely.
So let's it be open, not to say that I am going to talk about. So
no.
That question can be addressed by Pat. I think that would be good.
No, if there is a paucity of time, we could. I said this is an integral part of a lecture. But if there is time, I totally agree with you. Different perspectives are always better, and to collate is even better.
Sure. In the context of development of radioprotectors, I think it is important to show there is no tumor protection. And there is no tumor protection, which eventually should manifest in the clinical outcome of quality of life as well as efficacy, right? So if you are using a radioprotector, let us do the tumor growth delay. Dr. Dwarkanath is going to go and give the specifics, what the issues are and what different types of models, maybe, I don't know what he's going to talk about. But I think let's assume that all those things are going to be covered in the context of radioprotectors. This is important. Also, it is important, does your protector protect against cognitive injury? That might happen 90 days after the treatment is over, but you are not actually able to, in a mouse model or in a rat model, you are not able to see in a tumor-bearing mice, because the tumor also will have an effect on the outcome of the quality of life. Okay. So having said that, you are required to sacrifice the animal because the tumor is now grown so much. You're not able to see the added value of the radioprotector bringing in, or the mitigator bringing in, a better quality of life. That would happen post the observation period. So there are some challenges like these. In a nutshell. Did I answer your question?
Yeah, I think it's quite clear. I also have one I believe it's a provoking question for all of us, because I'm not aware of it. Has anybody studied the senescence with respect to the membrane of the cells? That kind of studies have been done, because now it's known that under certain conditions, there is a membrane bevelings, etc. happens. Whether such kind of thing can be considered as a marker that now cells are entering this kind of situation. I don't know whether such kind of studies have been carried out. I mean, I'm just bringing one concept.
No, it's a good question. the SASPs are mostly the proteomic changes, and you will see the morphological changes under the microscope, and which is also dynamic changes. And you will see some membrane changes too. So, but is this membrane changes that is happening, is it a cause or an effect? We don't know. And how can we address these things? You know, this is an area.
Yeah,
but to my knowledge, I would have, it could be anything. To my knowledge, if something was looked at specifically, you know, when I was putting that paper together, that workshop together, that would have come up as a topic. And to my recollection, no, nobody brought that up. But since the proteins are involved, and proteins are the building blocks of the cells, as we taught, and the membranes are part of it, and you see that, you know, they're also involved, and why not look at that? But the membrane the cell membrane is a very dynamic entity that does a lot of things, and that allows certain things to come in selectively, that allows across, there's an osmosis, and there's a lot of things happening, the receptors and whatnot, and what role these do play, metabolically, especially, this is a very important area in my opinion, and especially the cells are metabolically active but reproductively dead. So,
there is another a concern for which we don't have solution. Are there questions from younger colleagues? Otherwise, we can continue this discussion because these are important.
Yes.
Viva, do we have any question from younger colleagues?
No.
All right. So I think another point which worries me all the time, that we start an area of research with a lot of enthusiasm. No, I'm sure you all agree that for 33 decades and 4 decades, people worked on the telomeres as senescence markers, cancer development, lot of things happened, and $1 million, $1 trillion research funding has gone in the telomeres. We got the papers in Nature, Cell, PNS loaded with these areas. Eventually that has died down. You don't have any marker coming out from telomeres, etc., whether you can use it. Like that, you use a word microRNA. I'm actually I'm afraid that that microRNA could be a hot area for fetching lot of funds and things for growing the science and getting good quality publication. But that will lead to a marker, etc. One needs to be. I mean, the worry is there that why some field is growing so fast and then dying down, almost same speed. What could be the reason for many of us actually to get a comment on that?
I don't know. But everything has a life cycle. You look at your iPhone, right? You got an iPhone 4 and you have something. The technology has a life cycle, and I think the field also has a life cycle. Some of these things are people get excited, and a group get excited, and takes a direction. But I think what is fundamental, in my opinion, is at least the quality and scientific rigor is necessary. Some of the lot of academic labs are free to do what they think is right to do. Not that intentionally there's a built-in effort to mislead the public. It is not the case. But lack of scientific rigor, sometimes people do get excited and take the science in a field, and eventually it doesn't get what it needs to. We have to be cautious. We have to be cautious and look at the literature very carefully, and I wouldn't say selectively. Look at everything that gets published in a field, and at an individual, at my level or at your level, it is not possible. But for the students, it is possible. That's why we divide the field into smaller areas and have the students, have the postdocs, get into the depth of it, and come back and tell And look, this is not probably, I'm not convinced that this is the way to go. And this is perhaps the way to go, and come up with a solution for these kind of things. Superficially, based on my very broad understanding of the landscape, and I think looks that microRNAs may be anything that is amenable non-invasively, including imaging, as Dr. Dwarkanath was pointing out, is the way to go, because we know that there is a temporal spatial heterogeneity, and the only way that you can overcome is, not that the tissue banks and biobanks are useless, don't get me wrong, they have the value, but how you use these things are more important. It is a tool. How you use the tool, how you use a model, that is more important than
right. I think our hanging has paid a good dividend. We got 2 colleagues. They are having question now for you. So I can go to Palak. Palak, can you ask question now straight to Pat?
Yes. Good evening, sir. I have a very basic question. I wanted to know, what is TCD50 and how is it correlated with tumor growth delay? And what is the basic function which I can use to calculate TCD50?
TCD50, I think you have to ask your mentor. It is a very specific question about tumor control. What is it, TCD?
Tumor
TCD,
control dose.
Tumor control dose.
Tumor control dose, yeah. I think it is better you ask your mentor and get more information than I can try to answer it, but I'm afraid that I will have to get back to you on that, because there are a lot of issues how people do it. Okay, so is the methodology, has it been harmonized? What is a harmonized methodology, right? How long you will grow the animal? How many cells you inject? Is it a xenograft model? Is it a orthotopic xenograft model? Is it a PDX model? What is the model that you are using? And all these things will, will be somewhat contextual, and I don't want to state here something.
I think
give you an impression that
I think this question Dr. Dwarkanath can take it in case, or Dr. Thullar. Since there is a question from audience, let's satisfy mentors can do.
Yeah,
so anyone.
This, as I said, is an integral part of what I'll be discussing. As Dr. Prasanna rightly mentioned, instead of occupying time here and verbally answering, probably with the help of a slide. So keep this question.
Very good.
The student, I don't know who the person is. We'll discuss this tomorrow.
Sir, I have one more question. Is there any possibility that a molecule can act as both radioprotector and a radiosensitizer?
Yeah, it is possible, but do we know all the differences? What are the hallmarks of cancer, and how is it different from the normal tissue? And what are the molecular pathways that makes a normal tissue take a path towards becoming a cancerous tissue or a cancerous cell? Understanding these things are fundamental to understand and exploit those differences so that you can come up with a radiosensitizer for the tumor and that can also act as a radioprotector. Is there one? Yeah, there was a lot of noise, especially as our respected chairman mentioned earlier, WR 2721. It became so debatable, Dwarkanath is laughing at it. And it became so debatable. And it was a radioprotector as well as a radiosensitizer. There are a few natural compounds, possibly yes, theoretically yes, but at what dose? At what dose? And what is the dose that is necessary for it to act as a radiosensitizer for the tumor? That dose may be different for the normal tissue to serve as a protector. So there are possibilities, and the sequencing, not only the dose, when do you administer it as a radiosensitizer and a radioprotector. So it is a complex area, but if you can solve that problem, it is great. Go after it, and that is the kind of work. The higher the bar in terms of challenge, the higher the reward is.
Yeah, let's go and the last question. Mr. Vikas Maurya, you can ask your question directly to Pat.
Hello.
Yes.
Actually, my question was how a molecule, if it is a radioprotector, then specifically goes into the nearby cell, not to the tumor cells. How it is differentiate between these two cells?
Yeah, again, exploit some of those molecular pathways. For example, you don't have the targeted therapeutics, right? Some of the targets are only available in the tumor, which are not available in the normal tissue. So some of the targets are only available in the normal tissue, might not be available in the tumor. But it is not 100%. There is a distribution. For example, EGFR receptors. Okay, EGFR receptors. There's a heterogeneity between tumor and normal tissue. EGFR receptors are also there in the normal tissue too, but they are more in the tumor. So it becomes again the dosing issue, again the dosing and scheduling issue. So how do we exploit these things? How do you to conjugate drugs to a protein that targets a certain receptor? So there's an active area. Dr. Sunil Advani, for example, he is in one of our grantees in the University of California in San Diego. He does what is known as ADCs, antibody drug conjugates. All his career so far, all his publications, high impact publications, are all in ADCs. It's a very active area of research. Can you deliver the drug specifically to the tumor? There's tumor vasculature issue, the hypoxic core where senescent cells or the cancer stem cells reside are not accessible for the drugs, but we can get them with radiation. So see, there is a benefit of radiation. So but how do you get the radiosensitizer to the tumor core when there is poor vasculature and poor blood supply? These are the issues. I think it's a lot of work is necessary, and I think philosophically speaking, what we have understood with our limited abilities and knowledge is very little. And there is so much to understand, even with our limited abilities.
Thank you. Thank you so much. Let me hand over this responsibility to Dr. Sanjay Thullar to conclude this session, and then followed by Mr. Vibha Tandon.
So, Dr. Dwarkanath, I think we have to have an offline conversation sometime to get into the precision radiation oncology, and we will do that sometime in the near future.
Looks like
can we do accurate medicine? Even I wanted that discussion. We have few minutes if our chairpersons allow.
Yeah, we can go ahead. No problem.
So if we can have that discussion.
Yeah.
You have few minutes.
So yeah, I was actually excited by Phil Lambin's work in Holland, wherein he's bringing up a novel concept called predictive radiation oncology. In terms of accurate precision radiation medicine, where we are in a stage is, in some organ sites, in the treatment of some organ sites like head and neck cancers, we are able to cohortize the HPV positive and HPV negatives, and HPV negatives are being more radio-resistant compared to HPV positives. But are you able to de-escalate the radiation dose to an HPV positive cohort. Studies are underway, the clinical trials are underway. So that is where the buck stops, right? In terms of precision individualization, there is a magnitude of work that needs to be done. That magnitude of work will require understanding the dose effect response relationships for radiation. At low doses, can you promote the immune modulation in certain individuals? Who are those individuals? Who have that ability to provide the optimal immune response to get rid of their tumors? That is a line of area that we could take. In terms of accurate precision medicine, do we have enough of data set from which we can understand via, say, machine learning or artificial intelligence, this patient characteristics with this size of tumor, with this amount of oxygen status with this amount of hypoxia and this amount of whatnot. And this was the outcome for this patient under these treatment conditions, which involved this. So we are treating, what, about 7 million cancer patients every year. So pretty much in theory, I'm laughing, we have 7 million patients data, right? If it is an iPhone, if a patient is an iPhone, we get that kind of data coming from every patient. So we may be better positioned if we do the analysis of that amount of data. But again, in terms of AI, to use AI and bring in the precision radiation medicine concept, you need the volume of data, the quality of data, and what type of data and type of data. And these three things are very important to bring in AI. Otherwise you will end up in garbage in, garbage out type of scenario. So those are the two things that comes to my mind, maybe few other. I'm sure that you also have a lot of ideas in how can we bring in precision medicine.
Just for one more thing. So I have seen a couple of, projects or maybe one or two publications where they are doing the imaging, and they're collecting the data of imaging and connecting it with the AI, and they're trying to say that although it is not directly related to the radiation, but by creating such imaging data bank and reading it through the machine learning using artificial intelligence, they are trying to predict that this is a particular kind of a tumor. It can be treated, it cannot be treated, it can be treated by this kind of these kind of chemotherapeutic agents are required, they respond to this. So such kind of projects are there, and so is it like that? Is it really, have you seen such projects there also, or there is some success rate and that can be utilized for radiation also?
Yeah, I think Phil Lambin is the person who has done a lot of work. He published quite a number of papers in this area. I believe one of the Nature articles. Phil Lambin, Phil Lambin, Lambin. He's in, I think he's in, he's in Holland. In Amsterdam area. So it is possible. It is possible, but again, the underlying issues are, what is the data, the imaging data, how good is the data, and how much data you need, and the quality of the data, type, quality, and the amount of data. Those three are very important. And you can go wrong in AI big way, okay? I'm bringing this to a conversation mode, from a discussion mode. I was surprised when Nadal won the Australian Open against Medvedev. After after 2 sets down, in the third set, one break down, and in a 30-40, about including the IBM Watson, including all the AI pundits, and everybody predicted 95 to 96, 97% Medvedev is going to win. But that did not happen. Okay. So what went wrong there was, the type of data that did not include one or two particular aspects of Nadal. So this is possible in our field also. So this is the conversation mode. That's what can we learn from other fields and how well we can adopt it to our field. It is the type and amount and the quality of data.
So thank you very much, Dr. Prasanna, to be here, and thanks to Dr. Dwarkanath and all participants who are sitting here from 9:30 till evening 6 o'clock, and especially Dr. Prasanna. And over to both the chairpersons, Dr. Tulkar and Hari Mishra. They are also here from 1 o'clock. So I would like to thank both of you. And now you both can just conclude the session.
Yeah, thank you very much. It was a very nice session, and we thoroughly enjoyed it. And both the speakers were very expert and learned in their field. And I hand over to my co-chair, Professor Mishra, to formally conclude the session.
I thought you will do that, but anyway, back to me. Thank you so much. I think both lectures we had very nice one. And as you said that both are experts, no doubt. We enjoyed it. Colleagues also enjoyed it. And we hope that in future we'll keep interacting. There is a question already raised by Professor Tandon to my earlier speaker, Prashanta, that what is the area in which he is working, so that some collaboration interaction can be created. And similar kind of thing, probably we'll see that some of us will contact to Professor Prasanna also and see that if some, if our science in this area of India, what we are doing, if that can be advanced with their help, we'll be grateful to both of you. I think with this, I would like to thank to all of you and Professor Vibha Tandon for giving this opportunity to both of us for chairing the session and actually enjoying, though I am in the Department of Atomic Energy for last 32 years, but I learned a lot of things in both these talks today, which probably might have not been possible without hearing and chairing and being around for 4-5 hours. Thank you so much.
Thank you. It's a long day for you, and I enjoyed the meeting as much as you did. Thank you so much.
Thank you, Dr. Prasanna.
Thank you.
We'll stay in touch, maybe
we'll stay in touch,
some other context, and have a nice Sunday.
You too, for the rest of the Sunday. Thanks.
Okay, thank you. Thank you, everybody, and we'll be back tomorrow at 9:30. Thank you, everybody. Thanks.
Bye.
Okay, thank you.